database. I'm not going to say it's a huge or voluminous database available with us, but it's just started. The reporting has been started now. Earlier, when the program was launched in 2015, there were a lot of issues in getting the data because of administrative issues. The National Coordinating Center was Richitra Tirunal Institute of Medical Sciences, Thiruvananthapuram, but unfortunately, due to administrative and various reasons, it could not pick up. The reporting were low, but once again the IPC took as a charge as a National Coordinating Center for Material Vigilance, the gradually reporting is increasing. Similarly, the centers are also reporting. But here we cannot always compare with the reports with the drugs, because there are certain reasons which I would like to highlight here. The reason is, this is one of the best example I would say why the device related adverse events reporting are comprehensive or complex in nature. Recently we have come across some of the examples, some of the issues related to intraocular lens related adverse events. So we have received some reports, around 13 reports related to TAS, that is toxic anterior segment syndrome related to intraocular lenses. So then it is related to the product quality actually, because the intraocular lenses are made up of polypropylene, acrylic, silicone, hydrophobic or hydrophilic acrylic or hydrogel. But when we, the reports are put under the causality assessment, there were something related to the quality related issue, lens quality related, but then we thought it should be discussed with CDSU. Then immediately the experts committee of CDSU and FDPA discussed together, and they have recommended that these samples, suspected samples, should be sent to the CDSU testing laboratory to get the quality data. So this is how the assessment related to lenses or medical devices are concerned as compared to the devices. And another example, the adverse event related to orthopedic implants, the commonly reported as metallosis. But we do have some reports related to metallosis, but the information we received is not up to the mark. Say this is related to the quality of metal used or the metal leaching. Generally, the orthopedic implants are prepared or made up of by using the metals like titanium, cobalt, chromium or nickel. If the quality of the metals used in the devices, and the quality doesn't comply with the ISO standard or BAE standard, that is Bureau of Indian Standard, or any other pharmacopoeial standards, there will be a chance of metal leaching. Then the patient end up with some inflammation or swelling. Then it needs to be diagnosed either through metal ion testing, cobalt or nickel or cadmium level needs to be established or estimated by using the ICP-MS, that is inductively coupled mass spectrophotometer, or we can go for soft tissue imaging. Then only the causality. So in order to do the causality or in order to confirm the root cause, we require this kind of complete information, and then only the adverse events confirmed are coded as metallosis or associated with orthopedic implant. So it's a comprehensive process. And then who are the stakeholders which program needs to connect or needs to integrate with various stakeholders? Of course, doctors and nurses are reporting quite a good number as compared to other healthcare professionals, but there is a lot of need, to address the issues among the pharmacists, because pharmacists are the one they can able to handle the medical devices of implants where the drug device combination is involved, and the biomedical engineers, of course, the adverse events monitoring and reporting related to the machines like X-ray machine or MRI scan, pacemaker. There are so many devices, you might have seen the class C and class D. So here there are another issues like instrument calibration or instrument maintenance. These all are needs to be accounted while doing the causality assessment. So therefore the biomedical engineer or if you look at other countries, that is called, nomenclature is known as clinical engineers. Their role is very much important in monitoring the devices. But wherever the drug device combination comes up, or the devices related to polymer based or chemical based, then naturally the pharmacist or nurses also can provide their inputs. Of course, doctors are the primary stakeholders for every device are concerned. But here, unlike drugs, in devices, the reporting depends upon the, depends upon the product to product. So, for example, ocular implants, then we will have a different.
Recording in progress.
There are few examples, and some of the examples are already taken, action being taken by CDSU, and the CDSU instructed the concerned manufacturer to include this information in the IFU, that is information brochure for the user. So, and another interesting fact which I would like to share with you that, recently the Secretary Health, Ministry of Health and Family Welfare, Government of India, written a letter to all state and union territory governments to ensure the effective implementation of MBPI. Subsequent to the letter from the Secretary Health, Government of India, there are 4 state government taken action while enforcing or while implementing the establishment of MBPI in their respective hospitals or medical colleges. States like Kerala already started this processing, and we have received a letter of intent even from the Taluk level hospital from Kerala to function in the material vigilance program, and they are also reporting started. Similarly, union territory like Puducherry, Tamil Nadu, Karnataka also issued an order to various hospitals of their states, and currently we are working with the state government of Odisha for implementing this program, and I think the AIIMS Rishikesh also took an initiative to facilitate this process at the level of Secretary, Government of Uttarakhand. I am thankful to the AIIMS Rishikesh team as well. I am sure the months to come, in a couple of months, Uttarakhand also may issue the same kind of order for their hospitals to participate and report the adverse events related to medical devices. And this is about the in vitro diagnostic. Since the in vitro diagnostic kits also notified or considered as a medical devices are approved by the Central Licensing Authority, we need to focus on monitoring the safety of medical devices related to in vitro diagnostics. But here the adverse events reporting just being started, and there are adverse events are different as compared to other products. Here we can expect false positive, false negative. This is related to most of the in vitro diagnostic kits, and then error in the reading. This is a common in glucometer or pulse oximeter. Then malfunction. It is also again applicable to all other devices. Then another adverse event or issue or incidence is disappearance of control or test line. This is the kind of adverse events reported with pregnancy kit so far our experience says. And it poses to huge health burden and economic burden to the patients if in vitro diagnostic kits are not doing well, or if we found any incidents with the in vitro diagnostic kits, it leads to socio-economic burden, and then interaction with the healthcare system, and of course medication burden, lifestyle changes, So these are the burdens to patients, and therefore we are equally important and equally committed to monitor the safety of in vitro diagnostic kits. So according to our experiences, since in vitro diagnostic kits are also notified as medical devices, now there is a need to identify or enroll in vitro diagnostic labs or testing labs also as one of the center. Accordingly, the letter of intent or enrollment form of MBPA also being revised. We must include the in vitro diagnostic kits labs also, and we have trained around 30 labs also for monitoring or to function as a material vigilance center. And then another the interesting fact which we found in the PubMed or ResearchGate and other search engines, saying that India is one of the country leading in the scientific articles or publications related to medical devices safety or even the material vigilance. There are a lot of studies are going on or published, by bringing or by creating the awareness among the healthcare professionals. The studies are related to knowledge, attitude and practice of doctors about the medical device safety monitoring, or the knowledge, attitude, practice-based study among the nurses about their level of understanding or awareness about the medical devices safety. And of course, some of the case studies are also found available. These are the evidence are very important in terms of vigilance, any vigilance, let it be pharmacovigilance or hemovigilance. We need to document the incidents. So currently, India is one of the leading country in having the article or evidence is available with medical vigilance. And this is, of course, this is challenging, I would say. So far, we have seen how the program is being establishing or running, how we are coordinating with different stakeholders. But this is very challenging one, and this is the take-home message for all of us. Left-hand side, if you look at the figure, shows the devices, list of devices are notified or approved by the Central Licensing Authority, that is CDSU. Say to name few, if you look at the total number of devices notified or regulated under the gastroenterology, there are 163 class of type of devices are being regulated. But if you look at the reporting part, right hand side, you can see the current reporting practice at MBPA. There is very few reports are available, or even in fact I would say the reports are not available for most of the devices which are notified by against the notification. Again, if you look at the software which are used in the diagnosis or treatment, again considered to be a medical devices, but here the reporting is not yet. Similarly, there are number of devices are being approved in the physiotherapy or occupational therapy, again the reporting is not there. So here the challenge for all of us, whether pharmacovigilance center or material vigilance center or the entire stakeholders, our responsibilities lies with, we have to connect with different stakeholders including physiotherapists. So far we have not addressed the issues among the physiotherapists or occupational therapists. The volume of physiotherapists available in the country is huge. There are a number of devices are approved under the physiotherapy, but still the reporting is not there. So I think this would be one of the challenge and the way forward for all of us. And another would be the practice of material vigilance should start or should evolve from the primary healthcare level also, as some of the devices are being used even at the primary healthcare. So currently the system, our focus is to establish a center or the unit at super specialty or multi-specialty hospital only. But if you look at the primary health center level, number of devices are being even used in the primary health center level. Those devices are notified as Class A devices. So thanks a lot. I have completed my talk. Over to the organizers.
Thank you, Dr. Kalaiselvan. Are we audible?
Yes, madam, you are audible.
Thank you so much for giving us such a comprehensive overview of the material vigilance program in existence that you have helped establish, and you are helping it establish further. And highlighting the issues which are in the program of pharmacovigilance, which make it different from the from material vigilance, you know, especially the complexity of reporting and establishment of causality. You highlighted the multidisciplinary nature of the team that would be needed in causality assessment, which is there in pharmacovigilance, but in material vigilance, you've highlighted the role of pharmacists who would help us better in understanding purity of the materials involved, etc. The role of biochemical, biomedical engineers. And so that is a very, very important thing for us to realize, because in the medical institutes, we are still struggling with pharmacovigilance. So material vigilance, to understand causality assessment in itself is going to be a big task. The other thing that makes us feel good with what you've told us is about the reporting of publications, as regard India leading in publications in the area of material vigilance, which is a very, very feel-good factor. I'm sure you're feeling wonderful about it. 60% publications globally reported is by the Indian setup is a wonderful achievement in the, you know, in just, I think, in about 5 years. 2015, I think the NVPI began, and 2017 and 2020, Government of India has formulated the medical device rules and regulations. Now, before, you know, I'm very sure the audience would have some questions for you, because you've spoken on such a relevant topic. So if there are any questions, Dr. Kalaiselvan would be very, very happy to take. Are there any questions? Yeah. Dr. Jitendra?
Hello, good morning, Dr. Kalaiselvam. Am I audible?
Yes, ma'am, you are audible. Namaste, madam.
Yeah, good morning, namaste. Very nice to hear you. Thank you for that talk. I have a very, very rudimentary question. I hope you're not going to laugh at me for asking that. What I want to know is, if a medical device has an additional drug, like a drug-eluting stent, so do we classify that as a device or a drug, and where should the reports go to?
Actually, drug device combination, drug-eluting stent also comes under the class D medical device, high-risk device.
Okay. So,
And currently we have exactly, I don't know, currently approximately I would say around 100 reports are available for the year 22 and 23 related to drug device, cardiac stent related adverse events.
okay, so even if that adverse event is actually because of the drug, should we be also reporting
No.
to pharmacovigilance? No. Or we could
No,
do
actually,
that.
you are asking whether it is causality is related to the stent or not.
Yeah. You know, like it could be tacrolimus eluting stent, and the problem is the tacrolimus, not the metallic part or any other part of the stent itself. So is it a pharmacovigilance problem or a material vigilance, report?
Ma'am, actually, when these reports, when these findings were submitted to CDSCO, in CDSCO, the causality assessment, the subject experts committee of drug and device both were seated together and they were analyzed. But majority of the device, the drug device combination, found to be the error in the procedure error. What concluded the terminology causality, it is not certain or probable here.
Okay.
It is related to the procedure error. So this is what the causality outcome or the report's analysis outcome says.
Okay. Okay. Thank you so much.
Sir, I have a question, sir.
Yes, sir. Please identify yourself.
Dr. Jagdeep Singh from GMC Chandigarh.
Ji.
Sir, thank you for a nice talk. It was quite revealing for us. So I want to just ask regarding the reporting of SAEs by these material vigilance program. Is it the same as it is for reporting SAEs in our routine?
Criteria remains same, Dr. Saab, the same criteria, seriousness and non-seriousness, and again the seriousness, death, hospitalization, congenital anomaly, the same criteria. Criteria remains changed, and even in the medical device rule also the same criteria is given.
Sir, do we then, I just wonder that how we label something as SAE, for example, most of these type C and D type of instruments or whatever device you are told. Because in that, they will be hospitalized first of all for undergoing such type of procedures and so on. And there's no chance of congenital type of abnormalities, mostly with these.
But see, that is what here the criteria differ or changes devices to device. When you ask MRI machine, there is no chance of, I don't know whether congenital anomaly may be possible or not, but we have to apply our logical practice and logical thinking. Then the defined criteria changes product to product.
So regarding some of the orthopedic, commonly used orthopedic processes, are they all included in that rheumatology part you have just told?
Yes, some of them are in the rheumatology, some of them are in the physiotherapy, some of the orthopedic implants are in the physio also.
So because most commonly what we see that people after stents, I think most common we have are the orthopedic implants. So they are mostly in class C, is it sir? Class C?
Class D.
They are all in class D. So
Yes.
they are the most severe type of, you can say.
They are all under high risk devices.
Right. So I was wondering, was as part of ethics committee, how do we go with that? So what we receive first of all, now we're receiving some trials for these devices also. And then secondly, how to go about that, sir? Just
Ethics committee point of view, I don't have any anything to comment on the ethics committee because I am not into the hospital setting practice. But I am sure somebody will help me. The chairperson or co-chair can help us how the ethical issues to be handled related to the device.
Thank you, sir. Thanks a lot for me.
Thank you.
Just to answer what I think what the question was, ethics in related to implants and all would come in when before the implant is put by the doctors, the consent of the patient is always taken. There is an informed consent form which the patients, in which the patients are informed about what procedure the doctors are going to do, what is going to be put, and what all can happen. So far, I don't think that this informed consent is very elaborate. The clinical informed consent, unlike the informed consent we have for clinical research. I don't know, Dr. Jagdeep, whether your question was in relation to research or clinical practice.
Yeah, it was related to how we will handle when we get such type of proposals to oversee this part of ethics.
Yeah, so no, in ethics, the patient information sheet or the subject information sheet will be even more elaborate. All details about the device, the material would have to be given, the quality standards, whether it's fulfilling the ISO mark, etc. It will be even more elaborate than what you have for clinical trials involving just drugs. More detailed.
Yes, again, ma'am. So,
yes.
So we'll have to devise some new ICFs, I suppose, and PIES forms. So we have to devise newer ones.
More detailing, and for which even the doctors and the research, the people evaluating the protocol would need to be aware of what is needed. Material quality, sensitivity, allergy. These are things with which creed terms. It's because it's not been handled. So yes, your patient information sheet will have to be very, very elaborate.
Thanks a lot, ma'am.
Are there any more questions? Okay, just before we conclude, Dr. Kalaiselvan, can you just tell us, since many of the issues related to material vigilance, the reports you're getting, maybe related to quality issues of the devices. So are there any penalties? Manufacturer is not fulfilling the quality assurance standards for which the device has been approved. Are there any penalties put into the Act?
Penalty, I don't know penalty, but CDSO regulators also, they have a lot of plan and activities to improve the quality related issues like recognizing through QCI. They are doing a lot of activities like ensuring the ISO standards or BIS standards. Then recognizing more number of laboratories for testing, because that is another major challenge. Currently, we have only limited number of CDSO notified laboratories for testing of the medical devices. Let it be orthopedic, cataract related, lens related testing laboratories or glaucoma devices. So this is one thing. They are coming up with more notified laboratories to test the quality related issues, and with the help of QCI, there is a program called certification bodies, identification of certification bodies to ensure the quality of medical devices. So the regular inspection or audit can be done to ensure the quality of the products. So there are lots of planning. I'm sure the years to come there will be a lot of capacity building and schemes to ensure the quality of medical devices as well. Since the adverse events reported so far, most of the device related issues are procedure error or quality related, or there are some issues the device meant to be used for single use only, but knowingly or unknowingly the devices are used multiple times. Then it is reported as device breakage. So there are a lot of issues. I agree with you. The quality is one of the main concern and the adverse events related to medical devices.
Thank you so much, Dr. Kalaiselvan. I think.
Thank you, Dr. Kalaiselvan, for your very informative lecture. Regarding Dr. Jagjeet Singh, I would also like to add in relation to ethics. We also discussed the same thing in our ethical committee in our institute. So we found that class A and class B usually do not require any ethical clearance, but for class C and class D, and as Dr. Bandha has mentioned, because we take the informed consent from all the patients whenever we have some implants. So for class C and class D, there is informed consent. Otherwise, for example, we have one PhD student, and he is doing some prospective studies in relation to the adverse events of the cardiovascular medical devices. So he's already taken a permission from the ethical committee, and ethical committee has given a permission only whenever he finds any untoward reaction or adverse event, he needs to report to the ethical committee. So for research purpose, we should have an ethical committee clearance. Otherwise, for class C and class D, informed consent is enough. So thank you once again, Dr. Kalaiselvan, for enlightening us about the about your program which you are running. And I found 2 or 3 terms very new. For example, whenever you said clinical engineer, so it is new term for me in place of biomedical engineer. So once again, on behalf of the organizing committee and on behalf of the Society of Pharmacovigilance, I thank you for accepting our invitation and delivered such a nice talk. Thank you so much.
Thank you so much.
So we move on to another lecture, and I hope Dr. Joan D'Souza is already online.
Sir, Dr. Joan D'Souza, she is not able to join right now. So we'll welcome
No, she's already online, I think.
Oh.
But, huh?
Yes, I am online.
Yes,
Okay,
all right. Thank you so much.
Okay, ma'am. Okay, ma'am.
Dr. Joan, thank you so much for joining from... okay, so it's my privilege to introduce Dr. Joan as the next speaker. She is basically working again on the medical devices, and she is pharmacovigilance consultant and regulatory medical writer. And good thing is she is very closely associated with International Society of Pharmacovigilance as chair for Switzerland and Austria, and the EMWA member and the team PRRC member. So she is very active and has already published many papers, especially in Drug Safety Journal, which is an official journal of the ISOP and EMWA journal. And her major achievements are she is a Juris Doctorate licensed to practice law in the state of Minnesota, USA, and the Dean's List of Academic Excellence Awards, which she has received for clinical research, University of Cincinnati, USA. So I once again welcome you, Dr. Joan, and request you to please start your lecture.
Thank you so much. I have to share my screen. Can you give me the rights, please? Since you cannot share. Okay. Thank you. So it was a bit early for me. I'm sorry. I thought it was 10 o'clock my time, and you're actually in Europe right now. So sorry. I will put it in my slideshow mode. Okay. Can you see my screen?
Yes, we can see.
Hello, can you answer please? Can you see my screen or no?
It's visible.
Okay. But I want to just put it in slideshow mode, so then from the beginning, yeah. Okay, can you see now? Okay.
We can, it's visible.
Okay, so so I'm going to talk about today post-marketing safety surveillance. Actually, we'll focus on that. And my topic is on drug device combination products, and that is the area that I really want to receive my expertise. And to the overview of today's presentation is like we are going to talk about key definitions of drug device combination products in the EU and the US market. And we are going to give an overview of the current laws and regulations and guidelines for combination products, classifications of drug device combinations in the EU and US market. And we are also going to talk about the similarities and differences in drug device regulations in the EU and the US. Next, we will talk about what are the certain challenges in actually globally harmonizing drug device regulations. And we are going to then focus on pharmacovigilance strategy, which is based on current regulatory requirements. Okay. So we know what are medicinal product and medicinal devices as well. Medicinal products are basically when we use certain products in order to diagnose. And can you just give me 1 second, please? 1 second, because the reason is I have to. Can anybody tell me how to see the notes? Actually, I don't want to read the notes while I am. I don't know what is the problem with my computer.
Ma'am, here we can. Ma'am, here we can see your slide in full screen mode.
No problem. Please go ahead.
Hello.
You're audible, Dr. Joan. We can see your screen and the notes.
Can you hear me, everyone?
Yeah, very clearly.
Yes, ma'am.
There's no sound at all coming from anybody.
Hello.
Yes, ma'am.
Can you hear us? Is she mute? No.
Hello.
Hello, can you hear us? Yeah, but she needs to know that, we're going to do this. But she can't hear us. That is the problem. Is there any way of contacting the speaker?
Ma'am.
On the phone or on a WhatsApp and tell her that we give her time to get her material in order while we can request the other speaker to come on board. They'll have to do it, sir.
Okay, I think Syed sir can do it for us. So has her contact number.
So I can request Dr. Joan to be the next speaker, na? And in the meantime she
Yes,
can,
sir.
so I can write to her. Okay.
Who's the third speaker?
It's Nusrat Shafiq ma'am.
It's ready or not?
Yeah, please confirm.
So can anybody get in touch with Dr. Nusrat? Okay.
Can you please unmute ma'am?
But she must be on mute and all, na? If she can.
Yeah, can you please unmute Professor Nusrat Shafiq
Dr.
ma'am?
Nusrat? Dr. Nusrat? Okay, can we have your are you okay if we have your presentation now?
So I think I have shared my screen, ma'am. I hope the organizers can state.
Thank you so much. Yes, we can see you, and we've got in touch with Dr. Joan. I think we need to inform her before we begin the next.
That's okay. Okay, she's already online, so we can start, ask Dr. Nusrat Shafiq to start her presentation. Okay. So, Dr. Nusrat Shafiq, you're most welcome. And on behalf of the organizing committee, and again from the society, I welcome you. And we would love to hear you. So before your presentation, I would love to introduce you as a speaker. Dr. Nusrat Shafiq is a professor working in the PGIMR Chandigarh, and she has already published more than 200 papers. A very active person and involved in many activities, and she has expertise in clinical pharmacokinetics, pharmacodynamics, bioavailability and pharmacokinetic modeling, and also clinical trials of pharmaceuticals. So I request Dr. Nusrat Shafiq to please start your lecture.
Thank you, sir. Thank you for the kind introduction. But I think I would like to add, because antimicrobial stewardship has been something which has been my forte for the last many years, and is also the topic for today's discussion. So, so that's what I would consider myself to be really working in. And having said that, I would like to thank you. This is the first time I'm finding myself in a conference on pharmacovigilance. I have landed up in all kinds of other subjects, but pharmacovigilance, this is my first time. So I hope I stay relevant and talk pertinent things as was expected of me, because I myself took some time to decipher why and how of things. So in any case, thank you everyone for inviting me to this wonderful sequence of deliberations. I happened to hear a bit of the previous talk, and I really enjoyed it. So okay, with that, having said that, the topic that was given to me was antimicrobial resistance, documenting failure of therapy, and a needful new vertical. So I'll be tweaking it a little, and I hope the chairpersons would be okay with this. But I'd like to, with having the broad topic in mind, points I will waver a little with your due permission, sir. So I hope I have that from you.
Go ahead, Dr. Nusrat.
Thank you. Thank you. So the slide, can someone? Yeah. So I think a lot of you would be familiar with this annual report from our. This is an annual exercise that they do. They have a big network of microbiology labs, and where they collect from tertiary centers. This is important. It is largely from tertiary care centers, though they are increasingly adding other care centers as well. They do an analysis of the resistance patterns in the microorganisms that are isolated in these facilities, and the situation is quite formidable. Formidable in the sense that if we just took out the example of two very common organisms which affect either us in some ways, or our relatives, or people around us. One is the E. coli, and the other is Salmonella typhi. We can see that from these data here, we can appreciate that the situation is quite scary. In fact, E. coli, only 60% of the isolates would be susceptible to meropenem, and meropenem is one of the reserved category drugs, so to say. So this makes things very difficult for us to handle. This reflects on the way we treat patients, the way their outcomes play out in subsequent to appropriate use of antimicrobials. Similar is the case with the Salmonella typhi. Again, we see that for some of the drugs to which they were very conveniently susceptible, ciprofloxacin was one of them, the susceptibility has been reduced dramatically, and this was before azithromycin became very commonly used for during COVID pandemic. We don't have the data following that. I mean, I'm quite sure the results are not going to be very encouraging. This is an example of some of the organisms which have not been affected all that badly. If we just go a step beyond these organisms, the things are even more scary. We just have something like polymyxins to which they are susceptible, and they show resistance to this. So basically, that was just to emphasize upon the point that AMR is a big problem, and it is not without reason that it has been identified as one of the global threats. So that is a big designation, and that's why all the attention that it is getting currently. So, I mean, how does, as pharmacologists, how does it affect? It gets reflected in the way we use our antimicrobials, and if we just see some of these evidence, which are global evidence, we see that our use of access and watch antibiotics, our use of watch antibiotics is increasing, and access antibiotics is decreasing by the day. That is a reflection, and in fact, it is a kind of an indirect measure of the situation. This is not a direct measure of antimicrobial resistance, but an indirect and important measure. In fact, it is a very important contributor to the current situation that we are facing. It would be desirable to have access use increase and watch use decrease. But the scenario we have at the moment, especially in the lower and middle income countries, is just the reverse. So the situation is quite grim. And if we come closer home, we see that we carried out this study just 2 years ago, just prior to the pandemic, and we did it with 5 centers within our country, and we realized the situation is, data reflects quite a similar pattern. We have an abundance of multi-drug-resistant organisms. Our use of watch and reserve antibiotics is quite high. We have a considerable amount of hospital-acquired infections about which we need to be worried about. So all this is just to show that, yes, it is indeed a very important issue. When we talk about global data or the national data, we really don't understand the impact of it. In order to understand the impact of it, I will try to bring out some real-time stories. We all of us have statistical meet and mortality meet in our hospitals. For the last 4 to 5 years, I've been going there pretty regularly for the medical stream, and what is noticeable, and I'm pretty sure that would be the situation in many of your settings, a patient comes with an ailment which is very different from an infective condition, but the way the patient goes is usually because of sepsis or septic shock, and that too because of some kind of infection that the patient would have acquired in the hospital. For something like, somebody would come with acute coronary syndrome and would end up with sepsis and septic shock. So that is the situation. Our medical management has improved considerably, but our outcomes perhaps are getting affected because of him. We have this facility for clinical pharmacology consultants, SR, to be on call for clinical queries which require bedside visit. And I just very cursorily reviewed all the details of all the calls that were responded to, and more than 80% of these calls were for issues related to management of difficult to treat infection. And this is just to tell how at our own levels it is affecting. The hospital stays are increasing because the primary condition gets cured, but it is the subsequent infection that a patient acquires leads to increased hospital stay. If some of you who would be working in this area as part of our antimicrobial stewardship activities would have realized that indications for which people are put on prophylaxis in the hospital settings, even in secondary care, are increasing by the day. You want to ask, you go ahead and you ask, why is the patient on antibiotic? They say, the patient is in the hospital, and that's a good enough reason for us to keep it. patient on antibiotics. Patient is receiving IV infusion, and that's why we're scared we're keeping the patient on antibiotics. So these are some of the indirect ways we understand how is AMR affecting us and others on a day-to-day basis. The profile of community-acquired infections is changing. Some time ago, I think around 4 to 5 years ago, there was very indigenous data which suggested that patients coming to emergency settings with suspected staph infections had a very high percentage of methicillin-resistant Staph aureus. Mind you, this is not something which is very native in nature. It was there globally. There, gram-positive infections and resistant gram-positive infections was a big problem. But over the period of time, with a lot of multi-pronged activities, they have been able to bring down all these things. And we are trying to achieve that kind of a situation. So basically, what is there in our hand? We can perhaps carry out antimicrobial stewardship as an intervention. And if we do antimicrobial stewardship, these are some of the things that we try to achieve. Some of the immediate things that we try to achieve are in terms of making the antimicrobial use more rational. Some of the little distant things are we help in better management of infections which occur out of hospital-acquired infections. To some extent, we are able to affect the cause, the side effect profile, duration of stay, and some data to suggest. We have this data from our trauma center where the mortality and patient outcomes have also got affected. But at this point, it is very important to understand this is just as much as stewardship can achieve. If we want to have better benchmark achievements, it is the infection control and prevention which will make the difference. So when we want to impact AMR will be impacted with integration of stewardship practices and infection control practices. So since I predominantly work for stewardship, which sometimes also join hands with infection control, and we think that our antimicrobial use is not going to show a change unless and until we allay the fears related to antimicrobial hospital-acquired infections. So I would like to emphasize upon I would like the attendees to understand that it requires very different kind of activities. I mean, so far you would have heard about pharmacovigilance. It's a different ballgame, I would want to say. Here you need to have different role players. It is not just one person can run the show. It has to be multiple players there. And there are very different kind of activities, not just one kind of activities. You have to do audit and feedback. You have to make policies, devote a lot of time in an education and training, then you have to keep tracking. So then you have to increase the practice of de-escalation. So this requires liaising with the hospital administrator, it requires liaising with the microbiology, with the clinicians, of course. So this is just to emphasize that we have to understand various activities and understand that they come with a very different kind of a need. I mean, it is not that they are not doable, but the team has to be totally oriented. And then it requires a commitment. It is not one time of commitment. Things start showing change very gradually. I'll show later with examples. Then it requires actions which are very distinct. It requires tracking and reporting. And even the experts need to be educated on that. So that's just to emphasize that the systems are very different. And now since it is AMR just doesn't affect hospitals, it affects across the streams. And the origin of AMR is also across streams, and not just medical, but it spreads across to veterinary health, the way veterinary care is provided. It affects to various other practices like the use of antimicrobials in poultry and farming. And so those things also need to be taken care of. The way antimicrobials are pushed down over water bodies, that affects the way antimicrobial resistance eventually unfolds. So you just have to imagine that it goes much beyond hospital setups. It goes much beyond your own departments. It goes much beyond, say, allied specialties too. So that is one thing that needs to be understood. This is to understand how exactly different it is from pharmacovigilance and other pharmacological activities. Then it requires the depth of understanding. So different groups have to understand the system very, very well, and that requires working over a period of time, consistently on a day-to-day basis. Then, as I already said, that it requires multiple role players, which need necessarily be just these three highlighted there. And one of the important cog in the wheel is the clinician, and they are difficult to manage. It's not that it's impossible, but I won't say that they are in fault or anything, simply because they are the ones who are actually facing the consequences of that fear of AMR. The patient's outcomes going bad is a concern to them very deeply, and that's something which needs to be understood quite early in the beginning. Then an understanding of structures and resources is very important, because we say this is one of the ways of addressing AMR, and it indeed is, but the kind of resources that we have to understand the ways in the pharmacies are operating, what are the turnaround times of microbiological labs, other supporting labs, because not all answers for infections and identifying focus of infection are coming from microbiology labs. They're coming from many other distinct labs, so one has to have this understanding, and a very thorough understanding. Then the way medical record systems are there in any institute, if electronic health records are there, more often than not, in our kind of setups, they're conspicuous by their absence. Then training and education resources, if one wants to carry it out, where does one go to? I mean, it hasn't really received the kind of attention that, and the financial support that pharmacovigilance has received. In fact, most of the times it's operating without any kind of support. Then, importantly, how the patients are going, what's the flow of patients? I mean, when do they seek treatment? If you start right from the community, we have a very poor understanding of it. Why a patient is seeking in treatment, where does he go, what does he get advised by? So all this has to be understood in granularity.
Dr. Nusrat, sorry to interrupt you, you have 5 minutes more.
Fine, I'll finish it, wind it up in the next 5 minutes.
Thank you.
And then there are an understanding of different kinds of setups that have to be understood in order to say that it is not just one kind of intervention and one kind of approach which can address everything. So you have to tailor it according to healthcare setups. Just to give you a glimpse of one of the backbone activities of AMSP, which is one of the tools for addressing AMR, I would like to run you through some of the slides for prospective audit and feedback. And that itself is so among us. It has to be thorough or it could be focused. It requires development of a plan which helps you capture the way the prescriptions are written and devise the strategies for giving feedback. We depend a lot on this form which captures all the patient details, it captures the prescription details, microbiology details, which help us to provide feedback. Then where does the resource for implementing it come from? It's your foot soldiers who are important there. And how expansive you can get in doing this. We've been able to achieve this much, but I'm quite sure it would be a Herculean task with minimal residents available for carrying out these activities in most of the centers. So maybe we can skip this slide. This was just to emphasize. Then a lot goes into making people understand the process of making this decision about starting any antimicrobial and stopping any antimicrobial when we do not have tools for that. This is one of the tools that we developed for admitted patients, but a lot needs to be done for settings in outpatients where prescriptions come in this kind of example. And it is important to maintain a continuum. Just a quick look at this work that we had done in surgical intensive care unit. Over 5 years, we started with small changes, and I would like to confess that till date we have not been able to achieve the kind of benchmarks that we would want to achieve. Though we have joined hands with the Infection Control Committee and we have added the element of infection prevention and control, but still the progress is quite slow. The feedback has to be given in various forms, in meetings with all the consultants there, so it gets very huge, the way things are read out. And then one has to evolve. In fact, this was just an example of the way we carried it out during the pandemic, when we thought the antibiotic use was just going haywire. So we had to evolve, and there myself and one of my colleagues, Dr. Nanda Gaman, who was a senior resident at that time, we worked on a daily basis in our facility for managing these patients, and we were able to bring about some changes. Again, the changes were some only. Then the way the whole ecosystem works, it keeps throwing challenges. You run out of antibiotics, which are very important. So you have to resort to antibiotics which are perhaps less effective, can have potential to cause antimicrobial resistance. So those things also have to be taken into care. Training and education is not easy. So those models have to be shared, and it has to be done at various levels. Currently, we have been able to achieve it to a great extent over the period of time at our centers. We have several sessions, dedicated sessions running in different departments of the hospital. The list is a little longer than this. I've not been able to update it, but it takes time and it requires dedicated, concentrated work over a period of time. Then policy making is an integral part of this, and for policy to be implemented, you have to have people who are involved in using the antibiotics and writing the antibiotics involved right from the beginning. Otherwise, it becomes totally something which is at the beginning. So if you want to summarize if I have to summarize my talk, I would like to say that we already have a vertical in the form of National Action Plan on AMR, which has resulted in a few states to develop their own action plan on AMR. Within that vertical, we have several verticals. And this is being run by NCDC, is a body which takes care of it overall. AMSP is one of the verticals, infection control is another vertical. So there are verticals within those verticals. Basically, we have to consider it as a quality improvement initiative, much like pharmacovigilance, but it requires a separate vertical which is actually being recognized. It is not that we need to revisit it. It has been recognized at a national level, and work is in progress. It is slow to come back because of want of funds, because of a lack of understanding of how to go about it, and because it's a challenge which has been thrown upon us relatively recently. I mean, I won't say very recently, but relatively recently. So that would be all, and I would like to thank once again, and we will be happy to take any comments and queries. Thank you. Thank you very much.
Thank you so much, Dr. Nusrat, for highlighting the problem of AMR and what you have been doing as regards the problem of surveillance, monitoring surveillance, antimicrobial stewardship, as well as infection control policies at PGI and over the last few years. But just to tweak one of the statements you said at the end, that it needs another vertical, that pharmacovigilance is okay, but antimicrobial resistance needs another vertical. I just want to modify this statement. Actually, antimicrobial resistance is one of the verticals or one of the aspects that the pharmacovigilance topic, the umbrella term covers. So one of the areas that pharmacovigilance is expected to handle is that of AMR, and it's elucidated in the list of what comes under pharmacovigilance. And what I can see is that pharmacovigilance is the science of detection, assessment, understanding and prevention of ADRs or any other drug-related problems.
Excuse me, ma'am, I can't hear you.
You can't hear? Can you hear us? Hello, Dr. Nusrat.
Now it's clear.
Okay.
Yes, ma'am, it's clear. I missed out what you said.
No,
If you have time, if you could repeat it.
Yeah, I'll just kind of make it short. So, AMR is one of the aspects which pharmacovigilance, you know, that's an umbrella term, is already encompassing. So, under pharmacovigilance, we are supposed to monitor and try to work towards decreasing the problem of AMR. So, and if you look at the heading of the definition of pharmacovigilance, detection, assessment, understanding, and prevention of adverse drug reactions or any other drug-related problem. So AMR comes under any other drug-related problem. It's a huge problem. And what we are now trying to do is prevent this problem. So monitoring, surveillance, antimicrobial stewardship, infection control policies are all directed towards preventing the problem of AMR.
I lost you for some time.
Okay, I will, you know, for the brevity, because we are running short on time, I won't repeat, but I'll request the audience if there are any questions. Any questions? Dr. Nusrat, can you hear us? Dr. Nusrat? Are we audible?
Yes, ma'am. Can you hear me? Am I audible enough?
Yes, you're audible, but are we audible? No.
I was losing sleep, but I think I could make out the gist of what you said. I'd just like to make a quick response to it. It's very encouraging to know that pharmacovigilance does recognize AMR as one of the things within its umbrella, but all the same, it is very important to also mention that it is a separate vertical requiring things. There's some of the common points where it can work together, and perhaps the resources can be utilized, pulled in from places, as one of that of ADR monitoring. And I think antibiotics do come with their own adverse events, and they need to be monitored. It's not. In fact, if we review some of the data that I had a chance to look at, most of the adverse events that were reported for that particular group was for antibiotics only. So I think there are some commonalities, and it would make a good sense to pull in resources, but it is very important to understand that the kind of activities which are required are very distinct, very nuanced. And it will be encouraging to see more people get involved with those kind of activities. And I think pharmacovigilance has a very big pool of personnel which antimicrobial stewardship activities do not have. They have not been fortunate others. So I think it would make good sense to see how this pool can be utilized better for addressing the requirements for both. That much I think would be the something that we should all work towards.
Yeah, so just, I mean, you know, AMR under pharmacovigilance is not just about adverse drug reaction reporting, it's about the drug-related problem of antimicrobial resistance. That's what I was trying to emphasize. So, are there any questions? Does the audience have any questions for Dr. Nusrat?
Okay, thank you so much, Dr. Nusrat Shafir, for your wonderful presentation. Especially, I appreciate your work which you are doing at your institute, especially in relation to the antimicrobial stewardship programs, antimicrobial resistance related things. So thank you once again. So we move on to another lecture. If Dr. Joan is online, then I request her. Otherwise, it would be better if we ask her at the end of the third lecture. So Dr. Prasad Thota.
I'm here.
All right.
Here, sorry.
Okay, fine,
Yeah.
fine. Fine. Okay, fine. So we can ask, we can request you to please continue your lecture.
Sorry. I thought for the first time you couldn't hear me, so I logged out. Yeah, sorry about that. So I will share my screen again. Yeah.
All right.
Can you see my screen now?
Not yet.
Okay. No. I can say participants can now see your screen. That's
Yes,
what.
we can now see your presentation.
Okay. Okay, good. Thank you so much. So I apologize. I thought the first time you couldn't hear me or it was frozen. So my topic today is drug device combination product, and I'm going to speak about, especially going to emphasize on post-marketing safety surveillance of drug device combination product. A combination product is actually comprised of 2 or more regulated components that are physically or chemically combined or mixed and produced as a single entity. So here today we are going to learn about and I'll be giving you an overview of key definitions of drug device combination products in the EU and the US markets. Then we will look into certain laws, regulations and guidelines for combination products. And then we will classify the drug device combinations in the EU and the US market. We will also study the similarities and differences in the drug device combination regulations in the EU and the US. And then we will also look into some of the primary challenges in globally harmonizing drug device regulations. And then we will emphasize especially on the PV aspect of current regulatory requirements and how the robust pharmacovigilance system here in US, EU. So going to the next slide. Okay, let me just open 1 second. Yeah. So first of all, let's define what are medicinal products here, which are any subs. Before we go ahead and define what are TDCs or drug device combinations. Medicinal product is any substance or combinations of substance which may be used in or administered to human beings with a view to treating or preventing disease, or to make a medical diagnosis, or to restore, correct or modify any physiological functions by exerting of pharmacological, immunological or metabolic actions. On the other hand, the new MDR, the second article of the new MDR states a medical device is any instrument, apparatus or appliance or software implant, reagent, material or any other article intended by the manufacturer to be used alone or in combination for human beings for the purpose of diagnosis, prevention, monitoring or prediction, prognosis, treatment, alleviation. I mean, there is a list of things that medical devices could be used. So beyond just treatment or alleviation of treatment. So the next, we will go on understanding in the EU and the US market how drug device combination products are actually classified. In the EU market, a medical device could be either an integral or classified as integral or co-packaged or referenced in the PI or package insert. And there is a strong need in order to have the notified body involvement and a C certificate or declaration of confirmation to be certified for drug device combination drug device combination products. Whereas in the US market, drug device combinations are actually defined in the regulation 21 CFR. So in the EU market, we see a single integral product which is intended exclusively for use in the given combination and it is not reusable. Whereas a co-packaged medicinal product is packed together in a single package. Whereas referenced in PI, medicinal product and medical device are packaged separately, but according to the labeling intended for use together. Okay. The next is under, again I said, so the definition under the EU MDR, we have these 3 types: single entity, co-packaged or cross-labeled. We have what are certain examples here. In single entity, we see pre-filled syringes or transdermal patch or drug-coated stents, whereas in co-packaged, we see convenience kits or surgical kits. Cross-labeled are examples, we see certain lasers or light-activated drugs that are separately distributed but labeled for use with one another. So, so the idea here is to understand how how these various notified bodies are involved here. In integral or the drug device combination, we see that they form a single non-reusable product intended exclusively for use in the given combination. So here what happens is they are defined under Article 1, Articles 1, Section 8 and Article 1, Section 9, and they are defined in the MDR. And there's a notified body opinion which is required as part of application to competent authorities here. And while we are kind of applying for this, the integral drug device combination, but at the time of placing on the market, the Article 117 of the MDR plays a very high role, and it requires compliance with the GSPRs or safety or performance of the medical device. And there's again notified body involvement here, notified body opinion is required, or C certificate or declaration of conformity. They are considered basically as a class 3 medical devices, regardless of whether the medicinal substance acts directly on the body or not. So, and the co-packaged one, which has again medicinal product and medical devices, are again regulated under MDR. The regulations are not that stringent, although we do require a C certificate or declaration of conformity. And for the referenced in package insert or medicinal product and medical device, which are separated but intended for complementary use, they are also regulated under MDR, EU MDR, the C certificate or declaration of conformity is again required. We'll just focus a bit more on the first, the integral medical device, which is again a medical device with an ancillary medicinal substance. It is certified in accordance with the MDR, as I said earlier, and as established by the first paragraph of its Article 1, Section 8. And these devices are now considered as obligatory as, they are as class 3 devices, regardless of how the action is, and under the MDC. Hello. Okay. And clinical investigations are needed in order to prove the clinical benefit. Also, an electronic system is covering clinical trials is performed in the EU, and it is justified. The level of clinical evidence which is required is pretty high. And it is necessary to demonstrate conformity with the MDR. Also, there is a strong post-marketing surveillance need is required to report the serious adverse events and FSC as well. And we also required post-marketing PMCS as well as a PSUR that has to be updated annually, and a UDI system. There is a need also here for summary of safety and clinical performance, which is required, or SSCP, which lists main safety and clinical performance and the outcome of the clinical evaluation. And furthermore, when it comes to the information supplied by manufacturer, there is also a requirement by the MDR that devices containing or incorporating a medicinal substances must be included as must be indicated also on the label. Okay. So what goes in the technical documentation here? 1 minute. Sorry. So we have the GSPR, as I mentioned earlier, and safety performance requirement, and RMP, which is required very strongly, as well as the PMSR. Post-marketing safety surveillance report required, PMCFs are required, and it's a certain procedure which has to be followed by the competent authority. Now we kind of shift our gear from US to the EU, EU to the US, where in the US it is defined under the 21 CFR. The term combination product includes a product comprised of 2 or more regulated components that are physically, chemical, or otherwise combined or mixed and produced as a single entity. So basically, there is a combination of either drug device or biological or device, or drug biological or drug device biological. So they are all called as combination product. So how the regulation goes is we kind of have the primary mode of action, which is more important here. And the medicinal product is included in the combination product, and biological products and human cellular or based maybe again be constituent products here and are regulated as either drug, device or products. Everything is based on the primary mode of action here, and again we also have a streamlined approach here, where co-packaged and single entity combination products that are subject to both the GMPs and the device QS regulations. So that there is another streamlined approach as well, which has been adopted by the US. So let's kind of go ahead and understand more about primary mode of action here. And in primary mode of action under the FDA is defined as single mode of action of a combination product that provides the most important therapeutic action of the combination product. So the primary mode of action is either the principal action of the drug device combination product, or which one can have a significant impact on the safety and efficacy of the product. So the therapeutic action is the mode of action expected to make the greatest contribution to the overall intended therapeutic action of the product. So again, in the US approval application pathway, for example, adrenaline autoinjector is submitted through the medicinal pathway, or a drug-eluting stent or coronary stent is submitted through the medical device pathway. So it again all depends on the PMOA. Then we have certain here examples of medical device examples which are based on primary mode of action. When the primary mode of action is the drug component of the drug device combination, such as pre-filled syringes or intrauterine contraceptives whose primary purpose is to release progesterones, they are again certified, or they come under the umbrella of a drug, and then again how it has been certified, or how the performance of the medical devices how they are come under the classification. So we have these, the primary mode of action is the wound treatment products, which comprises a matrix whose primary purpose is the administration of medicinal products, of wound dressings. They all are classified as under the drug. For devices, we have, if the primary mode of action is the device, then they come under the classification of a device and not classified as drugs. So it all depends on which one is the primary mode of action in the US. So we'll kind of just touch base upon the similarities and risk and differences in EU MDR. It the intended purpose of the device and the inherent risk. Whereas in the United States, we see that they are all products under 21 CFR. Both require a very robust pharmacovigilance system, but at the same time, there are similarities and differences in how they are regulated. So both require EU and US require an adequate or robust PV system, but the differences is that in EU, the reporting is to the EMA or the competent authority, and there is no actually obligation to comply with the EU MDR. Articles 83 to 86 of the PMS of single integral device that is not CE marked is governed by Article 83 to 86. Whereas in US, it is always governed by 21 CFR section 4B, and it depends on the application type of the device as well as who is actually applying for it, the applicant type. So we'll go ahead and understand again the US regulatory pathway to ensure there is one set of documentation across EU and US. So as I said in the initially that the drug device combination in the UAR, if it's a biological or if it is a CDR, which is a drug, which is the primary mode of action, it is consulted with the CDRH. If it is a device, and again, the standalone devices directly go directly to the CDRH. And we will next go again and understand the post-marketing safety reporting requirement depending on the application type, which is NDA or BLA. So it all depends on the primary mode of action, as we mentioned, and who gets consulted based on the primary mode of action. So if it is the primary mode of action is the drug, then we go to the CDR, or if it is the device, we go to the CDRH. So it all depends on what is the primary mode of action. So depending on that, we have a different application types as IMDA or BLA. Okay?
Dr. Joan?
Yes?
Sorry to interrupt you, but you have just 2 minutes more. Can you wrap up?
Okay.
Can you wrap up?
Okay, yeah, sure. Okay, sure. Okay. Then we have the safety and the efficacy depending on the tracking. Hold on 1 second. So the tracking of the deviations, or how we track the deviations or adverse events or serious adverse events or protocols. It again depends on how there's a strong, robust pharmacovigilance system that we have, and how the mode of action is here based in EU as well as the US. And there is also a need for compliance assurance. As I said, post-marketing safety reporting, there is a need to again avoid unnecessarily duplicative reporting here. And the handling of communications as well differs in the EU and the US. Again, EMA's dedicated system for reporting quality defects. And as I said, in the US, how the handling of communication, depending on the primary mode of action, it goes to the CDR or CDH. And the device safety reporting requirements are described in the 21 CFR part 803 and 806, supplemented with specific reporting elements for each of the other constituent parts of the combination products. Again, the risk management here is in EU and the US, there is an ongoing risk management based on the ISO 14791. There's slight difference here in the EU MDR, which is defined in the Annex 1 of EU MDR 2017-745, as well as there is a requirement of ISO 13485. Then we have how the global management of risk we have, like in the it is governed by the FDA or EMA. In Japan, we have the the MHLWA, or the labor welfare system, which governs the drug device combination products. Now here the primary challenges we kind of govern are the difference in opinion on whether the device is integral or co-packaged, and on the classification of medical devices. And there is basically no legal definition of a significant, substantial, major change to a part of an integral DDC device. There is we also there is a challenge how to comply with the device labeling requirements. And there is also an unclarity of roles and responsibilities between notified bodies and regulators. Also for labeling for co-packaged devices, we always face this challenge, as well as to how to label the co-packaged devices. The next step here is the EMA, along with the small industry, actually is trying to develop a focus group to provide scientific advice. But it is basically only for scientific opinions, and there will be no advisory board here in order to how for those administrative procedures. The focus group will actually in the EMA, along with the industry, will focus basically on how to only provide scientific advice, and the pilot program is going to get kicked off by the end of December 2023. Which was actually agreeable in December of 2022 here in the program between the EMA and the small industries here, in order to help them, the small industries, in order to become more robust in the drug device combinations. So in order to summarize, we learned today the key definitions of drug device combination products. We had an overview of the current laws, regulations, and guidelines. We also classified drug device combinations in the EU and the US, and we learned the similarities and differences. We also learned the primary challenges in globally harmonizing drug device regulations and the pharmacovigilance strategy, which is based on current regulatory requirements. So we also learned today there was a focus group which will be established between the EMA and the industry trade organizations to have a pilot program to provide scientific advice for the drug device combinations. And basically there is a global harmonization which is needed for risk-based safety and quality management of combination products throughout their life cycle. So yeah, this is my presentation, and you're free to ask if you have any questions. Thank you so much for your time.
Thank you very much, Dr. Joan.
Sorry if I have exceeded my time limit, and I apologize. Thank you.
It's okay. Are we audible?
Yes, yes.
Okay.
Yeah.
Thank you very much for a very interesting overview of bringing in front of us a new term, drug-device combination. We talk very frequently about fixed dose FDCs, fixed dose drug combinations. Now there's another combination, DDC, drug-device combination. So that's a new term for people to be aware about. Thank you also very much for highlighting the definitions, the definition of DDC, the regulations involved with as regards DDC in Europe and in US and the challenges therein. I think your talk may have helped answer some questions which may have arisen in the first talk on material vigilance, with which this session 6 began in the morning. There were some queries on drug-eluting devices, etc. So we do have some regulations on that in the new Medical Device Regulatory Act 2020 under the Drugs and Cosmetics Act India. But it's evolving, this area is an evolving area. So I think some of the aspects that you've highlighted will help us in going ahead. Now, if the audience has any questions, Dr. Joan is available to answer. Are there any questions?
Okay, Dr. Joan, I'm just curious to know, did you see our MDR 2017 and found some variation? And is there any difference in the MDR in the US FDA or Indian? Did you try to compare? Can you comment on MDR 2017?
I'm sorry, I'm not very familiar with the Indian regulations as such. I'm trying to focus on EU and the US.
Okay, fine. I'm just curious to know if there's any difference, or you want to update something on our MDR 2017 rules. So if there is no question, and because of the lack of time, we can move to another lecture. Thank you so much, Dr. Joan, for joining from US, and we are very fortunate to hear you. Thank you so much once again.
No,
so
I'm based in Switzerland, so yeah. But yes, thank you very much for having me. Appreciate all your assistance, and apologize if there is a morning. It was a bit. I thought you couldn't hear me, so I just logged out. Sorry.
It's okay. It's
thank you so much. Yeah.
Okay, thank you so much once again. So we move on to another lecture by Dr. Prasad Thota. So Dr. Prasad, are you there?
Yes, sir. I'm available.
All right. So it's my honor to introduce very young speaker, Dr. Prasad Thota. He is a scientific officer, again working in Indian Pharmacopoeia Commission, and we are very fortunate that we have 4 speakers from Indian Pharmacopoeia Commission, and Dr. Prasad is one of them. And as a young scholar, he has published 48 papers, and he is very active in conversion of VigiFlow from WHO ART to MEDRA. And he has detected 5 Indian specific drug safety signals, which is very good. And he's a member secretary of the core training panel of the PAPI, and he has been a resource person for many programs, especially skill development program on pharmacovigilance for medical products. He's a member secretary of PAPI and has significant contribution for recognition and identification of NCC PAPI as a WHO collaborating center for pharmacovigilance in public health program. So it's very we are fortunate that WHO has recognized IPC and has designated IPC as WHO Collaborating Center for Pharmacovigilance in Public Health. And Dr. Thota has also published a paper on approach to alternative rapid microbiological methods. So I request Dr. Thota to please start your lecture.
Good morning, one and all. It's my pleasure to be part of this program. First of all, I would like to thank Chairperson Professor Vandana Ma'am and Rahman Sir, as well as the Sopicon team, as well as the AIIMS Rishikesh team organizing such type of events like this. So directly, I'm entering into my topic. Global drug safety signal versus Indian drug safety signal at a glance. So if you look at the global networking system about the pharmacovigilance, that program we can call it as WHO Program for International Drug Safety Monitoring. So it has been established in the year 1968. So if we look at the 55-year story behind the WHO Uppsala Monitoring Center, as on today, there are 155 full member countries directly they are reporting the adverse drug reactions to the Uppsala Monitoring Center. Apart from these 155 countries, there are 21 associated countries. They are taking the help of Uppsala Monitoring Center how to enroll those as one of the nationwide center under the ambit or umbrella of the WHO Uppsala Monitoring Center. So these are the demarcation of the full member countries and associate member countries. As on today, 176 countries being involved in the WHO Uppsala Monitoring Center program. So this is a current status about how the Uppsala Monitoring Center is working for the program for International Drug Safety Monitoring Center. So what is the story and what is the strength and what is the volume of the reports so far collected by Uppsala Monitoring Center? If you look at the data of the last 55 years being collected by Uppsala Monitoring Center, there are 3.6 million, there are 36 million reports so far collected by Uppsala Monitoring Center with the help of 151 full member countries. So if I look at the percentage wise the data which is being collected by these countries, if I talk on 85% of the reports, that means 32 million reports being collected by the developed countries. So rest of the 15% that I can say, that is 15%, that is around 4.9 million reports, it is collected by low and middle income countries. So as on today, there are 36 million reports which is available in the International Drug Safety Monitoring Database, that is we can call it as vigilance. So what is the strength of different 151 countries being so far reported since many 55 years? So if I look at the last year data, this is the annual report which is published by Uppsala Monitoring Center. If they segregated what are the top 10 leading countries, hardly I can say there are 10 countries has been scored under the maximum volume in reporting of adverse reaction. Thereby those are converted into ICSRs, individual case safety reports. This being gathered in vigilance data. So India is a country, it stood at 9th position. So the volume of the Indian ICSR contributed to Uppsala Monitoring Center for the year 2022 is 2%. So this is a good number and as well as percentage. So India is one of the toppest country among 10 countries being pillars for Uppsala Monitoring Center's program under the database that is vigilance. So what happens? So further after the collection of these reports, so after collection of these 36 million reports, so Uppsala Monitoring Center has to identify signals, those signals being published by Uppsala Monitoring Center, this further communicated to pharmaceutical newsletters who are the National Pharmacology Center being involved under this program, being receiving those signals, further these signals being circulated through this National Pharmacology Center newsletter, thereby nook and corner of the every healthcare fraternity, these signals being aware by different healthcare professionals when they are giving or they are taking medicines by bedside of the any patients. So out of 36 million reports, 25,000 reports or ICSR has been contributed in identification of 136 signals so far identified by Uppsala Monitoring Center, or we can call it as WHO signals. Either the both terms we can use. As on today, Uppsala Monitoring Center, they have identified 136 global drug safety signal among the consideration of the total 25,161 ICSR by deep medical review or clinical relevance between the what are the drugs reported due to the causality assessment, what are the reaction happened by combination of this drug-ADR combination through this 25,000 reports, they have identified 136 signals among the whole volume of the database, that is VG-based, that is 36 millions. So this is a background about what is the volume, strength, and percentage of the reports being reported by various countries, as well as under the umbrella or ambit of the Uppsala Monitoring Center program. So what we have to look at into this one. So for example, any drug ADR combination has been reported as adverse drug reaction. The drug which comes or which fits under one of the category, that category we can call it as WHO ATC. So drug definitely it comes under the categories WHO ATC, anatomical, therapeutic, and chemical classification of the medical products. So if you look at this slide, all the medicinal products which has been categorized into 14 categories. So the 14 categories, they have alphabetically categorized, that is from A to B. So for example, let me take one example. So the alimentary tract and metabolism disorder. If I talk on the proton pump inhibitors, so which comes, as we know in general pharmacology, which comes under proton pump inhibitor. So definitely it is acting on the alimentary tract system. So it is not as categorized as proton pump inhibitors as under pharmacology category. But apart from this pharmacology category, it has been categorized based on the ATC classification. So anatomical, therapeutic, and chemical classification, all the medicinal products has been categorized into A to V. So what these 136 reports has been further categorized based on the drugs reported so far. So if I look at these slides, so these are the alarming of this 136 nature. The 136 drugs safety signal identified due to the category of the L category and N category, followed by the R category. The L category, it is anti-cancer drugs which is causing a maximum. Apart from anti-cancer drugs, there are immunological medicinal products also available. If I look at in deeply into what are the 34 drugs which cause the adverse drug reaction, there are so many monoclonal antibodies has been introduced into India and globally for various type of treatments. Those monoclonal antibodies maximally they are causing the new adverse drug reaction. So those new adverse drug reaction with due to this new drugs has been considered as drug ADR combination, which further leads to new ADR. So that indirectly implies these are the signal has been generated. If I categorize these all drugs which prone to signals out of 136, there are 34 categories, which is anti-neoplastic as well as immunological drugs, that is monoclonal antibodies, which cause the maximum number of adverse drug reaction. So subsequently, rest of the categories it is divided. And if I talk on this N category, if you can see, apart from the anti-neoplastic drugs, the second category, that is N category, the nervous systems means the nervous system related drugs which cause the second top leading adverse drug reaction due to this respective category, that is nervous system. Followed by another category, the third category, that is R category. If I see the R category, the respiratory system related drugs cause the maximum number of signals. So this is the split of the 136 signals based on the ADR combination. The ADR has been further categorized based on the anatomical, therapeutic, chemical classification of the drugs. So what about the reaction part? So if you look at the adverse drug reaction has been picked up with any of the terminology, that is we can call it as a MEDRA terminology, medical dictionary for regulatory activities. So if you look at the MEDRA, there are 27 category has been subdivided. So if I talk on this 136 signals, there are maximum 3 category where the adverse drug reaction has been happened. The first one, which is related to skin and subcutaneous tissue disorders, and followed by gastrointestinal disorders, then followed by psychiatric disorder. So these are the maximum number of the organ where the drugs which is causing a maximum number of the adverse drug reaction in the human body. So that's why it comes under the category of the system organ classification. Why I wanted to highlight these are the 2 slides. For example, if I am the MD student, if I am a DM student, if I am a pharmacy students, though I am a clinical pharmacologist, so I am doing any projects which is relevant to the pharmacovigilance. So these are the drugs and these are the alarming. So these are these new drugs is giving some alarming nature. So you can focus on these drugs. This can be further monitored, which can help in the pharmacovigilance program of India to identify these are Indian specific signals rather than depending upon the WHO signal. So what is the signal? So far 36 million has been reported, and there are 176 countries being involved in this Uppsala Monitoring Center program. So what they do? So as the ICSR, it comes to National Pharmacovigilance Center through Regional Pharmacovigilance Center, then it comes to Vigiflow data. Further from the National Pharmacovigilance Center, the Vigiflow data, it will be entered into the Vigi-based data. So once the Vigi-based data has been taking the full information, there is another channeling mechanism from the transcribing of the data from the Vigi-based to Vigilize. So Vigilize is the data where there are so many statistical tools are inbuilt. Initially, those statistical tools are made as algorithm-based. So those algorithm-based mechanisms are involved. Those algorithm-based mechanisms, they have implemented as tools. Those tools are being implemented or involved in the identification of the signals. So what is signal? The reported information on a possible causal relationship between adverse event and a drug, the relationship being unknown or incompletely documented previously. So let us say, for example, any drug A, it is causing reaction B. So the drug-A combination AB. So the reaction B is unknown to the drug A in the past, or it is incompletely documented with the use of the drug A. So the new reaction with the use of the drug known A, so it is incomplete documented previously, or it is new one due to take of this drug A, that can be considered as signal in the pharmacovigilance perspective. So signal means the identification of unlisted ADR from the reported drug safety data. So how the signal can be identified? There are two mechanisms. Signals being identified at the central level, means that is in national category or international category. So how the national pharmacovigilance signals being identified? So every country having their own database, or they are depending upon the Uppsala Monitoring Center Vigiflow database. So once the data being collected at the Vigiflow, they are doing themselves the clinical relevance spotted through this Vigiflow data, rather than then they are going into the vigilance data just in order to identify. So the disproportionality analysis basis. So if you look at the right side of my slide, there is a visualized data is there. This visualized data base being given to every country. They are doing with the help of this visualized data, they are doing their statistical analysis. So if you look at this second, right side of the slide, the statistics parts is there, ICSR parts is there, the data mining. This is the art of the tool where this data mining tools are inbuilt in this visualized software. It definitely gives the country level alarming to those particular drug ADR combination with the help of information component. This is the core statistical tool which is utilized for the identification of the drug safety signal. So in nutshell, I want to say at National Pharmacovigilance Center, they are identifying signals based on the Vigiflow, that is the raw data. Further, with the use of the tools, that is Vigilance tool, they are implementing the data mining techniques. They are pulling the data mining techniques values from this Vigilance, combining together the National Pharmacovigilance Center. They are identifying the safety signal. When it comes to, as I quoted here, 136 signals has been identified in Uppsala Monitoring Center. So this is the Vigilize is the data and the only tool. It is identified so far 136 signals out of the 36 million reports. So this is the nutshell I want to segregate and differentiate how the National Pharmacovigilance Center, how they are doing, and how what are the databases involved in the pooling of the data as well as data mining tools with the help of the Vigilance. So in general, as a thumb rule, how the signals being identified? There are two eyes for these signals. The left eye we can say qualitative manner, and the right eye we can say it has quantitative manner. The qualitative manner means it is purely depending upon the clinical relevance. So once it is related to the COVID, let me take one example. COVID vaccine, it is causing the hearing loss or tinnitus. So it is an alarming nature. If the number of the ICSR reported, it is more than one. Let us take as 3 examples. If the drug ADR combination, the COVID vaccine, which is causing the tinnitus, are hearing us. Those 3 reports we are putting in front of the clinical people who are involved in the identification of the signal. Those committees, we can call it a signal review panel. In that particular signal review panel, the particular person or their relevant field which is related to ENT, those persons being involved for the clinical relevance between the whether this COVID vaccine is causing the tinnitus or loss of hearing. So those are the clinical part which is being taken by the signal review panel. When it comes to second eye, that is the right eye, we can call it as quantitative basis. So as I mentioned, the quantitative basis poorly depending upon the visualized data. From this visualized data, we are doing the data mine. From this database, we are analyzing and pulling the data with respect to the IC value, information component. So the information component, that is the based on the algorithm that we can call it in other term, that is we can call it as BCPNN, Bayesian Conference Proper Neural Network. So this is algorithm, this is the model which is adapted to Epsilon Monitoring Center for inbuilding the information component tool in the visualization. So these are the 2 parts which is being involved in the identification of the signal. So as I mentioned, there are 136 signals being identified Epsilon Monitoring Center. So these are the global drug safety signal identified Epsilon Monitoring Center. So these are the some red portions. So these are the data which is pulled from the 155 countries. So whether the India has been reported any case reports or ICSR out of these 136 signals, but it is very cautious to us. All these red color portions, it is being reported, it is being published somewhere in Indian journal by Indian authors. But in fact, those reports have been not into the ambit or under the umbrella of the pharmacovigilance program of India. These red colors, which is identified by the global signal, this global signal, there is no ICSR from the India, but rather than as ICSR, those red color portions as available as research article or meta-analysis or review article or case studies also, even those being published in Indian journals and foreign journals, though they are the authors and the clinicians from India, but those reports have been not reported to into PVP database. So this is one of the challenging area, how to identify the signal of the Epsilon Monitoring Center, where the evidence based from the Indian ICSR. Though it is a voluntary program, this is a suspected program, so that's why we cannot predict that this can be reported by every healthcare professional. So, but once it is giving some alarming message that, for example, as I am a clinician, though I am a PharmD, though I am a MD, though I am a MBBS student, so if you are reporting case report, immediately you have to send such type of the new adverse reaction related cases immediately to the PPA. Thereby, those can be immediately as at the earliest stage, immediately those can be considered as drug safety alert. Then further, those going to be strengthened as drug safety signal. So these are the different drug area combinations which has identified with Epsilon Monitoring Center as signals. So if you look at the last COVID-19, the COVID situation taught us a lot, but COVID-related vaccines also, it causing the hearing loss and tinnitus. The second one, that is COVID-19 vaccine, that is, it is causing the myocarditis patient. They have enrolled and they have monitored the 6,78,000 reports, but in fact, there are 134 ICSR reported due to COVID vaccine, which is caused the myocarditis. So this is the alarming nature, and as we know that pharmacovigilance, it is not only related to drugs and even drug-related problems also, even in specialty group of people also. So these are the signals which is brought up, and they have identified not only based on the drugs, which is based on the different category of the people and different drug-related problems also.
Dr. Thoda, your time is nearly up.
Yes, ma'am. Just I am going to finish it now. So these are the different 136 signals being identified by Epsilon Monitoring Center, and these are the list of the which is available in Epsilon Monitoring Center database. And this is with reference to drug-related problem as well as drugs. Even one or two are related to medical device and drug-related challenging problems also. Those has been identified signal. So drug-drug interactions also. So these are the especially out of 136 by combination taking of these two drugs together, there is a consequence of severe ADR. This has been identified out of this 136 there are 9 definitely and particularly the drug-drug interactions where as signals has been identified by there is a relation between the drug-drug interactions. So these are the list which is depicted here. And now coming to Indian basic signals. So based on the there are more than 7 lakh adverse drug reaction or ICSR we can say. So as of today, India has been reported. So these are the signals being identified by Indian system, that's Conference Program of India. If I connect and compare between these Uppsala manufacturing signals and Indian signal, there is no relation between these two. Those based on the international database, this is based on the national database. So that's why there is no data which is being categorized, which is being involved for the international. So this is the information just I wanted to have. I wanted to aware the particularly clinicians who are involved in the clinical practice. So India we are identifying, and apart from the signal, we are identifying the PA location also. Information update. Here the laboratory drug which is used for the chronic seizures. So we have identified SJS. This is being identified. It has been circulated to DCGA. Further, DCGA has taken action. It is given to marketing authorization holder. Further, those marketing authorization holder updated their PK that is 10 and
Dr. Thota, can you hear us? Can you please unmute yourself?
yes, ma'am, I can hear you.
Okay, now we can hear you. So you've concluded your talk?
Yes, ma'am, yes.
Thank you so much for letting us know about the global database and the differences between, the contribution of the Indian Pharmacovigilance program to the international database. That it makes us feel good to know that we are among the top 10 countries that is contributing to the global database. 2% at 9th position is a wonderful position to be, but as you've highlighted, we have a long way to go. Out of the 136 signals which the Uppsala adverse drug reaction UMC has identified, there are 4 you said which are based on publications and data sent by Indian authors but not through the Pharmacovigilance Program of India. So as you rightly said, we should, while we publish, but we must try and send all the ADR reports, whatever we have, through the Pharmacovigilance Program of India, so it comes out as a national, it becomes a national repository as well as a contributory source to the international signal determining database. So that's a very, very important thing that you've just told us. Now, also what makes us feel very good is that I think 10 or 12 signals have been identified by the PVPI based on Indian data which don't have a link with the international data or the international signals. That in itself is very confidence boosting because it makes us realize that the PVPI is now working for the population, sending back signals to strengthen the hand of the clinicians and all the people who are using the drugs. So thank you, Dr. Thota. And are there any questions in the audience for Dr. Thota? He's shared a very vital repository of data which PVPI has. Any questions, Dr. Rahman?
Yeah. It was very nice hearing to such a good talk by Dr. Thota. I really appreciate the hard work he has done. And we are actually got a lot of things about what our system in India has and what all new types of signals sort of cautions they have created. So it will be a lot of help for us, especially in the Indian context, to know about the site-specific, geography-specific, site-specific, geography-specific types of signals which we should be looking for and question our practitioners regarding the same. It was a really enlightening talk. Thank you, Doctor.
Thank you, sir.
Thank you, Dr. Jagdish, for your comments. So I think once again, Dr. Prasad Thota. And okay, now.
Thank you, sir. I express my sincere gratitude on behalf of the organizing committee of Sopicon 2023 to our speakers, Dr. Kalai Selvan, Dr. Joanne D'Souza, Professor Nusrat Shafiq, and Dr. Prasad Thota. We will be sharing a token of appreciation via post to all the speakers. May I now request Professor Ratinder Chaj ma'am to kindly come upon the dais to present a token of gratitude to the chairpersons. Welcome, ma'am. Thank you, ma'am. Thank you, sir. Thank you, ma'am. I now invite all for tea at level, which is served at level 2 of this auditorium. May I please ask all the delegates to kindly proceed for tea. Yeah. I request you all to come back within 5 to 10 minutes. I request all the delegates to proceed and kindly join us back for the prestigious KC Single oration, which is going to be delivered by Professor Daya Kishore Hazra, sir. Thank you. Welcome back, delegates, to the illustrious Casey Single oration. I would like to invite our chairpersons for this session. I welcome Professor D.C. Tashmana, Head Department of Pharmacology, Himalayan Institute of Medical Sciences. I extend my welcome to Professor Ratinder Jaj from All India Institute of Medical Sciences, Bhopal. Welcome, sir. Welcome, ma'am. Now I invite Professor Syed Ziaur Rahman to the podium to introduce the oration to the delegates. Welcome, sir.
Thank you so much. Before we start our second oration, I request our President, Society of Pharmacovigilance, Dr. Sandeep Agarwal, to please take your seat and come over the dais. I also request our distinguished speaker, Padma Shri D.K. Hazra, sir, to please come over the dais. We are honored that this oration is after the name of Professor K.C. Singhal, and he is also here. So I also request Professor K.C. Singhal, sir, to please come over the dais. So thank you so much. Before I request our chairperson to chair this session, I would like to inform you that this oration is after the name of our founder, chairman of the Society of Pharmacovigilance, Professor K.C. Singhal. It was started in 2005. This oration was started in 2005. Yesterday I told you that the John Orton oration was started in 2002, and I also named some distinguished speakers who delivered the John Orton oration. But you know, this is another very prestigious oration of our society, and in this we have earlier invited many people, stalwarts from the field of pharmacovigilance, including from International Society of Pharmacovigilance and Uppsala Monitoring Center. Like we had earlier speakers, Dr. Shanti Pal, Dr. Stan Olsen, RHB Miboom, Eugene Pan-Brook, Jampalu Vilu, who was the president of the ISOP, Ruth Savage from Australia, and Chris Boxtel. This is just a few names from the list of the K.C. Singhal oration. And this oration, as I said, was started by the society, and I would like to inform you that Professor Singhal deserves this oration because he not only started the society but also played a significant role in the field of pharmacovigilance in India and abroad. So just briefly, I would like to tell that Singhal sir has started his course from Aligarh Muslim University. Then he did graduation in medical science and MD in pharmacology from King George Medical College, Lucknow. He also did PhD in 1976 from Sardar Patel Medical College, Bikaner, and also got honoris causa DSc from Aligarh Muslim University. He served as the chairman Department of Pharmacology, Jawaharlal Nehru Medical College from 1990 to 1996, and then from 1999 to 2002. After retirement, he was the founder, vice chancellor of the NIMS University, Jaipur. He has made extensive and highly significant contribution in the field of pharmacology also. Like he developed Cyteria cervi as one of the model for the screening of anthelmintic drugs. He was the foremost pioneer in the field of Indian pharmacovigilance also, in addition to in general pharmacology, and organized many scientific meets in the field of pharmacovigilance. You all know that he founded the Society of Pharmacovigilance in 1999. He has been the recipient of several awards, including the orations like B.C. Sharma Memorial Research Award of ICMR, S.S. Parmar Foundation Award of Indian Academy of Neurosciences, Major General S.L. Bhartia Oration Award of the Association of Physiologists and Pharmacologists of India, Colonel R.N. Chopra Oration Award of Indian Pharmacological Society, Platinum Jubilee Oration Award of Indian Science Congress Association, G. Achari Oration Award of Indian Pharmacological Society, John Orton Oration of this society. And he was also the distinguished visiting professor of the University of Tennessee, the guest of John Orton. So with this brief introduction about the oration, I very humbly request Professor T.K. Hazra, Padma Shri, and also, sorry, before I invite him, I request Chairperson to please start this oration ceremony.
Thank you, Professor Rahman. It has been the responsibility given to me is to introduce our guest speaker for today's Professor K.C. Singhal Oration Award. I'm thankful to organizers, especially Professor Handu, for giving me this opportunity. With that, let me introduce our guest speaker today. And the guest speaker is Professor Dayakishore Hazra. His specialty is nuclear medicine as well as medicine. He is awarded from Padma Shri in his field. He had done his MD in 1968. Afterwards, MSc Nuclear Medicine with distinction from London, PhD from London, and then various other degrees. Other awards that FNAMS, FICNM, and FICP. As far as his academic positions were concerned, he has a long experience. He was professor and head of the Department of Medicine, as well as professor of nuclear medicine at SN Medical College, Agra. And along with that, he was having post of ICMR research scientist and advisor to nuclear medicine, government of UP. He held many illustrious positions in the past. He was President, Society of Nuclear Medicine in India. He was Dean, Indian College of Nuclear Medicine. He held the position of Vice Dean, Indian College of Physicians, President, UP Chapter, API, President, Geriatric Society of India. He is Editor, Nuclear Medicine Communications, London. He has been Editor to Indian Journal of Nuclear Medicine, Consultant to International Atomic Energy Agency and WHO, as well as member WHO Steering Committee on Radioimmunoassays and Standardization. He is member Nuclear Medicine Committee of Government of India. He has over 250 publications to his credit, and he has guided a vast number of MD, MS students, as well as 12 PhD students in his career. Still, he is very healthy, hearty, and doing a lot of service to the society by serving them. I welcome you, sir, for this guest lecture, and his title for today's lecture is Pharmacovigilance in relation to diabetes interventions. So, Professor Kishore Hazra, sir, please.
How do I get this? Yeah. Professor Singhal, distinguished members of the Society of Pharmacovigilance, I am conscious that delivering the lecture is a real honor, because this lecture is named after Professor K.C. Singhal, who I regard as the John Auditorium of India, and also the founder of this society. He is the creator in our country of the physician's interest in pharmacovigilance. It was initiated originally as science and activities relating to the detection, assessment, understanding, and prevention of adverse effects of any drug-related problems, focusing on phenomena noticed after the drugs are released for use. It has led to many concepts such as clinico-pharmacological reconciliation, drug safety assessment, recognition of warning safety signals, and the recognition of the harmful effects on the environment, such as drug-resistant bacteria, and even things like vultures being wiped out by diclofenac. Why are we interested in pharmacovigilance? The knowledge related to the safety profile of a product is initially assessed through the drug in the phase 1, phase 2, and phase 3 studies. But after it is released for general use, there's expanded use, and then the number of patients exposed and the types of patients exposed is radically altered. In particular, during the early post-marketing period, the product might be used in settings which are very different from clinical trials, and there's a larger population exposed in a relatively short time frame. So this is the knowledge which forms the basis of pharmacovigilance. This, in addition to ensuring safe use, it improves the benefit and risk balance of using any agent on a human being. It gives timely information feedback to the users of the medicines. And equally important, it gives feedback to the designers of the molecules which are being used, so that very often by tweaking them, one can reduce or eliminate the potential dangers noticed during pharmacovigilance. So it has a benefit not only to the patient, not only to the physician, but even in drug design, in making newer molecules. Now, these activities must continue throughout the product life cycle. There is a scientific approach to risk documentation which has been promoted by pharmacovigilance. Regulators, scientists, innovators of new molecules, and industry must all collaborate in the organization of pharmacovigilance. There are special populations in whom the drug may be used or could have been used, which were not addressed in the initial trials. And these are, for example, children, very old people, pregnant women, lactating women, patients with relative comorbidities such as hepatic or renal disorders, subjects with disease severity very different from that studied in clinical trials, either much greater severity or even much lesser severity, subpopulations which carry known and relevant genetic polymorphism, and subjects who are of different racial and ethnic origins. So, because these special populations are now being exposed to the drug, new phenomena may be observed which help to contribute to the knowledge of pharmacovigilance. Also, one must remember that when one notices a side effect after use of a drug, you must compare it with a background incidence. Suppose patient condition X is an important adverse event when using drug Y for disease Z, then one needs to go back and see what was the incidence of the same condition in patients with the same disease who had not been treated with drug Y. Therefore, comparison of background incidence is extremely important. Information on risk factors for an adverse event would also be useful to include if available. Now, in the context of diabetes, I must emphasize that today many drugs which were initially introduced for diabetes itself now have expanded indications. For example, the GLP agonists both the injectable varieties and now the oral varieties, the notable drug being Ribelsis, are being used to treat obesity. Not only diabetic obesity, they are also being used in non-diabetic obesity. Similarly, the SGLT2 inhibitors, which were initially used to treat the diabetics, are now being used in heart failure as well as kidney failure. And not only when these failures complicate diabetes, but they are now being used in heart failure in general and in renal problems in general. So today, dapagliflozin and empagliflozin, the leading SGLT2 inhibitors, are being prescribed by cardiologists and nephrologists to patients who have no diabetes at all. Similarly, fatty liver has many causes, including diabetes, and glitazones and sarglitazar have had a great success in treating fatty liver, but now they are being used in fatty liver of any origin, non-diabetic, alcoholic, post-hepatic, in addition with cirrhosis. One more example I would point out is that metformin, one of the most commonly used anti-diabetic drugs, is now shown to have advantages in anti-aging. Some claim it may have advantages in the prevention of Alzheimer's, and thus metformin is being tried out in various anti-aging and Alzheimer prevention trials. So what I'm emphasizing is that these drugs, which were originally introduced for diabetes, now have expanded indications: obesity, heart failure, renal failure, fatty liver, anti-aging. Similarly, the drug, when it is introduced for a limited population. But as practitioners use these drugs, they tend to extend the indications. For example, metformin was originally used only in non-pregnant diabetics. Today, it is being used even in pregnant diabetics. Similarly, the alpha-glucosidase inhibitors such as acarbose, which were introduced for diabetics as such, have now been used even in pregnancy because they moderate the hyperglycemia. So metformin and alpha-glucosidase inhibitors are today believed to be safely used in pregnancy, in addition, of course, to the sheet anchor of insulin. Similarly, polycystic ovarian syndrome is considered to be the sister of diabetes, and there are many therapeutic aims such as fertility, such as the regulation of the menstrual cycle, which can be achieved by anti-diabetic drugs, even when the patient is technically speaking non-diabetic. So this is an extended indication. Similarly, I was mentioning the use of the glitazones for hyperglycemia was noted that they also helped fatty hepatomegaly, and therefore they are being used in all types of chronic liver disorder. Erectile dysfunction, one of the most important causes, is diabetes, but many of the anti-diabetics, particularly metformin, have been found to be useful in erectile dysfunction of non-diabetic origin or cyclinic origin. The same remarks apply to the use of anti-diabetic drugs in coronary artery disease in patients who come from a highly coronary family for preventing major coronary damage. So these are extended indications. How does one look for the new adverse effects? One is, of course, passive surveillance, case reports, oral discussions, spontaneous reports, case studies, and this morning you heard an account of how the centers in Uppsala, which monitor all these and analyze all the reports mentioned regarding a particular drug, which can unearth the occurrence of a pharmacovigilance significant effect. One can also look for reports actively. Apart from that information gathered through case reports, active online reporting, surveillance, sentinel sites whose job, I think, like the center at Uppsala, whose major job is monitoring drug events. The data from insurance claims, where patients come with financial claims arising out of complications, can also provide a lot of data in relation to what are the drugs these patients were on. Did they include anti-diabetic drugs? Was there a relationship between them? So insurance claims are a fertile field for information. Drug exposure formal registries are also being set up in various countries, and you had this morning, you heard about examples of this. And there are cohort follow-up studies, either disease-wise or drug-wise. And these can give information not only about drugs and the pharmacovigilance arising out of that. It can, may I suggest, also give information about things like what is the best diet. That too, information can come from cohort follow-ups. So whether it's a Mediterranean diet, whether it is veganism, whether it is timed calorie intake. So if one has cohorts on these, one can pick up a lot of information which can be used to supplement our pharmacovigilance. So this is why I call evoked or stimulated reporting. Now what are the comparisons which can help? Cross-sectional studies, case control studies, cohort studies, and targeted clinical investigations. Today, the FDA insists on cardiac safety follow-up as mandatory for any new drug introduced, particularly in diabetes, but also in many other diseases. It insists on a long-term follow-up study. And then, very, very important, looking for congenital malformations in patients who have been exposed to it. Remember the thalidomide story. Remember that Europe had a lot of thalidomide disasters, but the US escaped because the US Surgeon General saw the European reports and decided not to permit the use of thalidomide in the US. And she was later decorated with the Congressional Medal of Honor for this foresight. So that is how pharmacovigilance is an example of being wise. What are other sources? Natural history of disease studies, drug utilization studies, drug event monitoring, for example, skin reactions, exposure drug registries. How many patients are there who are on metformin? And having a register of that. And I feel that clinicians in India who have access to one of the largest body of diabetics can play a vital role in maintaining proper data on whatever side effects they see in relation to the drug exposure. I would also suggest, incidentally, that in India, we have a fascinating combination of various cooking oils. We have mustard oil in Kashmir and West Bengal. We have coconut oil in the south. We have groundnut oil in Gujarat. And therefore, exposure to these oils, the same registry which is looking at exposure to drugs, can also decide 10 years down the line as to which cooking oil is in reality better or worse. Now, I should particularly mention that with the newer anti-diabetics, which are quite potent in lowering blood sugar, the foremost danger which has to be picked up in pharmacovigilance is hypoglycemia. This can not only precipitate cardiac arrhythmias, because hypoglycemia triggers catecholamine release and rise of blood pressure and triggers arrhythmias, aggravation of ischemia of the heart and brain, and all the anti-diabetics, because all of them lower blood sugar, should never be given without caution about these, because they can all aggravate hypoglycemia side effects, arrhythmia, ischemia. Some of the anti-diabetics can precipitate renal failure. Glifosins and biogonines, for example, metformin. Hence, in any diabetic on a drug, it's important to watch the EGFR, the effective glomerular filtration rate. If we see a decline, it may be unacceptable, and it may be a signal that you have to change from that drug. For example, it has been shown that linagliptin, although otherwise a very useful gliptin, it has been shown to lower the EGFR. Incidentally, it has also been shown to trigger pancreatitis. Another very important thing to look for is fluid overload. The glitazones are famous for fluid overload, and the result is edema of the feet. Now, this is especially important not only from the point of view of pharmacovigilance experts, but because in North India, edema of the feet is feared by the lay public as a harbinger of approaching death. So, relatives of a patient who is diabetic and who develops feet edema, they get extremely worried. As doctors, we also get correlated with aggravation of cardiac failure or renal failure, and that's why we too look for fluid overload. Urinary and genital infections are of course common with those agents which increase glycosuria. Now the glifosins, they increase glycosuria. They are known to cause genital infections, melanocystitis, phimosis, paraphimosis, vulvovaginitis, and they are usually easily treated by antifungal drugs. But one needs to be aware of this, and one needs to withdraw the glifosin agent being used. Surprisingly, while genital infections increase in frequency, urinary infections do not. Or if they do only for a short time. And this is, people are suggesting that urinary infections are related to blood sugar, while fungal infections are related to the urine sugar. And therefore the genital infections go up. But the urinary infections, they may develop, but they are easily treated. Sometimes they have very dangerous complications, like the Fournier's gangrene of the scrotum, noted with certain glifosins, and a difficult condition to treat, but fortunately very, very rare. Sometimes the clinician increases the anti-diabetic medication without need. He fails to recognize that the blood sugar is high, not because your drug is inadequate, not because your drug needs to be changed, but simply because there is coexistent infection. In India, tuberculosis is extremely common, and silent tuberculosis in the diabetic extremely common. Similarly, urinary infections, skin infections, and fungal infections, they all increase the blood sugar level and the apparent need for increasing medication, when the correct course would be to treat that infection rather than simply increasing the anti-diabetic drug. Also, there may be in India very commonly a failure to recognize dietary errors. There are many of our diabetic patients who have been told, no sugar, and instead what do? They take gur, jaggery, or they take honey, thinking that ये चीनी नहीं है, ये हम ले सकते हैं आराम से, not realizing that the same gur gets converted to glucose in the body. So the anti-diabetic drug requirement goes up, and the clinician who fails to note this, he increases the dose, causing more problems. Also, there are sometimes errors in timing, and this may be because of the clinician's failure to explain things. For example, if acarbose is used after meals, it is virtually ineffective. It has to be given before meals so it can prevent the breakdown of the polysaccharides to monosaccharide which get absorbed. Errors in insulin storage. The patient says कि साहब बस सुई तो लग रही है मगर शुगर में कोई असर नहीं हुआ, आप ज़रा ज़्यादा देके देखिए, and the obliging clinician increases the dose. What he needs to do is to find out how is the insulin being stored. Is it being exposed to outside heat? Is it being, instead of being kept in the wall of the refrigerator, is it being kept in the freezer? Is it outdated insulin transported by truck from other states? Now this is a phenomena peculiar to India because the sales tax rates in various states differs. Therefore, unscrupulous drug sellers, they will import insulin from Tamil Nadu to sell in UP, not being worried as to what temperature it was exposed to during its transport. How does it affect us in pharmacovigilance? The clinician who is not aware of these aspects and does not interrogate, he increases the doses, and that causes hypoglycemia. Now, so we need proper patient education, not only about mistaken increase in medication, but also the patient needs to be educated that hypoglycemia is a greater and acute danger as compared to occasional high sugars. I find sometimes patients who see a high blood sugar and increase the doses themselves, not realizing that hypoglycemia is a greater danger than occasional high sugars. We need to educate them about the target sugars. The fasting 80 to 120 and 90 minutes after a meal 160 to 175. That is our target. And the target has to be confirmed by self-home blood glucose testing. If that is done, then neither the patient's relative, neither the patient nor the doctor will increase doses and cause hyperglycemia. Similarly, we as medical students teach that insulin injections must be site rotated, otherwise they become ineffective. There must be proper injection technique. After giving the injection, you must count 10 to ensure that all the insulin is in, not simply do a prick and pull it out. Also, the patient needs to remember that medication must go hand in hand with diet control, so that rather than simply blindly increasing drugs, one needs to look at these factors and avoid many of the dangers of a low blood sugar. So, what are my take-home messages? One is that diabetes management is an object lesson in pharmacovigilance. Pharmacovigilance starts with blood sugar home testing and neat records. Initially, the clinician must review the record every fortnight. I personally find that HbA1c is too slow an index. It tells me what has happened in the past 3 months. I'm more interested in knowing what's happening now. What's happening now is told by the blood sugar, and fructosamine tells me over the last 3 weeks, which is much more valuable to me. So I tell the patient कि मुझे आपके पिछले जन्म के कर्म में कोई इंटरेस्ट नहीं है, मैं जानना चाहता हूँ कि इस जन्म में क्या कर्म कर रहे हैं. So that tells me how the control is over the last 3 months. And of course, creatinine, SGPT, cholesterol must be reviewed every 6 months. And it is the duty of every clinician to keep his knowledge up to date. That means attending meetings, reading literature, and staying awake. There are other subtle dangers of hyperglycemia. Precipitation or aggravation of dementia, loss of memory, depression, cerebrovascular accidents, and confusional states. And these confusional states can lead to many things, like diabetics in a low blood sugar, getting off at the wrong station in a long distance journey, or wandering into traffic and being hit by a fast moving car. So these are the subtle dangers of hyperglycemia. Another very important cause of inappropriate hypo is that we have not adjusted the target. Many people believe, in accordance with the textbooks they studied 20 years ago, that HbA1c of 6 should be aimed at. Sometimes, if the person is old, then HbA1c of 7 to 8 may be more desirable, and 6 may constitute hypoglycemia. In 3 major trials in the US of anti-diabetic drugs, it was shown that the heart attack rate increased if one tried to achieve euglycemia, inverted commas, rapidly. One has to do it gradually. The targets are to be achieved gradually under clinical supervision rather than giving in to patients' enthusiasm or relatives' unwise enthusiasm. Finally, one must remember that there is a phase of supernormal drug sensitivity after an episode of hypo. If a hypo occurs today, then the clinician must reduce doses for at least 2 to 3 days. Otherwise, further hypos will occur. So even the dose needs to be reduced because of the supernormal drug sensitivity which occurs after a single hypo episode. And what are subtle indicators of hypo? Irritability, raised blood pressure, headache, memory impairment, oppression in the chest, although the EKG is normal and the trop I is normal, unexplained breathlessness. So, my final slide is this. An alert physician is the keystone of pharmacovigilance. He keeps an eagle eye on the patient. He insists on regular follow-up.
I think we have to mute all the participants on the online mode.
And helpful clinical staff or relatives of patients, they try to aggravate or increase the treatment without asking the doctor. Therefore, it's important that the patient, the relatives, the family, and the clinician must share their knowledge and keep up to date. Thank you for your attention.
Thank you, sir. Now I request next chairperson, Professor Ratinder Judge, to please for your concluding remarks.
Thank you, Dr. Hazra, for that very informative talk. In fact, so he's told us a lot of given us a lot of insights of pharmacovigilance, especially pharmacovigilance related to anti-diabetic drugs, and this is also a kind of a emphasis on the fact that drug surveillance is not only about the adverse effects. Drug surveillance may lead to findings which lead to extended use of a drug. which at the same time extended the use of the drug, meaning we are using a drug for an indication other than what it was approved for, in turn means that now we have to be more vigilant about it. So with that, not taking any more time, and I also want to congratulate the organizers, because it's not in every conference that we get the honor and the privilege of listening to a Padma Shri awardee every day. So yesterday we had Dr. Balram Bhargav, and today we have Dr. Hazra. So thank you and congratulations to the organizers for that.
Thank you very much. I now request Professor K.C. Singhal, sir, to please present a shawl to our guest distinguished speaker. I also request our President, Professor Dr. Sandeep Agarwal, to please present the certificate on behalf of the Society of Pharmacovigilance, in fact, the oration certificate, and also the plaque. So for photograph, I request Professor Shailendra to please come over the dais and have a photograph with our distinguished speaker. So thank you. Now over to the organizers, please.
Thank you, sir. I would once again express my gratitude to Professor Daya Kishore Hazra sir for sharing his knowledge and perspectives. I would now request President SOPI, Dr. Sandeep Agrawal sir, to present a memento to Professor D.C. Dashmana as a token of gratitude. I request Secretary SOPI, Professor Syed Ziaur Rahman sir, to present a memento to Professor Ratinder Jaj as a token of gratitude. With that, we conclude the KC Singhal oration.
Continuing with the 8th session, Pharmacovigilance for Ayush, I invite Professor Tarun Sharma from Indian Institute of Technology, Roorkee, and Dr. Sandeep Agrawal, President SOPI, to chair the session.
Please mute all the participants online. I invite Professor Taruna Sharma, Head, Himalayan Institute of Medical Sciences, to kindly chair the session. Now I request chairpersons to preside over this session.
The next session is on pharmacovigilance and Ayush, and we have two eminent speakers in this session. This would be a very interesting session, considering that the use of herbal medicines in our contemporary practice of medicine is increasing by the day. The interest in this is increasing, and henceforth the safety of usage of such kind of preparations carries still more importance. I would invite Dr. Taruna to introduce the first speaker.
Thank you so much, sir. I'll take this opportunity to invite Dr. Ravi Narayan Acharya, Director General, CCRAS, New Delhi, for his talk. Okay. Topic basically for this session is safety related to traditional medicines.
Okay. Since it will take some time for Dr. Acharya to organize his slides, may I invite Professor K.C. Singhal for his talk first, and this can be followed up with the talk of Dr. Acharya.
So this session on safety will be taken up later. So in the meanwhile, I invite Professor K.C. Singhal, sir, for his session on promoting use of traditional medicines, safety basically. Sir is former chairman, Department of Pharmacology, Jawaharlal Nehru Medical College, AMU, and founder, president and patron, Society of Pharmacovigilance of India. Sir is recipient of various awards, P.C. Sharma Memorial Research Award for IMA, Colonel R.N. Chopra Oration Award for Indian Pharmacological Society, G. Achari Oration Award for IPS, John Ocean Oration Award for Pharmacovigilance India, and many more awards and orations are to his credit. So, sir, we invite you for your talk.
Of which I know very little. And also, when I am going to present it before two stalwarts, Ravi Narayan Acharya, who is Director General of CCRM, and Padma Shri Professor Zillur Rahman, who is the expert in Unani. So I've got expert in Unani, expert in Ayurveda, and I know very little about it. But anyway, I'll make some efforts to do something. So first we have to see what the Ayurveda texts say. Ayurveda texts say, I think because of the fount, we could not show you the Sanskrit text. But in Ayurveda, in Charak Samhita, it is clearly mentioned that the drug which is given produces adverse reactions. First, it does its karma, which is done by virya, or the potency of the drug, and thereafter, the drug is transported into the body cells, as we study in physiology. After transportation, it is metabolized, and then it reaches the site of action, and its action begins. So that is exactly what we have learned in allopathic system of medicine in the Department of Physiology that Charak had said long back. So this is just the same thing, that the concentration of the drug is important. So we call it dose-related adverse reactions. If concentration is more, the effect will be more. The drug has to reach the site of action. And the site of action means the receptors, what we call, and there a drug-receptor combination takes place and the action initiates. So these are same thing which we are having. Then Charak has also described that drug is fatal if we do not know what it is, how it acts, where it acts, when it acts. It could be fatal. So this is just like a poison. हम कहते हैं दुधारी तलवार होती है दवा. It can do good if given in the proper dosage, in the proper format, at the proper time, and in proper patient. But it could, at the same time, if it is not properly used, it can be fatal. So we should very well know what the name of the drug, what the action of the drug, and what are its complications. So it is drawing of the attention of the physicians. Charak has very simply exemplified and beautifully narrated what a physician should do and how the drug should be used. So this is just pharmacovigilance, nothing else. So latent poison, that means the drug, we do not know what is latent in the drug. It can produce pustules, kitibas, urticarial rashes due to disorders of the blood. Thus, poison takes away life soon by affecting each dose. So what we are doing it now in pharmacovigilance is already written in our text long back. Similarly, exogenous swellings are caused by juices of the fruits, flowers, and so on. What happened with synthetic drugs? We had many worrying experiences. 1946, when streptomycin was first introduced for the treatment of tuberculosis, it caused deafness and renal failure. Then, if you, Professor, the senior people can understand what I mean to say, streptomycin was in two forms. One is streptomycin and dihydrostreptomycin. So it was thought that if we do the combination of the two, the adverse reactions will reduce. But it did not happen. So now we are left with streptomycin alone that we are using, but that causes these adverse reactions. Isoniazid-induced hepatitis, which is usually fatal. Chloramphenicol induced aplastic anemia, and thalidomide-induced phocomelia. So these were the worrying experiences which opened the eyes of medical community. Then phenylbutazone very commonly used for pain relief, aplastic anemia and bone marrow depression causing agranulocytosis, which was again a fatal reaction in many cases. Then aspirin induced peptic ulcer. If you look at the peptic ulcer induced by aspirin was identified about 50 years after the aspirin has been in use. Then oral contraceptive induced thromboembolic phenomena. Cleoquinol induced SMON, subacute myelooptic neuropathy. Although this myelooptic neuropathy was not seen in Indian population, it was identified in Japan. But B.J. Vakil in Bombay, with retrospective study, he found that some patients, they had symptoms just like subacute myelooptic neuropathy after being given enteroquinol. So that is now history, and we do not use this drug anymore. Then pectolol induced oculomotor neurosis. Then ketoconazole induced hepatitis, valproate sodium, spina bifida, the fetal abnormality, ofloxacin caused psychosis, ceftriaxone, biliary symptoms, so on. So after 1948, the data of causing side effect after a death caused by an anesthetist. Food and Drug Administration that came into, I'll go to it. Thalidomide was introduced in 59-61 as a sleeping pill or for morning sickness. Its safety was proven before marketing. UK yellow card system did not show any adverse reaction. Then 1967 WHO International Monitoring Program was there, but thalidomide had done its job mainly in Germany and around countries and in Europe. Luckily, we are late in introducing new drugs. So thalidomide did not come to India, otherwise there would have been havoc in our country as well. So if you look at the babies who were born, thalidomide babies, some did not have limbs, some had cleft palate, and so many other abnormalities. And thalidomide babies were sometimes kept in a different place, given a special treatment so that they could survive. So these are, you can see some of the photographs from that. So these are the photographs of catastrophe of thalidomide. Birth defects were there, deafness, blindness, disfigurement, cleft palate, internal disability, and so on. And this we do not go to. Now I come to Unani drugs. Unani is a system of medicine, which is a holistic system of medicine. Holistic means that we treat patient as a whole. We do not treat disease as we do in allopathic system of medicine. So there is a difference. And this system is based on mizaj or temperament. I'll come to that later. So there are 4 types of mijas: hot and dry, hot and moist, cold and dry, and cold and moist. And they have got, each has got 3 degrees. This is Unani system medicine. And if we identify a particular disease related to, say, hot and dry temperament, mijas that we call in Unani system medicine, if somebody has got hot and dry mijas, and if we given some medicine, that the medicine has to be given a reverse medicine. That means cold and moist. For hot and dry, we give cold and moist medicine. So, now I come to that. Now, I was talking about this. Unani drugs, they are given in a definite way. The preparation is different and so on. But what is there in Unani medicine? One thing, that there is a al-abdal, principle of al-abdal. Al-abdal means badal ke dena. ये दवा नहीं है तो हम उसकी जगह दूसरी दे दी. That is principle of al-abdal, and this was first identified in a book, Maqala fi Abdal al-Adwiya al-Mustamala al-Tib wa al-Ilaj. So in this book, Rezi, the Muhammad Zakaria, or Rezi call, the physician of Persia, 240 Hijri. He studied Indian medicine also. He was chief medical officer of Baghdad. He became blind at the age of 60, and 29 books have been written by him. Now, he first said that there are 122 drugs for which we can use a substitute. Now, if we use substitute, then they have given the method of substitution. And this is the book. All the drugs required for the treatment are not available everywhere. So if the physician is unaware of substitutes, which may be used in place of the original drug, the objectivity and benefaction of this medical profession will cease. So Unani system clearly indicates that if this drug is not available, you have to use an alternative. Even the compound formulations, they use a substitution. Dawal Kas, Dawal Kurkum, Rogane Hina, Rogane Marjarush, and Rogane Gul, Rogane Banafsha. So these are the substitutes which can be used if the original is not available. Now they have given the scientific principles also. Substitutes should be used when original drug is not available. No drug can be a complete substitute of the original drug. Drug should be substituted original drug for a specific activity. Different drugs may be substituted for different activities, and substituted drug should match the original in mijas or temperament. As I said, that a patient has a mijas, hot and cold, dry and moist and so on. Similarly, a drug also has a mijas, according to Unani system of medicine, and we have to give the reverse for the treatment. For hot and dry, we give cold and moist, a drug. So that is how the substitution is done. And Ibn Sina, he further propagated this principle. So I think I leave this. By middle of 19th century, 80% of the medicines were herbal. Even today, 25% of the medicines are derived from plant sources. Most of these drugs, they come from the folk knowledge of treatment, and some of these could not be substituted despite the enormous advancement in the synthetic chemistry and so on. Now, I told you about 122 substitutions were suggested by Rhazes. We went into the Unani literature and explored what are the substitutions used other than what Rhazes said. From the literature, I could find out that 50 more drugs in India of Unani system of medicine, they are substituted because original is not available. So the list becomes 172. I published this data earlier. Now, why pharmacovigilance? Pharmacovigilance of herbal drug is safe. It is a general conception. Then there could be drug-drug interaction, herb-drug interaction, which we do not take into account generally. Toxicity of the isolated ingredients, we find out. A traditional process like shodhana and so on, that could be there. So what are the problems? I think Professor Acharya will tell you in more detail and in more explicit manner. But thing is, then in allopathy we use single drug. Aspirin, paracetamol, chloramphenicol, streptomycin, valproic acid or whatever. But here we are using plants. And when we are using plant, it is a composition of several chemical substances. And when this is a composition of several chemical substances, multiple ingredients are there in the formulation, it is difficult to separate out these two, which one has caused. And for this, we require a very large database. And large database may find out where we are. Now, in Ayurveda, we have got, just like in Unani, we had Bijaz. Ayurveda is also a holistic system of medicine, and this is based on Vat, Pitta and Kapha. These are temperaments. Personality, Vat है, पित्त है, कफ है, कौन सी पर्सनैलिटी है. इसी तरह से दवाएं भी कुछ पित्त को बढ़ाएंगी, कुछ कफ को बढ़ाएंगी, कुछ वात को बढ़ाएंगी. तो ये एक समस्या यहाँ है. मगर Ayurveda and Unani, they are different in one sense. In Unani system of medicine, as I said, 122 substitutions were proposed by Zaki, and 50 more we identified, so total was 172. This was for active ingredients. But in Ayurveda, active ingredients are not substituted. It is not the excipient which could be substituted. Myself and Professor Ravina and Acharya together worked, and we have published a paper also that in Ayurveda we have taken several nighantus. Nighantu means materia medica of Ayurveda, and from that we have concluded that only the excipient can be changed in Ayurveda. Substitution for excipient can be done, not for the main drug. So, just like This is a book I translated the WHO adverse drug reaction terminology into Hindi. So, and this was published by Society of Pharmacovigilance India. And this is the meeting which was held in Delhi. Professor Ravina and Acharya was present in that meeting. I was there. It was before 2010 when the program was launched. And so we thank Professor Charak for giving us such a vast knowledge which we have done today. Thank you so much.
Thank you, Professor K. Singhal, for such a lucid explanation and insight about the usage of Ayurveda and Unani preparations. I would now like to invite the, you can join on the dais itself. Next speaker.
And also the key issue in the beginning, that's you highlighted very well like that. All 6 R's, they are really very important. Otherwise, whatever drug we are using for the benefit of the patient. But if all 6 R's are not identified in a proper manner, then it could lead to rather harm to the patient. Thank you so much, sir. Thank you. Now we invite another speaker.
Hello, namaste to all. Can I have the slides, please? At the outset, I thank AIIMS Rishikesh for giving the opportunity and SOPI to speak on pharmacovigilance aspect, and I have been allotted a title that is Safety of Traditional Medicines. I am associated with SOPI since last more than 10 years, and I thank Professor Singhal what Professor Hajra told. I think he is the father of pharmacovigilance in our country, and since 2007 I am associated with SOPI, and a session is being dedicated in every year for pharmacovigilance purpose. And in the next 15 to 20 minutes, I will be saying what pharmacovigilance, particularly the topic allotted is safety aspect of traditional medicines and Ayurveda in particular, and drug-related and disease-related. And safety of traditional medicines, how the contemporary needs different aspects are in the present society or presently how they are dealt with, and pharmacovigilance, how the National Pharmacovigilance Program for Ayurveda, Siddha, Unani and homeopathic drugs is presently working in the court. I will start with a note as told in the last slide by Professor Hajra, that a person needs to be eagle eye. Any physician must have an eagle eye. That Charak says 1000 years back. That safety monitoring, Charak highlight the word that muhur muhuscha roganam, that not only the vigilant aspect, every minute a physician needs to be vigilant, not that while administering drug. For total the patient's treatment procedure, Charak says that a physician needs to be vigilant throughout the treatment protocol and also highlight that nothing is safe in the world in terms of medicines. Not only in terms of medicines, but every aspect, nothing is safe, but it is only depends upon, as Professor Singhal rightly told, that thalidomide was a disaster, but thalidomide has been repurposed and used as medications also. So it is up to the person, up to the physician, how you are using, in which purpose and which conditions in that aspect. So safety of traditional medicines, Ayurveda being 1000 years old, knowledge that no paper writing system or public, as on today was available, it was only chanting and remembering all these things. So in maximum, all are in sutra form, what we call in the Sanskrit word, or what was in very short form, and it is up to us how people interpret it, and they have everything, the guidelines in a very concise form that. So the safety aspect of a drug, Ayurveda says, identifications, because a drug of herbal origin, it is to be collected or to be processed, all these things from that aspect, how the safety Ayurveda is maintaining, and processing and storage. And particularly drug-drug interaction, herb-drug interaction part has been well highlighted in the classical text, recommended contraindications and mineral drug and other drug reactions. Let us start with the first part, that is the drug-related and improper identifications. Ayurveda, Professor Singhal has rightly told that stanza, Ayurveda says everything is every drug is a poison. And not only poison, Ayurveda correlates the drug with the thunderbolt, and in that aspect, if it is used judiciously, it is a nectar. If it is not used judiciously, it is a poison. In that way, Ayurveda considers both a poison can be converted to a nectar, or nectar can be converted. It is up to the judicious use. Ayurveda always emphasizes on judicious use of the drug. And it says a physician must be well versed about three components of a drug, not only the nomenclature, that is identification part also, but its pharmacological properties and actions also. The concept of pharmacovigilance was quite vibrant at that point of time, and Ayurveda says in the last stanza, everything a physician must be aware about the pharmacological properties, actions, how to draw, all these things, indications, contraindications and indications, etc. But knowing all, doing wrong administrations can cause the word is anartha. That point of time, Charak says, anartha is nothing that everything, if you are not knowing all the things, but not doing administrating the drug in a judicious way, and how to administrate the drug, it has been well described. So, if you are not administrating the drug judiciously, then anything can happen, and that is the new concept, the word termed after 60th thalidomide disaster is the pharmacovigilance. Regarding the collection, storage, all these, a single stanza of Charak describe everything what WHO now says, good collection practice, good storage practice, good cultivation practice, good clinical practice, or any good practices WHO gives international standardization. Everything has been well noted in the single stanza of Charak Samhita, where it says that evang upaskrutyam, evang prakriti. I am not going to that in detail, but everything, every good practices, how starting from the collections till processing and administration, not only in the clinical practice, also in storage practice and processing practice, everything, it has been well noted in the classical text. And also regarding the processing of the materials, Ayurveda is very meticulous. Yes, we know. Many research papers now coming up, particularly after the COVID-19, when the Tinospora caudifolia with Ania somnifera has been highlighted for their different activities. We got many news, many articles are coming up on that. But all these things, every drug, as I told, every drug is a poison, and every drug that may be most effective drug like Guruchi or Ashwagandha, they have also adverse effect when they are not used or improper used or not properly processed. So Ayurveda gives much emphasis on processing, what we call as a shodhana in that term. So processing part is also needs to be very meticulously noted in the classical in the pharmacy sector part. And about another part of the drug, Ayurveda also that point of time, whatever the concept of now we call in the technical term herb-herb interaction, herb-drug interaction, or drug-drug interaction, like that. It has been well noted. I will cite some few examples here, how the medicinal plants, the vegetables, the drugs used at that point of time, that has been well noted. If this, we all know in our local practices also, we don't take the buttermilk or ghee in a copper vessel, we all know in our village or all these things. That has been well noted because it is observational and experiential science. So herb interaction, herb food interaction, all our animal origin interactions has been well noted in the classical text. This sample, this sage, these are the drugs. If you are Bacilla alba, this is the common vegetables, is taken by sesame, then it may cause adverse effects. Because those now many exotic vegetables are available. Tomatoes, potatoes, these are not the time of Charak Samhita there. All are exotic, and we are many exotic fruits. We are even papaya also not of Indian origin. But this type of, since last 200 years, there is no much research in Ayurveda sector due to colonial effect or whatever effect may be that. But the concept of herb interactions, herb-drug interactions are well noted in the classical text based upon those vegetables or those animals what have been consumed at that point of time. We know the concept of herb food interactions also. These are the concepts, these are the taken. We should not take garlic along with milk. We should not take radish along with milk, not to be taken wine. Also, some fruits, particularly not to be taken with. So there are many examples. I have cited very few in here, but many vegetables, many herbal interactions have been well noted, which we needs to be researched upon in this point of time. And animals also. Now, you can see how they have meticulously noted that if a person consumes the fruits or herbs or drugs in this way, along with those animals' product, then there is adverse effect. It has been. Not only that, Ayurveda is known for its metallic preparations, particularly the mercurial preparations. Ayurveda says if a person is consuming mercurial preparations, then he must not consume this Kakarashtaka Varga in Ayurveda, we told, that is drugs available with drugs or vegetables or fruits started with a K, that is Kantakari, Kadali, Kapitha, all these things. So, person consuming any metallic preparation, particularly the mercurial preparation, should not consume. But in the real practice, is it in nowadays it is not, we are blaming that Ayurveda drugs of our metallic origins having adverse effect. And as told by Professor Singhal, these are the classical texts. As Ayurveda has its own fundamental principles, that is Vata, Pitta, Kapha, and the Saptadhatu. So all the drugs has been highlighted. If you are consuming this, it will elevate Vata, it will tranquilize Pitta, or it will aggravate Kapha in that way. Every drug has been noted. So it is without knowing the pharmacological properties and actions and prescribing the drugs of herbal origin can get adverse effect in that aspect. And drugs, not only that, drugs of minerals, the dose, the diet restrictions are all noted in the classical text to have a safeguard on good clinical practice in that aspect. And not only to that, adherence to the fundamental principles, rational use of the drugs, dose-related and incompatible drugs diet, procedural errors. Procedural, there are chapters dedicated in Charak Samhita for metrovigilance or this other hemovigilance. All these are new concepts here, I can say 20 years back. But this metrovigilance has also been well devised, well described in the Samhitas, that how if you are processing or if the instrument is not properly developed, how the adverse effect, Panchakarma can adverse. Chapter has been dedicated in that part also. So these are the adherence of the fundamental principles, because Ayurveda is it's having own fundamental principles and the irrational use of these drugs, because Ayurveda concept consider every person as an individual, and person to person the treatment be differs. Not only the person, here the desha, the bala, the strength of the person, the time of administrations, the nature of the person, all these 10 factors are considered to prescribe a single drug. Not only that, a single drug of 500 milligram or 200 milligram BD, TID is not the principles of Ayurveda. Ayurveda considers all this in a holistic way, and then only the drug can be effective. That has been well noted in that. This dose related and exactly the dose. Never in Ayurveda science will find that this is a 500 milligram, 1 gram, 2 gram, 3 grams. It is up to the physicians, he or she decide based upon the strength of the person, but the maximum dose and everything has been, even if the low dose, what are the effects, that has been also described. And incompatibility of the drugs, I have already described there. Then procedural errors, that is faulty preparations of the instrument, like we all must be knowing there about the Panchakarma, about the Basti, the Virechana, the Bhamana. So how the procedural errors that has been well defined in the classical text of Ayurveda, patient-related, iatrogenics and drug-related also, it has been well described there. And now the question comes, why there is a pharmacovigilance concern among the public? Because many persons, we have a common notion that Ayurveda drugs are of herbal origin, it don't have any adverse effect. But the situation is not like, and many physicians without any degree, you can see that without a degree and all this practice, started practicing Ayurveda or Unani or Siddha system of medicines, and they started giving prescriptions without any proper degree or knowing the subject properly. And irrational prescription also, unregulated practice and irrational prescriptions are another cause of adverse effect. You can see the nature of this is one Ayurveda, another also the modern system or all this system, how the prescriptions are being written and irrationally. Even if in the Ayurveda, you can see a combo of medications without knowing the herb-drug interaction or drug-drug interactions, everything is being prescribed in that aspect. And another major because there is a global demand of Ayurveda, Siddha, Unani systems of now herbal drugs, in the name of herbal drugs, there is a global demand, but the natural resources are very limited, and as on today, in Indian scenario, more than 95% drugs are from natural origin, and only 5 to 7% drugs from cultivated sources. So, a poor quality maintenance is another major hurdles for pharmacovigilance concern, and direct internet sale without knowing the nature of the drug, where its indications, contraindications is another factor for this pharmacovigilance concern of and direct to consumer also. Direct to consumer, many without any hesitations, many online or social printing or online medias, all these things, and direct to consumer and over the counter also, availability of the drugs over the counter about the drugs is another concern, pharmacovigilance concern for herbal drugs. And another major is also concerning uninformed consumer. Generally, due to the notion that Ayurveda drugs are safe in that notion, there is no regulatory aspect, and there are not many pharmacies are not following the regulation part, and they are not mentioning about the ingredients and Schedule A. We have Schedule A. Many poisonous plants are there. We have the Schedule E1 drugs are there, and they are supposed to be used with a caution. But no, many of the drugs doesn't mention in that aspect also. And taking these into considerations, Government of India has started the pharmacovigilance program after this, just like in the thalidomide, make rage about the pharmacovigilance program globally. After this, one article was published by Dr. Sapper in the international paper in the 2004. After that, everybody's concern started about the herbal drugs or mineral drugs with metallic preparations having adverse effect, all these things. The Government of India started this program in 2007, and before that, it is Professor Singhal and his team, Professor Jia is here, and his team, and also some organizations started working on pharmacovigilance of herbal drugs. But Government of India took the lead, and after 2007, National Pharmacovigilance Program for Ayurveda, Siddha and Unani drugs started, and in 2018, homeopathy also comes to the fold, and now All India Institute of Ayurveda at New Delhi is the National Pharmacovigilance Coordination Center. Before that, through 2007 to 2018, Institute of Teaching and Research in Ayurveda, Gujarat Ayurveda University was the National Pharmacovigilance Center. And as on today, there are 99, but 91 functional about peripheral pharmacovigilance center. We have 4 intermediary pharmacovigilance center and National Pharmacovigilance Center at All India Institute of Ayurveda, New Delhi. And this one of the major component, this is working on this pharmacovigilance programs, and another major area considering this OTC product and misleading advertisement. The present pharmacovigilance programs have added, we have added that misleading advertisement about one of the major component there. And about creating awareness is another component of this pharmacovigilance program, because there is a common notion, as I told repeatedly, that even if in the field of Ayush practitioners also, that our drugs are safe. But to make them aware, also, lot of awareness programs are being carried out every year, and I am presenting the data. I thanks Dr. Galib for providing me data yesterday about the adverse awareness programs carried since 2018 to 2023. These are the awareness programs, and these are the reported adverse drug reactions of suspected adverse drug reactions from January 29 to April 23. These are the Ayurveda 47%, Siddha, Homeopathy, Unani, and Swaripa also. Not reported, but we have started reporting Swaripa also. And you can see the number of misleading advertisements reported. We have very vigilantly working and reporting about this misleading advertisement in newspaper. You can see during the COVID time, the number of misleading advertisements were peaked. Even if in a month we have more than 1,000, near about 1,000 misleading advertisements in the newspaper, but due to our intervention only and reporting all these things, they are reduced, because many people started claiming that these herbal drugs can cure COVID, these herbal drugs can cure that on this and that disease conditions. So this was a step taken by Minister of Ayush about misleading advertisement, and we are getting good result as on today also in that aspect, reducing the number of misleading advertisement. With thanks, I would conclude, there is no such thing as a safe drug. One should be cautious while handling a drug. It may be of synthetic origin, or it may be of natural origin or mineral, or because Ayurveda used the drugs of natural, herbal, mineral, metallic origins. So everything it's a drug, it must be considered in that way. Thank you. Thank you, Punitji, for your kind invitations in this aspect.
Thank you, Dr. Acharya, for your nice presentation. And we all like to congratulate, first of all, like the initiatives that you have taken for various awareness programs that are being conducted, as well as by Ministry of Ayush to basically to sensitize the population regarding various misleading advertisements and all. So these are great initiatives, and
Thanks for that wonderful presentation. I think we can take one or two questions.
Yeah. I'm here. If anybody have any queries or questions regarding this Ayush drugs pharmacovigilance aspect, I will be glad to accept.
I understand there are no questions.
Thank you.
Conclude this session, and thank you once again for the speakers.
Thank you, sir. Moving forward to the felicitation ceremony, I request Professor Taruna Sharma to kindly felicitate our distinguished speaker, Professor K.C. Singhal, sir. I request Dr. Sandeep Agrawal, sir, to kindly felicitate Dr. Rabi Narayan Acharya for his excellent talk. I invite Professor Shailendra Handu, sir, for felicitating our guests. So please felicitate the chairperson.
Chairperson.
Okay.
So as we are going through all these presentations, I think we have had extraordinary sessions and, interesting presentations by experts. So the organizing committee wants to felicitate the founder of SOPI, Professor K.C. Singhal, with a small memento for his contribution to the cause of pharmacovigilance. So he is a patron of the society as well as the founder. So as a token, small token of appreciation, we would like to felicitate him with a small memento.
Thank you, sir. A small announcement for the delegates. After the next session, we will have a group photograph. So I please request you all to have the group photograph first, and then we'll move on to the lunch after the next session.
Okay. Can you invite all the people for a group photograph?
So do we need to have it just now? Okay. We can have it right now. Yeah. Please, all the delegates and all the guests, kindly come up on the stage. We'll have a good group photograph.
Since we are a very small gathering, we can have it right on the stage itself.
Yeah.
Straight away. What do you say, Dr. Zia? Yeah, because
Yes, sir, we've invited
everybody.
everyone. Yeah, sir.
You know what?
Alina, this one. इसके बाद है. Now we proceed to the 9th session on environmental pharmacology.
I now invite the chairpersons for this session, Professor Shalini Rao, Head and Content of Community Medicine, AIIMS Rishikesh.
Now I request our chairpersons to kindly preside over this session.
Yeah, good afternoon, everyone. I would like to invite Dr. Balakrishnan, Professor and Head of Pharmacology, to deliver his talk, please. Dr. Balakrishnan is Professor and Head of Department of Pharmacology at AIIMS Bhopal, and he has more than about 100 publications to his credit and several other achievements. So he's our next speaker, and sir will deliver his talk now.
Kindly check if sir is available online. Yes.
Sir is delivering his lecture online.
Good afternoon, everybody.
Good afternoon, sir.
I hope I am audible.
Yes,
Yes, sir.
audible, sir.
Good afternoon to the chairpersons. Good afternoon to all the delegates. I hope I am audible and visible. Are my slides visible?
Sir, your slides are visible and you are clearly audible.
Thank you. The organizers have given me the topic on environmental pharmacology and introduction. I took the liberty of changing it, modifying it to carbovigilance, that is the carbon footprint of the pharmaceutical industry. I think all of you must be hypoglycemic. I'll make this a 10-minute talk. I'll make it a short talk, and I hope all of you find this interesting. I think everybody is talking about carbon emissions, carbon footprints, and you'll be amazed to know that the carbon footprint or the carbon emissions produced by the pharmaceutical industry, right from production, manufacture, tableting, storage, transport, and then finally till the time it reaches the patient and then disposal of the drug. The carbon emissions are much more than what emissions are given out by the automotive industry. So that is the scale or that is the magnitude of the problem we are talking about. Why should the pharma industry should care about sustainability? Because healthcare itself, including hospitals, account for nearly 5% of the net global climate emissions, of which more than 1/4 is from the pharmaceutical industry. And this sector emits about 55% more Scope 1 and Scope 2. Scope 1 and Scope 2 emissions are that which can be controlled by the pharmaceutical company. So it emits more than 55% more scope 1 and scope 2 emissions as compared to the automobile sector. And that's the reason why we should be concerned. So we support a small study during COVID time, and this is the paper which was published. And we propose a new terminology called carbo-vigilance, that is to have an idea of the expanding carbon footprint of the pharmaceutical industry. If you look at data, the data is quite old. The carbon emissions in the US was 540 million, and in the UK, 32 million metric tons of carbon dioxide per year by the pharmaceutical industry, which is significantly more than the automobile industry. Please note here, I am not talking about automobile industry when the car or the truck moves on the road. It is the manufacturing process and the delivery of the car to the customer there. I am not talking about carbon emissions of moving vehicles. This is only the manufacturing sector. So we are talking about pharma manufacturing sector, right? And this is a relatively untouched topic. So we selected 12 largest pharmaceutical companies in the world, and their carbon emission data for 2017 and 18 were collated. And this we got from a voluntary disclosure project called the Carbon Disclosure Project. And this can be Googled and found out on the internet. It's easily available on the internet. And what we did was to correlate the carbon emission per year with the income, the company's income. And this financial data of the company, we cross-checked and we got the data from Google Finance. So we express the results in terms of emission intensities of carbon, that is as usual million metric tons of carbon dioxide emissions. And we arrived at a single figure. Generally they take the carbon emissions in metric tons of carbon dioxide per total revenue. Now, what happens with total revenue is that the total revenue doesn't make any sense to the pharmaceutical company or to any other firm for that matter. So we proposed a modified emission intensity, wherein we took only the operating revenue. Now, operating revenue is an interesting entity, wherein the operating revenue is not the total revenue. It is the profit, net profit made with the company after everything, after investing for the next year, etc., after paying its employees, after income tax, after everything, what can be pumped back next year is what is the operating income. So in this graph, I'll show you the results. You can see that this dark is the operating income, and this is the carbon emission. This lighter is the carbon emission. Now, you can see that industry is with less carbon emissions. For example, Roche, AIIMS, Abbott, and in particular, LAP. Industries with less carbon emissions had a higher operating income. That is, the more they tried to reduce the carbon emission intensity, the more was their operating income. They didn't lose out on the profit. Except for one major reason, that is Bayer. Bayer was the only pharmaceutical company we found which had a lot of decrease in carbon emission intensities, then had a decrease operating income also. The reason for this is not the carbon emission intensity. The reason for this are different. So in the figure, we can see that for most of companies, the more they went greener, the more was their operating income. That is, there was a positive correlation between the amount of carbon emission intensity decreased and the increase in the total operating revenue. This is just for 2 years, and it cannot be taken as an indicator for what is to come or what can be actually listened. So this is just a small study we did, and to compare the carbon emission intensities with the operating income. So what we concluded from the study was that pharmaceutical companies can successfully reduce their carbon emissions and without having a fall in their operating income. And this study can help set up a standard for carbon emission for the pharmaceutical sector. In fact, results of the study was taken and then modalities have been proposed by the European Union for making it mandatory for companies to disclose the carbon footprint. And our study has been quoted in that proposal or the panel report. So, healthcare sector can stay green, it can reduce its carbon emissions, and this sort of an audit of carbon footprint of healthcare industry is much needed in today's world. Now, this or the importance of done. So, Sanofi Aventis has started a manufacturing unit site which is totally green, right? And by 2020, they have done it with the help of solar panels, and moving on to alternate sources of energy rather than hydrocarbon or fossil fuels. Now, they have a solar plant, and by 2026, they are going to go fully green or fully carbon neutral. Novartis is acting on its supply chain, that is after manufacturing, packaging, and then transporting. The major carbon emissions are for transporting and packaging, and then discarding this items. So Novartis is committed to becoming carbon neutral by modifying this supply chain, and there are a lot of modifications involved in the supply chain, including use modes of transport which have carbon emissions. Like, for example, water. Ships are supposed to have the least amount of carbon emissions among all the mode of transport. And then recycling the packaging items like thermocol and the cardboard cartons and the blister packets. They can be recycled. That is what is Novartis' commitment. There is Philips is a medical device manufacturing company, and Philips is shifting its economy to something called the circular model. This circular model means that end-to-end, when you know that many of the most of the instruments become outdated in 4 or 5 years. So Philips takes back this instrument and then reuses, recycles parts or so that there is no disposal of this instrument and no effect on the carbon emissions. So they recycle the equipment. So this is called the circular economy of Philips. And Pharmacovigilance's sustainable supply goal chain, this includes end-to-end including packaging, transport, making of API, because it is all these are chemical reactions. So making of the active pharmaceutical ingredient itself involves a lot of carbon emissions. This is my last slide, and I leave you with an interesting fact. All of us use cars. I think there's nobody in this audience who doesn't have a car, at least a car. Many of us have 2 or 3 cars. So for comparison, one car emits 4.6 metric ton of carbon dioxide per year, and for salvaging 4.6 metric ton of carbon dioxide in 1 year, you need to plant 240 trees. So you can leave you with this very sober thought that just taking a car out every day for 365 days a year needs 240 trees to actually circumvent the carbon emissions of the car. Then you can imagine how much one pharma company, if you look at the US data in the first few slides, to actually nullify this carbon emission. This is how serious the problem is, and that is the reason why I thought I will deviate a little, not talk about your hormone.
Have we lost contact with him? Dr. Balakrishnan?
Recording in progress.
Giving me this opportunity to talk on this topic which is close to my heart. Thank you.
Thank you, Dr. Balakrishnan, for sharing your data on the study which you had done. Quite impactful that changes are being brought because of the results of your study. This session is open if someone wants to ask Dr. Balakrishnan any question. Okay, Dr. Balakrishnan, you have come out with this, how this several companies, they had, they had reduced carbon footprints. So did you explore what are the reasons other than the recycling causes? What are the steps they had taken to reduce the carbon footprints? Dr. Balakrishnan?
Yeah, the carbon emissions start right from the active pharmaceutical ingredient. So the sourcing the active pharmaceutical ingredient, tableting, and then making it into a drug, etc. So now if you look at, there are two sources of drugs. One is chemical, the other one is small biological. Drugs made synthesized chemically and drugs made in a recombinant way. If you compare both these, now they are trying to use natural synthetic pathways to produce drugs. So this is one way in which a lot of carbon emissions can be other than the recycling, packaging, transport, etc. The way the drug is synthesized, or the molecule or the active pharmaceutical ingredient is synthesized, is what many pharmaceutical companies are looking at. That is a source of major carbon emission, as well as your API in the effluents and in river waters, etc.
Thank you so much. Thank you for.
Yeah, so after this interesting talk about carbo-vigilance by Dr. Balakrishnan, which was really enticing, now I would like to request Mr. Anup Nautiyal, founder of Social Development for Communities Foundation. He is also the brand ambassador for Dehradun for the Government of India, nationwide sanitation and cleanliness competition, Swachh Sarvekshan. He is also the country manager India for iconic Swedish global clothing company H&M, Hennes and Morris, alumnus of SRCC and member of several government committees that deal with the development and climate change issues in Uttarakhand. He has led 108 medical emergency services in PPP mode, and he'll be talking about preserving environment in view of healthcare needs of the community. Mr. Anup Nautiyal, over to you, please.
आपका बहुत बहुत आभार कि आपने मुझे यहाँ पर आमंत्रित किया है, और मैं धन्यवाद देना चाहूँगा डॉक्टर पुनीत धमीजा जी को। डॉक्टर साहब हैं नहीं यहाँ पे। मैं हिंदी में अपनी बात रखूँगा यहाँ पर, और मेरे लिए बड़ा एक हम्बलिंग मोमेंट है कि मैं आज एम्स के सभागार में उपस्थित हूँ आप लोगों के बीच में। मैं कई जगह जाकर के बात रखने का मौका मिलता है, लेकिन इस किस्म की गैदरिंग में मैं शायद पहली बार इतने सारे डॉक्टर्स के बीच में, और जो आप लोग इतना बेहतरीन काम कर रहे हैं, आप लोगों के बीच में मुझे मौका मिला है। तो मैं जो बात आपके साथ रखूँगा, मैं अपने राज्य उत्तराखंड के बारे में कुछ जो चिंताएं हैं वो साझा करना चाहता हूँ। और विशेष तौर पर डॉक्टर पुनीत धमीजा ने मुझे जब इसके लिए आमंत्रित किया तो उन्होंने मुझे कहा कि उत्तराखंड में जो बायोमेडिकल वेस्ट की जो सिचुएशन है उसके ऊपर मैं थोड़ी बात करूँ, जो एक्सपायर्ड मेडिसिन हैं उसको लेके थोड़ी बात करूँ। तो ये उत्तराखंड का, और ये मेरा सब्जेक्ट था, आप देख सकते हैं जिसको, प्रिजर्विंग एनवायरनमेंट, आगे बढ़ाना है इसको ज़रा। तो ये आप एक बार ये ट्वीट पढ़ लीजिए, ये जो लिखा हुआ है उसके बारे में मैं थोड़ा सबको बताऊंगा कि इसका क्या बैकग्राउंड है। जैसा आपने पढ़ा, ये वेस्ट डिस्पोजल गंगा नदी को लेकर एक उदाहरण है, और सामान्य तौर पर जो हमारे ब्यूरोक्रेट्स होते हैं वो बिल्कुल ओपनली किसी भी बात को ऐसा डिस्क्लोज नहीं करते, लेकिन ये पिछले साल 26 अप्रैल का रात के 9:15 बजे का चमोली के जो डीएम हैं, और यहाँ पर मैं ये भी जिक्र करना चाहूँगा क्योंकि आप में से हो सकता है कुछ लोग उत्तराखंड के बाहर के हों, कि जो हमारा राज्य है, हमारे राज्य में 13 जिले आते हैं और उन 13 जिलों में से 9 जिले जो हैं उनको मूलतः पहाड़ी जिला कहा जाता है। और उत्तराखंड राज्य के 2 हिस्से हैं मोटे मोटे, एक गढ़वाल क्षेत्र और एक कुमाऊं क्षेत्र। तो ये चमोली जो जिला है बड़ा बद्रीनाथ जी के लिए, वैली ऑफ फ्लावर्स के लिए, तमाम इन चीज़ों के लिए मशहूर है। और ये जैसा आपने पढ़ा, गौचर एक छोटी नगर पालिका है, वहाँ पर वाहन के द्वारा नदी में कूड़ा कचरा गिराया जा रहा था। अब ये नदी कौन सी है? गंगा नदी है। तो ये गंगा नदी में ये बहाया जा रहा था, और यहाँ पर ये एक बहुत बड़ी बात आती है कि उत्तराखंड में किस तरीके के साथ में कचरे का निस्तारण हो रहा है या डिस्पोजल हो रहा है। और अब सवाल आपके जहन में आ सकता है कि भाई ऐसी क्या परिस्थिति हुई कि डीएम साहब को जो है वो ये ट्वीट करना पड़ा। इसकी बात ये थी, ये इसमें डेट नहीं है, लेकिन पिछले साल की बात है, 24 अप्रैल को एक ये ट्वीट वायरल हुआ। इनफैक्ट एक वीडियो है, और जैसा आप पढ़ सकते हैं कि जिन्होंने ट्वीट किया उनको पता नहीं था ये कौन सी जगह है, लेकिन इस तस्वीर में आप नीचे गंगा जी को देख सकते हैं और ऊपर आप इस कचरे को देख सकते हैं। तो ये एक बहुत 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 गंभीर समस्या भी है, चिंता भी है। और यहाँ पर मैं आपके साथ में एक और उदाहरण साझा करना चाहूँगा उत्तरकाशी शहर का, जो कि हमारे राज्य का एक बहुत प्रमुख शहर है। और उत्तरकाशी शहर जो कि गेटवे है यमुनोत्री का और गंगोत्री का, जब आप उत्तरकाशी शहर में घुसते हैं तो बिल्कुल गंगा जी के तट पर जो है कचरे को गिराया जा रहा है। तो ये वेस्ट मैनेजमेंट की सिचुएशन है। पिछले साल जब चारधाम यात्रा, आपको पता है हमारे राज्य के लिए बड़ी महत्वपूर्ण है, और चारों धाम इस बार भी खुल चुके हैं। पिछले साल केदारनाथ जी के जो कपाट खुले थे 6 मई को खुले थे, और मात्र 14-15 दिन के बाद में 22 मई को ये तस्वीरें वायरल पूरे देश में हुई थी, और आप इन तस्वीरों को देख सकते हैं। और ये क्या स्थिति जो है इस समय ये केदारनाथ जी की क्षेत्र की है। इनफैक्ट लास्ट ईयर जो थी सिचुएशन इतनी क्रिटिकल हो गई कि ऑनरेबल प्राइम मिनिस्टर को अपनी मन की बात में, और ये जो मन की बात पिछले साल हुआ था ये 29 मई को हुआ था, 29 मई 2022 को, और उन्होंने अपने मन की बात में केदारनाथ में इस फैले हुए कचरे का उल्लेख किया था, और उन्होंने अपनी चिंता जो थी इसमें व्यक्त की थी। तो ये मैं इन उदाहरणों को आपके सामने इसलिए रख रहा हूँ ताकि आप कचरा प्रबंधन, क्योंकि जो बायोमेडिकल वेस्ट है वो भी हमारे वेस्ट का एक कंपोनेंट है, आप उसकी स्थिति से कुछ हद तक उससे परिचित हो पाएँ। आगे बढ़ते हैं। तो बायोमेडिकल वेस्ट और एक्सपायर्ड मेडिसिन्स का, जैसा मैंने कहा, मैं मुख्य तौर पर पहाड़ों की बात कर रहा हूँ, और जो हमारे 9 पर्वतीय जिले हैं, मैं अगर उनका भी जिक्र करूँ यहाँ पर, चमोली जिला, उत्तरकाशी जिला, टिहरी जिला, रुद्रप्रयाग, पौड़ी, अल्मोड़ा, पिथौरागढ़, बागेश्वर और चंपावत, हमारे 9 जिले हैं। और एक इतने बड़े राज्य में जिसका क्षेत्रफल लगभग 55,000 स्क्वायर किलोमीटर है, हमारे पूरे राज्य में सिर्फ 2 अप्रूव्ड बायोमेडिकल वेस्ट कंपनीज हैं। तो एक कंपनी जो है वो रुड़की में है, और रुड़की के माध्यम से, रुड़की जो कि ऋषिकेश से ज़्यादा दूर नहीं है, रुड़की की जो कंपनी है उसको पूरी जिम्मेदारी पूरे गढ़वाल मंडल की दी गई है, यानी 7 जिलों की जिम्मेदारी रुड़की की कंपनी को है, और कुमाऊं मंडल में गदरपुर एक शहर है उसको दी गई है। अब आप सभी लोग, चाहे आप उत्तराखंड में रहते हैं या उत्तराखंड के बाहर रहते हैं, आप सभी को पता है कि यहाँ पे कितनी बरसात होती है, यहाँ पे एक्सेस की कितने बड़े चैलेंजेस हैं, कितनी सारी आपदाएं हमारे प्रदेश में आती हैं। 12 महीने में से लगभग कई महीने तो जो हैं सड़कें ही बंद रहती हैं। तो जब ऐसे लैंडस्केप में और इतनी दूर, भाई कहाँ रुड़की और कहाँ ये छोटे छोटे शहर और छोटे छोटे कस्बे, तो वहाँ पर किस किस्म से बायोमेडिकल वेस्ट का निस्तारण होता होगा, ये अपने आप में एक सोचने वाली बात है। इस प्रेजेंटेशन को लेके मेरे एक पुराने साथी हैं जो कि उत्तरकाशी के एक सरकारी अस्पताल में काम करते हैं, तो मैंने उनसे भी फ़ोन पे बात करी। मैंने कहा डॉक्टर साहब थोड़ा मुझे बताएंगे कि वास्तविकता क्या है आपके अस्पताल में, आप लोग किस तरीके के साथ में करते हैं। तो वो कहते हैं कि भाई ठीक है, हमारा टाई अप तो है रुड़की के साथ में, लेकिन कई बार वो रुड़की की गाड़ी नहीं आती है, तो हम लोगों को एक कमरे के लिए बोला गया है कि भाई कमरे में जो है आप अपना बायोमेडिकल वेस्ट रखिए। लेकिन कई बार क्या होता है कि बायोमेडिकल वेस्ट इतना ज़्यादा हो जाता है कि हम फिर अपनी बात को पूरा बोल नहीं पाए। तो मैं खैर उनकी बात समझ गया था, उनके कहने का क्या अर्थ था। और एक ये बड़ी, जो आप तीसरा मेरा पॉइंट देख रहे हैं यहाँ पे, ये भी एक बड़ी स्वाभाविक बात है, एक सवाल है कि भाई कोई भी अस्पताल कितने समय तक बायोमेडिकल वेस्ट को अपने कैंपस में रख सकता है। वो भी एक, जिसे कहेंगे, एक डबल एज स्वॉर्ड है। जब हम उत्तराखंड की बात करते हैं, गंगा नदी की बात करते हैं, तो बार बार नमामि गंगे का जिक्र आता है। लेकिन नमामि गंगे की उसकी वैसे तो खैर उसका बहुत सारी चीज़ों को लेकर उसका फोकस है, लेकिन जब हम उसको बायोमेडिकल वेस्ट के दृष्टिकोण से देखेंगे तो नमामि गंगा जो है वो एसेंशली जो सीवेज है या ड्रेन्स हैं उनको रोकने में उसका ज़्यादा बड़ा उसका फोकस है, हम ये कह सकते हैं। और वैसे भी ये जो कचरे को फेंका जा रहा है तो नदी के ऊपर से फेंका जा रहा है। अब उसमें कौन कौन सा कचरा डाला जा रहा है वो कॉमन सेंस की बात है, खुद हम उसको अज्यूम कर सकते हैं। और अगर मैं इसको समराइज करूं अपने 5वें बिंदु को, तो बोलना खैर बहुत अच्छा नहीं लगता है, लेकिन जो ज़्यादा बड़ी तस्वीर जो पहाड़ों की हमारे राज्य में है, या तो उसको जला दिया जा रहा है कचरे को, या उसको फेंक दिया जा रहा है, या उसको डिग कर दिया जा रहा है। तो ये एक हल्का फुल्का एक परिदृश्य है। लेकिन बात यह है कि देखिए, खाली हम चुनौतियों की बात करें और कुछ समाधान न तलाशें, वो भी हमारी बात पूरी नहीं होती। तो अगली स्लाइड में मैं कुछ 10 किस्म का एक 10 पॉइंट प्लान फॉर बायोमेडिकल वेस्ट इन उत्तराखंड देना चाहता हूँ, सामने रखना चाहता हूँ। बड़ा सिंपल सा है ये, कोई बड़ा टेक्निकल नहीं है। लेकिन इसमें पहला बिंदु यही है कि भाई ये जो हमारी सरकार है, ये जो हमारी अथॉरिटीज़ हैं, वो आइडेंटिफाई करें कि भाई इसके स्टेक होल्डर्स कौन हैं, इस बायोमेडिकल वेस्ट के और इस पूरे एक्सपायर्ड मेडिसिन के कौन कौन स्टेक होल्डर्स हैं, ताकि उनसे बात की जाए। दूसरी बात, अवेयरनेस और एजुकेशन। ये जो हमारा 8वां सत्र हुआ था उसमें भी डॉक्टर साहब ने एक अवेयरनेस को लेकर बात करी थी। इसके अलावा मुझे लगता है कि लोकलाइज्ड और डिसेंट्रलाइज्ड इंफ्रास्ट्रक्चर के ऊपर जोर देने की ज़रूरत है। जैसा मुझे लगता है आप मेरी बात से सहमत होंगे कि रुड़की से पौड़ी या चमोली या तमाम इन जगहों तक जो है वो गाड़ी कैसे जाएगी, कैसे पहुंचेगी। तो जब तक हम अपने पहाड़ी ज़िलों में लोकलाइज्ड इंफ्रास्ट्रक्चर को स्थापित नहीं करते इस लोकल बायोमेडिकल वेस्ट मैनेजमेंट का, तब तक कुछ होने वाला नहीं है या कुछ मजबूत नहीं होने वाला। ट्रेनिंग की आवश्यकता है और सोर्स सेग्रीगेशन। इनफैक्ट जब हम वेस्ट मैनेजमेंट की बात करते हैं, वेस्ट मैनेजमेंट की ABC ही जो है वो सोर्स सेग्रीगेशन को लेकर के होती है, चाहे हम नॉर्मल वेस्ट की बात करें, किसी भी वेस्ट की बात करें। और नेचुरली फिर उसी से जुड़ा हुआ जो मुद्दा और मसला ट्रांसपोर्टेशन का, डिस्पोजल का, एनफोर्समेंट और मॉनिटरिंग का और डेटा जनरेशन का। मुझे लगता है आप मेरी इस बात से सहमत होंगे कि हमारे देश में जो डेटा है, पर्टिकुलरली मैं अगर कचरे के डेटा की बात करूं, तो उसमें कई सारी इनएक्यूरेसीज हमें नेशनल लेवल पर भी देखने को आती हैं। तो खैर राज्य की तो खैर क्या बात करनी, लेकिन इसके ऊपर भी काम करने की आवश्यकता है, क्योंकि जब तक हमारे पास में डेटा नहीं होगा तब तक हम आगे प्रॉपरली नहीं बढ़ पाएंगे। और 9वां बिंदु भी बड़ा स्वाभाविक है। आपको पता है कि जब भी कचरे को लेके बात होती है तो हमेशा एक जन आंदोलन को लेके बात होती है कि भाई कम्युनिटी के पार्टिसिपेशन के बिना कुछ भी संभव नहीं है। जो मेरे से पहले अभी जो प्रेजेंटेशन था, डॉक्टर साहब बात कर रहे थे, तो उन्होंने EPR को लेके बात रखी थी, फिलिप्स के मॉडल को लेके बात रखी थी। और आज पूरी देश में, दुनिया में EPR को लेके, सर्कुलर इकोनॉमी को लेके बात होती है। मुझे लगता है कि पॉलिसी टूल्स का भी और रेगुलेटरी फ्रेमवर्क का होना ज़रूरी है। तो ये एक बड़ा, जैसा मैंने कहा, एक बड़ा सिंपल सा एक मॉडल है, बट चीज़ों को बिना ज़्यादा कॉम्प्लिकेट किए हुए अगर इसमें गंभीरता के साथ में काम करना है तो कुछ ऐसे बिंदुओं को आगे लेकर के बढ़ना पड़ेगा। दोस्तों, मैं अपनी आखिरी स्लाइड पे आ रहा हूँ और मैं, ये एक संजोग है कि आज 12 मई है, तो मैं आज अखबार पढ़ रहा था और मुझे अमर उजाला अखबार में एक कतरन दिखाई दी। मुझे लगा कि मुझे आपके साथ साझा करना चाहिए। लेकिन उस कतरन पर आने से पहले, उस अखबार की खबर पर आने से पहले मैं आपको उत्तराखंड की एक खासियत बताना चाहूँगा। और मुझे लगता है कि वो उत्तराखंड की एक बहुत बड़ी उपलब्धि है, बहुत बड़ी अचीवमेंट है, और वो अचीवमेंट उत्तराखंड की फार्मा इंडस्ट्री है। उत्तराखंड की फार्मा इंडस्ट्री का एक बहुत बड़ा योगदान पूरे देश की अर्थव्यवस्था में, पूरी दुनिया में है। आज ये अनुमान लगाया जाता है कि हमारे देश की 20% से ज़्यादा जो मेडिसिन्स हैं वो उत्तराखंड राज्य में बनती हैं। उत्तराखंड राज्य में फार्मा इंडस्ट्री में 1 लाख से ज़्यादा लोग जो हैं वो इसमें कार्यरत हैं, और बहुत सारी 300 से ज़्यादा यूनिट्स हैं। तो ये कुछ पुराने आंकड़े हैं, तो उसका एक बड़ा अहम योगदान, जैसे मैंने कहा। लेकिन आप ये आज के अमर उजाला की अगर आप खबर पढ़ेंगे तो फिर आपको पढ़ करके जो है एक ये आपको अच्छा नहीं लगेगा इस खबर को पढ़ के। और ये देहरादून शहर की खबर है। तो देहरादून शहर के पास में एक सेलाकुई इंडस्ट्रियल एरिया है जहाँ पर बहुत सारी मेडिसिन्स बनती हैं, और आप खुद पढ़ पा रहे हैं इसको कि नदी में मिला दवाओं का जखीरा, और ड्रग इंस्पेक्टर बोले कंपनियां फेंक रही एक्सपायर्ड दवाइयां, होगी जांच। तो इस सोबरिंग नोट पर मैं अपने इस प्रस्तुतिकरण को यहाँ पर मैं समाप्त करना चाहूँगा। मुझे लगता है कि आप लोगों ने तो खैर बहुत बड़ी बड़ी बातें करी हैं डॉक्टर साहब, और आप लोग शेयर कर रहे हैं इसको। तो आई होप दैट दिस वेरी वेरी सिंपल प्रेजेंटेशन मेड सम सॉर्ट ऑफ सेंस, एंड यू नो it somehow or the other fitted into your larger canvas, and you know what you've assembled here to discuss and deliberate and finally come out with some solutions. तो मैं एक बार यहाँ पे डॉक्टर पुनीत धमीजा मुझे दिखाई नहीं दे रहे, बट वंस अगेन आई एम वेरी वेरी ग्रेटफुल कि उन्होंने मुझे एक इतनी ऑगस्ट गैदरिंग में आप लोगों के बीच में इन्वाइट किया है। थैंक यू सो मच वंस अगेन, एंड ऑल द वेरी बेस्ट। ये मेरा ईमेल आईडी है, हाय वंदना। तो मैं पुनीत धमीजा जी का, ये मेरा ईमेल आईडी है। आप में से अगर कोई भी मुझे, मेरा बड़ा सिंपल ईमेल आईडी है, आप में से मुझे कोई भी अगर संपर्क करना चाहें, मेरा नाम अनूप नौटियाल है। और आपके कोई भी सवाल कभी भी उत्तराखंड को लेकर के हों, आप बिल्कुल निसंकोच और बेहिचक जो है आप मुझे संपर्क कीजिएगा, और हम निश्चय। डॉक्टर साहब वहाँ पर आ गए। डॉक्टर साहब, मैं आपको एक और कारण से बधाई देना चाहूँगा, एंड दिस इज समथिंग पर्सनल। So my father retired in the year 1979. So I did my schooling initially in Delhi, and फिर हम लोग देहरादून आकर के बस गए थे। तो वंदना एंड मी वर क्लासमेट्स इन स्कूल, और ये 1979 है, और 2023 से तो 44 साल के बाद में आज हमारी मुलाकात जो है वो एम्स में हो रही है। तो मैं उसके लिए भी डॉक्टर साहब का बहुत बहुत धन्यवाद और आभार व्यक्त करना चाहूँगा। थैंक यू सो मच।
Thank you, Anup ji, for speaking on such an important topic. Disposal of biomedical waste as well as the solid waste that we have at our homes, which many times contains biomedical waste. जो common man होता है वो अपना जो biomedical waste होता है, उसने कुछ दवाई खाई, कुछ expired दवाइयाँ हैं, तो वो general waste में डाल के dispose of कर देता है. There are other waste also which are considered as biomedical waste, जो general waste में dispose जाते हैं home basis पे. हॉस्पिटल्स की सिचुएशंस आपने बताई, जो कि वाकई बहुत ही दुखदाई है एक्चुअली, सही शब्दों में अगर बयान करें। एक्शन लेने की जहाँ तक बात है, बहुत अच्छे 10 पॉइंट्स अनूप जी ने ले आउट किए। इन 10 पॉइंट्स के अपने चैलेंजेस हैं, सारे 10 पॉइंट्स में अपने अपने चैलेंजेस हैं। स्टेक होल्डर आइडेंटिफिकेशन करने का अपना है एक चैलेंज, ओनरशिप लेने का है इशू। People don't want to take ownership about issues such as biomedical waste disposal. Waste disposal का ownership लेते नहीं लोग, generate करने का ownership रहता है, but आगे workup करने में मुश्किल है. We have conducted a study on e-waste generation and knowledge about e-waste in Srinagar, Garhwal, and also I tried to do an attempt of disposing e-waste properly within the campus. But I failed miserably, truly speaking, because I didn't get support. And as you told, there are only 2 sites जो ये सारा waste disposal के लिए help करती हैं, but we didn't get administrative support. So it is a real challenge if we really want to do something in a very systematic manner currently in the setup. So all of us have to really work hard and cooperate and maybe collaborate for working in this area so that we can take this thing ahead, which is affecting the lives of all of us. So we are affected, not knowing that we are affected, we are still affected by it. So really thankful for you to talk on such important topics, especially at this platform, so that it goes to people and we are able to make some policy for it. Thank you so much.
I would like to thank the organizers and the pharmacological vigilance body actually to include such an important stakeholder. Because when you as comprehensively, you have started doing this pharmacovigilance, and this is one of the most, another important aspect which should not be ignored. And I would like to ask, what are you, I like to ask the members, what exactly are you doing about this awareness for this eco-pharmacovigilance? This is going to be actually the next step ahead, because you will have a Herculean task. You're looking at the hospitals and you are, taking care of the pharmacovigilance there. But an equally important responsibility would be generating that awareness among the community.
Okay. Thank you, Anup Notialji. It was such an inspiring and such a sobering, presentation.
Thank you.
Because I think this gives us, a clue to, most of the time we are focusing on medicines and patients. So we administer the medicines to patients, and of course pharmacovigilance, the primary focus is drug safety. But then when we look at a wider picture, then all these important issues come up. That we are linked to the environment, and we are linked to the biological system, the ecosystem. So everything is interrelated. And our health, disease, it can be impacted by environment. So now we are looking at the bigger picture, and these things are evolving, like eco-pharmacology. We had the previous speaker talk about vigilance. So these are evolving areas, because ultimately they are going to impact our health. And they are going to help impact the whole ecosystem, the environment. And I think we have to, embrace all these newer areas which are emerging areas, and more and more sensitize the administration, the public, and the people in general. Thank you so much for your presentation.
Thank you.
Thank you, Dr. Handu, and thank you, Dr. Notialji.
Thank you.
Thank you, sir. I express my heartfelt gratitude to Professor Balakrishnan and Mr. Anup Nautiyal for their presentations. Now I request our chairpersons to kindly felicitate Mr. Anup Nautiyal, sir. Sir, kindly be invited. I invite Dr. Puneet Dhamija, sir, too. So please come. May I call upon Professor Vandana Roy, ma'am, to kindly felicitate our chairpersons. Thank you all. With the end of this session, I invite you all for delicious lunch served at level 2 of this auditorium. We will begin next session at 2:45 p.m. Hello. Welcome back to the 10th session of the second day of Sopicon, themed Vaccine Safety and Safety Outcome Research. I invite our chairpersons for this session, Professor Rangil Singh Raina, Principal, Haridwar Medical College, and Professor Nilotpal Chaudhary from Department of Pathology, AIIMS Rishikesh. I welcome you, sir. Now I request our chairpersons to kindly preside over this session by introducing our first speaker, Dr. Kapil Kudala.
Okay, good afternoon, everybody. So, first lecture of this post-lunch session is by Dr. Kapil Kudala. So, he is senior consultant in IQVIA. He has 30 number of publications, and about his major achievement award is a travel grant to attend international conference held by International Diabetes Federation. He is associated with the Asian Association for the Study of Diabetes, Internal Society of Pharmacoeconomics and Outcomes Research. He is also associated with the International Society for Pharmacoepidemiology. So now I think Kapil Kudala is online. Can you hear Mr. Kapil? Am I audible to you? Some connectivity issue is there. So please
Dr. Kapil Kudala?
Yes, we can hear you now.
Yeah, so we can also hear you.
Okay, Kapil Kudala, please, over to you. Please start your presentation. And we have 20 minutes for this presentation.
Here, good.
Technical team?
Can you hear me now?
Yes, sir. Yes, sir, we can hear you. Yeah, so you can start sharing your screen.
I'm already presenting my screen.
Okay, yes, sir, we can see. I request chairpersons to kindly ask the speaker to start.
Okay. Mr. Kapil, please, you can start now.
Thank you, sir. Okay. Today my presentation is on, why safety is important in terms of health economics and outcomes research. I'll be majorly focusing upon how the safety is included in the health economic analysis, in the health economic modeling, and also how and why. Why is also an important aspect to understand. Why the inclusion of safety in the health economic modeling is important. Okay, I'll be touching upon these two things. So this is a brief disclaimer of my topic. And as I said, my aim of this presentation is only to demonstrate how and why safety of products should be considered while performing any cost-effectiveness analysis. But I will not be touching upon, in detail various aspects of cost-effectiveness analysis. Okay. To achieve this goal, I will be briefly introducing to HTA, what is health technology assessment, and I will briefly touch upon what is cost-effectiveness analysis, how to interpret, because these two things are necessary to understand, how safety will be included in cost-effectiveness analysis. Okay. So after that, I will explain the importance of safety, and also I will briefly walk you through a case study. Okay, a real-time case study which I faced. So I will be presenting that case study. Okay, as everyone knows that the clinical trials start to establish the safety and efficacy of a drug in patients for various conditions. Okay, this has been done by using phase 1, phase 2, and phase 3 clinical trials. But once the drug get registration and when it got into the market, okay, there come the value add scheme. Okay, so basically in developed countries like UK, US, and Australia and other countries, these drugs are being reimbursed by the HTA body and by the Ministry of Health of their respective countries. So this is being achieved by HTA submissions. Usually HTA submissions are done by manufacturers, pharma manufacturers. In this HTA submission, we have multiple aspects, and one of the aspects is cost-effectiveness analysis and budget impact analysis. Okay, so this analysis, cost-effectiveness analysis, is being done to show the value of this drug in terms of how the new drug, the intervention is faring out with the comparator, both in the terms of the cost and also effectiveness. And recently, if you have seen, many of the drugs are being priced based upon value-based pricing. Value-based pricing is nothing but it is like the price premium which an innovative drug is being offered depending upon how much value it adds to the existing comparator, existing best supportive care. Okay, so based upon that only, the premium pricing will be done, for the innovative drugs. So to achieve that, we need to do cost-effectiveness analysis and budget impact analysis. Okay, so this is why we need to do this value-based pricing or the cost-effectiveness analysis, because if you see in various economies, all the resource allocation is very constrained. The amount that is that we have to fund for healthcare is very limited. Okay, so, but the needs and the wants of the patients and for the country is being increasing. Day to day, we are seeing a surge in the prevalence and incidence of various diseases that includes both acute and chronic diseases. But if you see many of the countries, the healthcare budget is hardly 5% of their GDP, maybe lesser than that. So, so to enter this, what we need to do is we need to take a very informed decision. It doesn't matter how much you are, funding it for the healthcare, which is in our hand. Definitely, there is one thing which we can control. That is how you spend it, how wisely you spend it for healthcare, on investing in multiple interventions or multiple health programs. Okay, so this is where the cost-effectiveness analysis and the HEOR and HTA comes into place. if you see, the main motto of HTA and HEOR is to maximize the utility of of the given intervention. Okay. For example, in front of the healthcare payer, there is a decision problem came into place whether to fund for deworming of 20 lakh children or whether to fund a new intervention for the breast cancer of one woman. Okay, which one to choose for the given healthcare budget they have. Okay, so this kind of decision problem, this healthcare economic modeling can help, and HTA can help in solving that. Okay, so this is a very brief introduction of HEOR and health technology assessment which I want to give. And I will quickly jump into the cost effectiveness analysis or health economic evaluation in a broad way. Okay, so what is health economic evaluation? When do we need it and how we do it? Okay, so cost effectiveness analysis is nothing but it is a comparison of two interventions, maybe intervention X and intervention Y, or it may be the comparison of health program 1, health program 2, or it can be a comparison of two diagnostic tests, or it can be a comparison of two prevention programs. It is just it is comparison between innovative intervention versus existing program. Okay, so we compared these two strategies in two ways. One is in terms of the cost, and another is in terms of consequences. Consequences are nothing but the healthcare outcomes, like number of deaths averted, or number of migraine episodes prevented. These are the different kinds of outcomes. Apart from that, we also have humanistic outcomes, that is nothing but quality adjusted life years. Okay, so these are the different outcomes we can model in the cost effectiveness model. Okay, so what we do is we assess the cost and outcomes of one intervention, and also we assess the cost and outcomes of the other strategy or intervention also. And we do the comparison of these cost and consequences between these two strategies. And we come up with the outcomes, which I will show in a couple of slides. And this is nothing but the economic evaluation. We call it as economic evaluation. There are several types of economic evaluation which we can do. There's, like cost effectiveness analysis, cost utility analysis, cost benefit analysis, and others are there. Okay. But for now, we will be focused on cost utility analysis. Okay. So what is cost utility analysis? It's cost utility analysis nothing but when we take consequences as the quality adjusted life years, then this kind of economic evaluation will become the cost utility analysis. As I said, the outcomes, the health quality of life. The quality of life is usually measured quantitatively in terms of utility. Utility can have a negative scale, positive, up to 1. Okay. Typically, we say that a person with utility of 1, he is having the absolute perfect health. Okay. And a person with a utility of 0 is nothing but dead. And some patients can have a negative utility also. I mean, like negative quality of life also. That means that the patient has suffering more than worse than the death. Maybe the patient who is on terminal ill care, that kind of patients where the quality of life is really deteriorated. In those patients, the utility can go to negative. Okay. If you can see the graph below, okay. So on the x-axis we have time. Okay. On the y-axis we have quality of life. Okay. So we are not talking about the extreme cases, but here we can see that the quality of life can range from 0 to 1 in the y-axis, and in x-axis it can vary from 0 to infinity time. Okay. When we block the area under the curve of, if you assume this is a curve, okay, and the area under curve is nothing but the quality adjusted life years, okay. So quality adjusted life years are nothing but it's a product of, the quality of life and the time spent by the patient with that quality of life. This is nothing but quality adjusted life years, okay. Suppose we are comparing two treatments, that is standard of care, and another is with new treatment. If you can see, the white area is nothing but the quality adjusted life years with standard of care. Okay, and if you could see this light blue color, area under the curve, this is nothing but the quality adjusted life years of the neo treatment. When you compare the quality adjusted life years between these two treatments, you can see there is an increment. There is a difference. This incremental blue color area is nothing but this is a value added. This is the quality adjusted life years added by the new treatment comparison to the standard treatment. Okay, so this is how a drug can improve the quality adjusted life years of the patients. Okay, so now comes, as I said, any of the health economic analysis, including cost utility analysis, the outcome, we call it as ICEF, incremental cost effectiveness ratio. Okay, so it is calculated using this formula. So what we do is, as I said in in this graph, said we calculate the total cost of the one intervention and the consequence of the other intervention, and similarly we do it for the second intervention also. And we put this in this formula. By the quality adjusted life years gained by the treatment minus quality adjusted life years gained by the standard care. When we take the ratio of this incremental cost and incremental effects, we get ICER, that is nothing but incremental cost effectiveness ratio. Okay, so different countries have different thresholds. What is this threshold? For example, UK NICE have a threshold of 30,000 GBP. 30,000 pounds. If the ICER, this comparison lies below 30,000, okay, below 30,000 pounds, it means that the new treatment or the intervention I touched upon, the cost effectiveness analysis and its, interpretation. I can just quickly walk through this, example. For example, if the new treatment is costing $20,000 and the standard of care is costing $10, and the quality is gained by the treatment is 3.5 years, but the quality gain on the standard care is 3. Okay, here the incremental cost is nothing but 20 minus 10 is 10,000 divided by the incremental quality is nothing but 3.5 minus 3, that is 0.5. Okay, that is 10,000 by 0.5. Okay, so that is how we calculate the ICER. Okay, the ICER is about, now it is about 20,000. So if we consider this as the health economic analysis for the UK, the treatment, the intervention is found to be cost effective because the threshold for the UK is 30,000 pounds. Okay, so this new treatment is cost effective and it can be reimbursed. So if we show the cost effectiveness, if we are able to establish the cost effectiveness of the new drug, then the Ministry of Health of UK or any other healthcare payer will fund for the reimbursement of those drugs. So that is how the the system works. Okay, now I will. Okay, so one of the most important aspects that is usually undermined or usually neglected in these kind of HTA submissions or in these types of cost utility models is the safety aspect. Okay, safety is very very much undermined in many of the health economic models because it is not usually there are no standard guidelines how we incorporate safety into these models. Although we have several recommendations by ICER and the CADIT and other HTA boards, but there is no universal agreement and there is no clear-cut guidelines how the safety should be included in the HTA submission. That's why you see there is a huge uncertainty. Okay. There's a study done by Lu et al. In 2023. So what these guys have done is they reviewed all the cost effectiveness analysis that were published in oncology about 467 such kind of cost utility analysis. Okay, what they try to do is they try to see how these in these models the utilities related to adverse events or models. Okay, so they have seen that only 54, that is only half of the 467 studies has bothered about the safety in the model. Okay, and it is pretty sure that compared to safety of two intervention is definitely be assured that it will be same in both the interventions. Okay, at the time if you are not sure, we definitely need to model the safety of the drugs into the model. But however, you see there are only half of the models that has included the adverse event component into the model. Okay. Out of them, only 6% of out of this 54%, only 6% of analysis has followed the recommendations by the major guideline bodies like ISPOR. Okay, only 6%. That is very, very, very less. And as we expect, to show the real value of a drug, we need to make it, the 100% compliance. But we can see only 6% of the studies that has shown. So this shows that the safety is undermined in most of the published cost-effectiveness analysis. Okay, and there are, if you see multiple aspects, these analysis, constantly undermine the, inclusion of safety into the model. Okay, just jumping on to, so how, safety of a drug is included into the cost-effectiveness analysis. I will talk upon that. Okay, the safety component in the cost-effectiveness analysis can impact or influence in 3 different ways. It can increases the cost to the patient, because once a patient suffers with adverse event, okay, it can be serious adverse event or a mild or moderate adverse event, okay, non-serious adverse event, okay. So once a patient has adverse event, or they might need a physician consultation. So this increases the burden of the patient in terms of the to manage adverse event. They will incur some cost. Okay, so this will also be included into the model, and this also have influence on quality of life of patients. Once a patient have adverse event, they deteriorates, and they are included in the model. And apart from that, if the patient has a serious event, they can lead to treatment discontinuation. So once the patient discontinues the treatment, they are losing the potential value of the new drug. So this is how the safety is included in the cost of analysis in 3 different formats. So what are the different inputs that are required to model safety in the CE models? We're requiring the incidence of adverse events, okay, and what is the severity of those adverse events in those drugs. And also we need to understand what is the impact of quality of life, okay. Generally, we use the term disutility here. Instead of utility, we use disutility. Disutility is nothing but, reduction in the quality of life. To what extent, the quality of life being reduced because of that adverse event. Along with that, we also need the duration of adverse event, and we have also need to understand what is the adverse event management cost. Okay, so these are the different inputs that are required to model the safety in the cost-effectiveness analysis. I will briefly touch upon, the calculation methods now, like how the safety is being included with these inputs. For example, for total cost estimation of a particular intervention, it will, the model will include both drug acquisition cost, the drug cost, what costs that the patient spend to purchase the drug, administer the drug, and monitor the drug. Along with that, the patient can incur the disease management cost, okay, to manage the disease. Apart from that, there is an increase in the cost because of the adverse events also. A drug having higher incidence of adverse events will incur higher cost,
Mr.
but
Kapil?
a drug with lower, adverse event
Hello,
management
Mr.
cost will have
Kapil?
a lower total overall cost. So adverse event management cost
Mr.
is usually
Kapil, you have
calculated.
3 more minutes. Please try to compile within this period.
Okay,
Sorry for interruption.
Yeah, no issues. Okay, so this is how adverse event management cost is calculated based upon the incidence and the cost to manage each adverse event. Similarly, we have for the disutility. Let me jump directly to the case study. So the case study here I'm presenting is for multiple sclerosis. The patients having the multiple sclerosis, and the intervention is natalizumab. Okay, this is the new drug. This is the new drug, and we have a comparator to the best supportive drug. Just taken a random word instead of using the actual word. So I just said XLE mode. This is a best supportive care. This is already in the market, and pharma manufacturer wants to launch natalizumab into the market. Okay. So the outcomes will be the typical cost-effectiveness outcomes. Okay. So PML is a common adverse event. It's a rare and serious adverse event that incur patients receiving the natalizumab. Okay, that is our intention. Okay, the incidence of PML in patients receiving natalizumab shows that the real-world studies shows that the incidence can range from 0.8 to 0.1%. It is quite wider in range, 0.8% to 2.1%. Okay, if we assume that 0.8% as the PML risk of PML of adverse event in the patients of natalizumab, these are the results. These are the other results. If you could see, there is a the difference between these two interventions, natalizumab and Axlimod. The adverse event disability is higher for natalizumab because of PML. At the same time, AE cost is higher for natalizumab. So I want to focus on the here. Okay, the ICER using 0.8% adverse events of PML can see that it is like 29,000, which is below 30,000, and the intervention is, then we can conclude as cost effective. But however, as I said, the real world evidence studies showed that the risk of PML can ranges from 0.8 to 2.1%. Okay, so if we increase the risk of PML from 0.8 to 1.5%, okay, just one input, you could see the ICER jump from the 29,000 to 41,000. It is way beyond the threshold. Okay, now if we by just increasing it to 1.5, then natalizumab is no more cost effective. And if I switch to highest range, the range of the adverse event of PML to 2.1, the ICER jumped to 50,000. Okay, there is again a huge increase in ICER. Okay, so this shows how important is safety in these cost effectiveness models. It is very, the modeler should take utmost care in understanding the the literature available for the safety of that particular drug, and also they need to they need to take the utmost evidence using the meta-analysis or network meta-analysis, and also they need to do proper safety analysis to demonstrate the value of the drug. Yep, this is all I want to present for today. So. Yes.
Okay, thank you, Mr. Kapil, for very precise and nice presentation. You have very clearly explained about the role of safety in this cost effectiveness model. So now it's open for the question session. Anybody, if have any query? Okay, Mr. Kapil, so we don't have any questions from the audience. So thank you very much again for the nice presentation. So now we are. Thank you very much. Now we have another lecture. Another speaker, Dr. Savita Verma is there. She is professor in the Pandit B.D. Sharma Postgraduate Institute of Medical Science, PGI, Rohtak. She has more than near about 70 publications, and her major achievements are, she is COVID-19 Warrior Award. She has got this COVID-19 Warrior Award. She has also got this Women Achievers Award. Another Ayushman India 2020 award she has also got. She has got the Best Teacher of the Year 2022 in 2022, and she has got this State President's Award. So I would like to welcome Dr. Savita Verma. Am I audible?
Thank you, sir. Yes, sir, you are audible. Am I audible?
Yeah, you are very much audible. So please, ma'am, you can continue.
Thank you, sir. And at the onset, I would like to thank all the organizers of this Sopicon for giving me this opportunity to speak on a topic which is close to my heart. And, it has been almost more than 4 years now that we are into the COVID pandemic. And COVID vaccine development has been one of the success stories of this pandemic. But having said that, those vaccines were developed at a very fast pace and underwent rapid development. So there was a lot of skepticism on the part of public when they were accepting this, and many felt that there must have been some compromises during its development. So I'll be talking about the safety of this COVID, and I'll be briefly touching upon some of the aspects, what was different in the COVID vaccine development and the clinical trials, and how safety was ensured in these trials. So this is the flow of my presentation, an overview. And what was unprecedented timeline for the fast tracking of the clinical trials and how safety was being monitored in the trials in the development phase and now in the post-market phase. So a brief overview, we know that first case of COVID-19 was discovered in cases have now crossed over 750 billion. Mass vaccinate the population. The success story being that the turning point was on 8th December 2020 when first dose of COVID vaccine was done in the world. And in India, the COVID vaccine started in 16th January. So this is a brief overview giving you about the timelines which happened at that time. And this is the global scenario now. At present, we have more than 750 million confirmed cases of COVID-19, with approximately 0.9% death. Million doses of COVID vaccine has been given. So what was different during their development? And there was a global response. These came together, including with the various bodies, to combat this pandemic. And at present also COVAX is working very hard to provide. There's many areas or the countries where still the COVID vaccine is not available. And COVAX is working really hard to provide these vaccines. So let's see what was different in this COVID vaccine development. Now, routinely in the clinical trials, it takes approximately 10 to 15 years for a product to come into approximately takes 1 to 2 years. But what happened during COVID pandemic, there were unprecedented times. So adaptive trials were adopted and there was a lot of changes which were made in the regulatory bodies. And most of these trials, they went in parallel to each other. And separate committees were made in the regulatory bodies, but nowhere during these trials, the safety or the primary objectives of the trials was compromised. So how safety monitoring was done in these COVID vaccine trials? These COVID trials were conducted on the same number of phases with safety checks and in the same number of volunteers. And routinely, the safety monitoring in vaccine trials is done with immediate adverse effects or events, solicited and unsolicited adverse events, treatment emergent adverse events, and serious adverse events. So what are these and how are they monitored in a clinical trial on vaccines? So generally we do solicited adverse events are observed for 7 days post-vaccination. And in this, we monitor both local as well as systemic adverse drug reactions. Unsolicited adverse events are, yes, monitored on these 7 days as well as throughout the study period. And once the vaccine dose is given, so within 30 minutes, immediate adverse events are observed. And serious adverse events are observed throughout the period of the study. And their timelines are same as that of other serious adverse events in other therapeutic clinical trials. So I'll just take an example of the journey of Covaxin, which is the first indigenous COVID-19 vaccine trial in India, to explain how these safety checks were in place and how the safety was maintained during the COVID vaccine development. Now, we know that by the end of March 2020, pandemic was declared in India as well. And the strain was provided to Bharat Biotech company in collaboration with NIV and ICMR in April 2020. And as you can see in the timeline, within 1 month, the strain was inactivated and the first preclinical study started in June 2020. By July 2020, we already had the approval from the DCGI office for phase 1/2 adaptive clinical trials. And by August 2020, the first phase 1. And by January, we all know that by January 2021, the COVID vaccination drive was started in India with 2 COVID vaccines, Covishield as well as Covaxin. So let's see how safety was maintained in the Covaxin preclinical profile. Now, the primary objective of preclinical profile is to identify whether the animals develop antibodies or immunity to the given vaccine or not. The same objectives was kept in the preclinical vaccine trials as well. And immunogenicity and safety of Covaxin was observed at 2 doses, 3 and 6 milligrams, in 3 different species, mice, rats, and rabbits. And all the 3 species showed excellent safety profile. They also did protective efficacy or challenge test in macaques, and these also showed good effect against the COVID vaccine. So Simultaneously, these results were submitted to the DCJ office to get the permission for phase 1 clinical trials, and development and reproductive toxicity studies were also started. And these studies generally go in parallel with the clinical trials. And now, obviously, we are 3 years ahead, so no treatment abnormalities were found in both prenatal and postnatal groups during the preclinical toxicity studies. And now we know that we have already given these doses to pregnant females as well as children, and we have not yet to find any congenital abnormalities or other abnormalities in developmental abnormalities in these patients with COVID vaccine. So let's come on to the Covaxin clinical profile and how safety was ensured during phase 1, phase 2, and phase 3 clinical trials. So like it said, we adopted adaptive designs. The primary objective of phase 1 clinical trial is to the vaccine is safe for human use because it will be used for the first time in human beings. And the primary objectives of phase 2 clinical trials are to find out the correct dose, whether the vaccine is effective against that particular disease or not, and is it safe or not. So these objectives were same for the vaccine, COVID vaccine trials. And there was an adaptive seamless phase 1 followed by phase 2 trial, which was adopted. So what happened in this trial was that 2 doses of COVID vaccine was chosen, like 3 milligrams and 6 milligrams. So initially in 50% of 50 individuals, low dose of Covaxin was given, and 7 days post-vaccine dose, solicited adverse events and unsolicited adverse events were observed. And the data of these post-vaccination 7 days was submitted to the Data Safety Monitoring Board. So once clearance was given from that Data Safety Monitoring Board, only then rest of the participants were recruited in the clinical trial. And once all the doses were given, day 28, the immunogenicity and safety data after clearance from the DSMB board was notified to the CDSU, and simultaneously once that report was submitted, the phase 2 of the clinical trial was started. So both instead of going in a sequential manner, because phase 1 trial was participants were observed for almost 1 year, and phase 2 participants were observed for almost 6 years. So had we waited for the completion of those trials, we would have lost a lot of good time. So simultaneously, within a month of starting phase 1, phase 2 was started, and there was no safety compromise. And on day 42 and day 56, interim report was also submitted to CDSU for both phase 1 and phase 2 clinical trials. And that report was given clearance, the clearance was also asked for phase 3 clinical trial of Covaxin. So this is just the results of that Covaxin study, phase 1 study. It was done on 375 healthy volunteers between 18 to 55 years of age, and only mild adverse events were noted after dose 1, and no immediate adverse events were noted after dose 2, and no serious adverse events were noted in phase 1 clinical trial. And this is just a table from the presentation showing all the solicited and unsolicited adverse events which were observed in this phase, which are available in this publication. And then these are the results of phase 2 clinical trial, which was done on 380 healthy volunteers between 12 to 65 years of age. And again, no immediate or serious adverse events were observed in this phase as well. Now, these the results again are available in this publication. Now, coming on to the objectives of the phase 3 clinical trial, which was started within 2 months of starting the phase 2 clinical trial. That means by November 2020, we had already started recruiting the participants in phase 3 clinical trial. Now, the primary objective of phase 3 clinical trial is to see efficacy of the given vaccine, whether it does protect the humans from the given disease or not. And again, safety is always one of the primary objectives of all the clinical trials. So, this Covaxin phase 3 efficacy and safety trial was conducted across 25 sites in India in 25,800 subjects. And the healthy volunteers, it also included 30% of the participants who were at risk, whether it was some comorbid condition or healthcare workers. So because ultimately this vaccine was to be used in the general public who might be suffering from some comorbidities or might be at risk. So there was provision to observe the effect of this vaccine in compromised patients or who were at risk. So the phase 3 trial also achieved its primary objective, and we found that overall the protectiveness was 77.8%, and there was 93.4% protection against the severity of the disease and hospitalization. And again, there were minor adverse drug reactions which were observed in this study. So after completion of all these trials, phase 1, phase 2 and 3, emergency use authorization was granted by the regulatory bodies who went through all the data. So as I said earlier, there was no compromise anywhere on the safety as well as on the number of participants or phases of clinical trials which were conducted for these vaccines. And after emergency use authorization, we now know that there were 4 platforms on which these vaccines were formed. And these are some of the vaccines which were granted emergency use authorization for COVID. And that's why we could fight against this pandemic, because we had so many molecules which were available, and all the countries collaborated together to fight this pandemic. So this is the summary of the Covaxin clinical trials, because the story didn't end over there. Obviously, since there was fast tracking of the clinical trials, and within 1 year, these vaccines were given emergency use authorization and were being used in the public. So the safety studies and the efficacy studies carried on further. And now we also have done safety studies as well as efficacy studies in pediatric population. Booster dose studies have been done for same vaccine or for heterologous booster doses as well. And this is the summary of the clinical trials which were conducted with Covishield in India, and 4 randomized trials were conducted. And again, they also found that the vaccine efficacy was approximately 70% with mild adverse drug reactions, and 2 mild cases were observed, out of which only 1 was possibly vaccine related. And they have also completed their heterologous as well as homologous booster dose studies as well with good safety profile. So this is a brief slide which is showing the reactogenicity with various products in the clinical trials. And obviously the reactogenicity was observed to be less with Bharat Covaxin, because maybe it was based on the killed vaccine platform as compared to other. But all these reactogenicity profile were minor when we look at the number of deaths and number of morbidity which was happening with the COVID per se. So what next? Because there was fast tracking of the trials, and within 1 year, like I said, these vaccines were available. So the active post-marketing surveillance is also being done, has been being carried out for these vaccines now, because millions of doses have already been given of this vaccine. And now a lot of data is available on the efficacy as well as safety of these studies. So the primary objective of post-marketing surveillance is to observe any unexpected side effect and to see whether the vaccine is still safe when it is being used in larger public. So WHO has defined adverse event following immunization as well as adverse event of special interest. So I'll just briefly touch upon the adverse event of special interest, because these were reported more during the post-marketing surveillance. So these are any serious or severe yet rarely life-threatening or long-lasting problems which are seen with vaccine use. They might be causally related to the vaccine product or might not be related to the vaccine product. So this is the flow where the WHO is actively following the COVID vaccine safety surveillance, where AIIMS reported from various sources, and all the countries are being accessed, and all the countries are linked to this online platform where they are reporting their AFIs after causality assessment to WHO, and all the databases are available at these websites. I'm just showing one example of various reports which have been published now. So this is a post-marketing surveillance report published in Ontario from December 18, 2020 to April 2023. And this is giving the percentage of various vaccines and the side effects observed from them during these 3 years of use of this vaccine. So all these are available online, and these data are available in public domain. And these, out of these, all the reports which have been published and all the data which has been provided to the WHO, so 5 adverse events of special interest have been identified by WHO as far as these COVID vaccines are concerned. These are anaphylaxis, thrombosis and thrombocytopenic syndrome, Guillain-Barré syndrome, myocarditis and pericarditis, and reports of death after COVID-19 vaccines, which are extremely rare. So active surveillance is being done for these for these adverse events following COVID vaccination. And apart from these, there were other reports which have been published, case reports have been published in for neurological side effects. And a review has noted that some of the neurological complications which have been observed with COVID vaccines, which they include intracranial hemorrhage or transverse myelitis, Bell's palsy, Guillain-Barré syndrome, and so herpes zoster, so on and so forth. But the incidence of these neurological side effects is very less when we compare to the protectiveness and effectiveness of these vaccines in counteracting the COVID pandemic. So India is also not far behind in its surveillance. And as of now, we have almost given more than 2 billion doses of COVID vaccines to our public, including the booster doses as well. And COVID vaccine surveillance in India is actively being done for post-marketing surveillance for adult population, pediatric population, and then spontaneous reporting on the CoWIN app, as well as causality assessment reports are being published by the National Committee from time to time for COVID immunization. And these are some of the examples of the reports which have been submitted to the regulatory bodies and the reports which are available in public. So for Covaxin post-marketing study, approximately 3% of the adverse events were observed, but most of them were mild in nature. And this is another report which is available on the CoWIN app. And then these are the causality assessment reports which are being published by the Ministry of Health and Family Welfare after doing their causality assessments. And overall, this has been a team effort, and nowhere the safety of these vaccines were compromised, as we have seen through the journey of Covaxin and other vaccines which are now available for vaccination against this pandemic. Thank you.
Okay. Thank you very much. It was a wonderful talk. Are there any questions?
Thank you, sir.
Okay. It seems there are no questions. It's you solved all the doubts in the.
Everybody was sleeping after the lunch break.
But it was a wonderful presentation, madam. Congratulations.
Thank you, sir.
Okay. So now the third presenter is Dr. Deepika Bansal.
I think, sir, ma'am is not able to join us today. Ma'am won't be able to join us today.
Okay, so the session ends over here. So the session ends now. Thank you very much.
I express my sincere gratitude to speakers and chairpersons of this session. I please invite Professor Ravikant sir to felicitate our chairpersons. सर आप ही देंगे सर आई Thank you, sir. Thank you, chairpersons. Now we move forward to our next session, which is panel discussion, where we will hear from experts in the field of drug safety and pharmacovigilance, who will share their views and insights on generic versus branded pharmaceuticals. I invite our panelists, Mr. Jay Prakash Mishra, DGM, Procurement and Quality Control, PMBIM. Ms. Komal Khandelwal, Senior Manager Marketing, Life Care Innovations. Mr. Abhay Patil, Vice President, Quality, Biopharma Division, Intas Pharmaceuticals Limited. Professor Dr. Ravi Khan, Head of the Department of Internal Medicine, AIIMS Rishikesh. I welcome you all. Now I call upon moderators for this session, Professor Sanjay Gaur, Professor and Head, Government Doon Medical College, Dr. Puneet Dhamija, Additional Professor, AIIMS Rishikesh. Now I request moderators to kindly preside over the session. I request Dr. Manjunath to please ask Dr. Abhay Patel to join us online. Dr. Manjunath, can you please ask her to join us online?
Sir, mic.
Good afternoon, everybody. This is Dr. Sanjay Gaur, and on behalf of Sopicon, I welcome you all to this panel discussion. This is an extreme privilege to moderating this session, and we have an extremely literate and educated panel members who will be joining us today. And on behalf of the organizing committee, I would like to welcome Professor Ravi Kant, who is head of the department in General Medicine, AIIMS Rishikesh. Welcome, sir. Mr. Jaiprakash Mishra, who is DG, Quality and Procurement, PMBI. Ms. Komal Khandelwal, who is from Life Care Innovations. Dr. Abhay Patel, Vice President Quality in TAS, who will be joining us online, and my fellow moderator, Dr. Puneet Dhamija. Just to set the tone, today's topic of discussion is generic versus branded. Just to set the tone, what exactly are generics? The WHO definition of generics is any pharmaceutical product intended to be interchangeable with the approved innovator product, and it should be bioequivalent, therapeutic equivalence should be present there, and the approval of generics is also dependent upon the demonstration of therapeutic equivalence. Now, the important thing is that these generic drugs can be manufactured without any licensing requirement from the original manufacturer, and they can be marketed immediately after the expiry of the patent or any exclusive rights. The generics are comparable to the original branded products or comparator products, and they are available at the fraction of cost, and thereby they have a potential to decrease the out-of-pocket expenses, which is the major expenses in healthcare needs of our population. The Indian pharmaceutical industry is third largest by volume, and it is playing a key role in driving better healthcare outcome across world through affordable and high quality generics. Now, there's an interesting document which was published by Indian Pharmaceutical Alliance called as Vision 230, Defining Growth Aspiration, which is estimating a market, a pharmaceutical market of for Indian industry of US $65 billion by 2024, which is to rise to 120 to 130 billion by 2030. So there's a huge potential in this market. And this will accelerate global healthcare across India and world by giving access to high quality and affordable generic drugs while supporting state-sponsored health schemes. Our industry is uniquely poised to leverage the patent cliff with increase ANDA share. The Indian generic drug market is poised, is around 24.53 billion. It was 24.53 billion in year 2022 and is expected to rise with a CAGR of 6.9%. So there's a huge potential for these generic medicines. There was an editorial that was published way back in 2015 by a fellow moderator, Dr. Puneet, and it was titled as "Only Generic Drug/Names. Is India Ready?" And in his editorial, he beautifully explained and discussed issues related to quality assurance, safety, efficacy, and of the generic drugs. And he also suggested various measures like empowering and strengthening of regulatory apparatus. And it's been a long way since 2015. A lot of water has flown through River Ganges. But doubts regarding the generic still persist in some of the stakeholders. These questions still stand, and we still are facing the same question. Is India still ready in achieving goals of universal healthcare access by providing quality assured generics? And the point is providing quality assured generics, not quality assumed generics. Now we have, I would hand over the mic to Dr. Puneet to have his opinion about this.
Thank you, Dr. Sanjay, and thank you, panelists, for being here. The idea for this panel discussion was to have a clinician among the manufacturers of the drug, how they perceive, how to instill the confidence among the prescribers about generic drugs so that healthcare can be provided at an affordable cost. What are the concerns that the clinicians have in their mind when they are prescribing? What are the things which a manufacturer, whether it is a generic manufacturer or an innovator manufacturer like Lifecare Innovations, they should take care so that clinicians start believing in the products which are manufactured. And so this was the idea behind keeping a panel discussion, and we will be, and Mr. Abhay Patel from Intas Pharmaceuticals, who is taking care of biosimilar division, will be joining us online. So biosimilars, we have kept it as a separate topic because that is entirely a specialized area.
I think Dr. Abhay has joined.
Yeah, it is good to see. Hello, hi Dr. Abhay.
Welcome, Dr. Abhay.
Hello everyone.
Yeah.
Good to see you and good to be part of this discussion. I'm happy to contribute as I can.
So the idea is all about how to build up the confidence among prescribers about generics, whether it is a generic small molecule or a biosimilar products, or new formulations of the already existing drugs. So I will ask the first question to Dr. Ravikant, who is a prescriber. Whenever you are prescribing a drug, what are your concerns, what are your thoughts behind prescribing whether a small molecule as a generic name or a branded name?
Yeah, so thank you very much, Dr. Sanjay and Dr. Puneet, for calling me here. In fact, this is a tricky question to answer. And all of you understand the psyche of Indian patients. And to a larger extent, what India is facing is a transition between, branded to generics. So I think it will take time to understand and to be confident. So when the cause of life or maybe the reason to save a life is there, which one to choose, which one not to choose? This is a big question to everybody, including me myself. So I don't deny the fact that even today, the clinicians are more inclined towards the branded medicines, right? Or the drugs which are uniquely prepared by some pharma companies under patents. We have seen certain companies which are bringing molecules which are quality assured, as Dr. Sanjay has already said. And the efficacy, no doubt, so far as bioequivalence of the generics is concerned, is good. The drug is acting nicely, but still there is some resistance somewhere in the minds of the prescribers. They are still tilted towards the non-generic or branded molecules so far as the life-threatening conditions are concerned. That is one aspect of the patient care where we deal with the critical care patients, where we're not supposed to take any chance with their lives. Second is, okay, OPD walk-in patients or patients who are admitted in general wards for illnesses of milder nature, where we can definitely, have time to act, to see, and then to react again. So this is a big question that every alternate patient asks us, because these patients are procuring the drugs from the generic pharmacy available with us, and they ask a simple question: sir, should I take this generic drug or I should take the one which is basically a branded drug, or basically which is under the patent, which is of course quite costly? I think the answer to the clinicians is very clear to the patients. This drug is effective. We are seeing the blood sugar coming down with the same molecule I prescribe as glyclazide in the generic name. So far it is controlling is fair. You please continue, and there is no reason to doubt about the efficacy of the drug so far as the generics are concerned. Now, what the problem as a prescriber I have seen, and I am emphasizing again and again to our pharma colleagues, we have seen some erratic responses also. We have seen some erratic responses in the patients in real life scenarios. Although we have a tight quality control, we have regulations, we have so many other laws before the drug is coming to the market, but still I believe until unless we have point of care quality assurance facility within an institute, I think totally relying will take time on generics. So what my perception about any drug, whatever drug we are prescribing, say for example, we need to have in-house quality control lab where we can check in what the bioavailability of the molecule is at the point of time the patient is receiving. Because we know that India is a varied climatic condition country where maintaining a cool chain or cold chain is quite a difficult task. So just to check the efficacy of drug at the point of, giving to the patient, at the time when it is being instituted to the patient, that drug should be tested in-house, and I think that is the point when we can say that we are 100% confident about the generics as against the branded medicines. Thank you.
But Professor Ravikant, as far as the testing, in-house testing of these drugs is concerned, we have this Jan Aushadhi initiative, which ensures that the drugs that are being procured by PMBI, as Mr. Mishra would second it, that and they are of the best possible quality. They are tested in NABL accredited labs, and then they are distributed to the Jan Aushadhi centers in case. So as far as the drugs that are available with Jan Aushadhi, the generic drugs that are available with Jan Aushadhi, there should be no doubt about the responses of the drug, efficacy of the drug. And one more thing that I would push in here is that you must be aware of the erstwhile MCI circular that was circulated in 2017, which stipulated that all the registered medical practitioners, they have to govern under the AIIMS Act. They have to prescribe drugs with generic names, legibly written, with rational prescription and use of drug. Now that is an old story, obviously, going by the date that it was circulated in 2017. But one of the recent development that I would like to bring to the notice of this house is that there's a standing committee on chemical and fertilizers of 2021-22 of 17th Lok Sabha. Now this submitted in its 29th report while it was reviewing the Jan Aushadhi Pariyojana, and they recommended that the prescriptions without generic names not be permitted at all. Further, to bring in a law to the House to enforce the same. Now, how do you see these two developments?
Yeah, so quite pertinent. I mean, so far as the law making and the enactment and the execution of the law is concerned, that is one aspect of the coin maybe. But I am a person actually who personally believe that yes, that is okay for 95% of the population, but there is 5% of the population, you know. We are living in a country where we have to give an approach to a patient which is free from the foundation. Like say for example, I am not opposed to generics. I am quite a supporter of generic prescription. We do favor the generic medicines, and our Jan Aushadhi is doing fine. Everything is wonderfully running, and our pharma colleagues are taking care of the Jan Aushadhi and other pharmacy in the institute. But even then, you know, from manufacturing till consumption, there is a huge, gap. And I just want to emphasize upon, although we have good laws and we are prescribing generics, but still, when the life comes, should I be not 100% confident? The answer is definitely yes. And the answer to this question is, so far as some good centers are doing, so what they are taking, what drugs should they make available in the pharmacy? Say for example, very good reputed institutes in this country only, they are testing these samples in their own labs of various, drugs of the same same pharma agent or maybe ingredient. I can put forward example of one, vitamin D, which claims to be having 60,000. But in hospital estimation by quality control analysis and the HPLC method, they have found that it is varying somewhere from 30,000 to 66 or sometimes 70,000. It's a huge gap. So yes, I think I am a strong proponent of a generic, but 2-point testing, manufacturing and consumption should be the end point.
Let's summarize this. The panel discussion is all about how will you get confident about generics as a prescriber. So let's have a viewpoint of PMBI, the largest procurement agency for generic drugs.
Yes.
What they are doing for ensuring quality of their products, and let's hear from PMBI.
Thank you. See, about generic, once as a movement it is started, to one of the IS officers, he was just explaining generic. He said that, so you want the milk, and somebody is saying you, no, you bring the milk of Ram's cow. You require milk, and now once it becomes Ram's cow's milk, then it is now the ethical. Ram's cow's milk they require, but you require milk. Basically, in case of generic, this is same. As far as manufacturing part is concerned, manufacturer same API has to be used. There is API test has to be done. As far as PMBI, the way PMBI procures the medicines, we have very strict term conditions. We invite tenders, and we procure from WHO GMP certified companies. Then we take COPP. Once we are taking COPP, certificate for pharmaceutical products, it does mean company has to provide the stability data to the regulations, to the regulatory authorities. And then COPP for each and every product every product manufacturers get. MSCs, market standing certificates for 3 years. So all these parameters, and then we defined packaging parts. And once the manufacturing companies manufactures the product, we do the inspection during the BMD. Once they have taken the batch manufacturing date, then our person goes there, they inspect each and every parameters, and once product is inverted, then they provide the COA report, certificate of analysis. But their certificate of analysis just only for the invert, that we have inverted in CWH. But that is not for sales. We have our own mechanism. We have, through the tendering process only, India's top laboratories, who is having GLP compliance as per Schedule L1 from Drug and Cosmetic Acts, and NAVL accredited lab. Along with we have the list of equipments and the analyst, who is doing the analysis, their certifications, each and every parameters are checked. And then from there we send our samples. Once they give the pass report that yes, product is of a standard quality, only then we are letting it to go for sales. So such strict parameters we have. Third thing is that yes, as Dr. told, that storage conditions, it is one of the important role it plays. We have GDP compliance, good GDP compliance warehouses, and even for transportations, that part we take care. So from manufacturing to laboratory levels, that yes, how the products in inverted, how it is kept, and each and every parameters after testing, then we send it for sales. And once it goes in market, we have such strict conditions that suppose if any product, if we get the market complaint, that yes, one product has got market complaint, we in tender itself, such strict conditions we have, that suppose we have procured for the 1 crore unit, 1 crore strips, and we have only now the 10,000 strips in my warehouses. But if we get the market complaint and our control samples, once we check it and we find that yes, there is defect and that is NSQ, then we issue the debit note of almost 1 crore strips, not 10,000 strips. So for the manufacturers, it's a big punishment. No manufacturer can think of doing any compromisations. So such a strict, and this year also I would say that despite having all this, so almost 15 crore of penalties we have imposed on the manufacturers on these parameters also. So the kind of strictness we have with the PMBI, I can bet that yes, none of the pharmaceutical industry, none of the company is maintaining such a strict parameters. And yes, I assure you that yes, use generic product with confidence, no problem at all, full confidence I give you. And this program, Janaushadhi program, this is from core of heart of our Honorable Prime Ministers. And slowly and slowly, there is very good increase in the sales growth, and people are accepting it like anything. Now, almost in every month, we are procuring around approx now more than 150 crores every month. And this way, every year we are growing by almost 40%. So such a phenomenal growth. So it is mainly because of confidence, and slowly, people are gaining the confidence. And doctors also, I would say that yes, use generic product, especially I would say that I will not talk of that, yes, what is available in pharmaceutical markets, other generic and all, that they also might be good. But for the other generic in pharmaceutical market, what is available? That is for enriching the pocket of chemist, that is retailers. For their, if they are procuring, they are purchasing it in 40 rupees steps, but their MRP is already 400 rupees. So that is not serving the purpose. But here we have kept MRP because government has given clear guideline that we are not here for profit-making organizations. We have, our success is basically measured that yes, how much out-of-pocket expenses we have reduced to people of our country. So this is our effort, and our all success is measured on this only. So our business success doesn't mean that is enriching the pocket of government. Our business success means that how much happiness we are bringing on the face of people. So this is our effort, and we are succeeding in this. So I would say that with full confidence, use generic, use PMBI product, Janaushadhi product with full confidence.
Mr. Mishra, you had mentioned the transportation and storage aspects of the supplies that are being procured by PMBI, and then they are distributed to various depots, warehouses and Janaushadhi Kendras. I had been instrumental in establishing one of the first Janaushadhi Kendra in Uttarakhand state that was established in Sushila Tiwari Hospital in Haldwani. The main problem that we witnessed was frequent incidences of stockouts. That was a real problem. So even if we are able to procure quality medicines, if these medicines are not being able to deliver at, they're not being delivered at the outlets at appropriate time, the whole mission fails. This whole mission, as PMBI, you have been interested in carrying out this mission, Janaushadhi mission. As far as transportation and storage of these medicine is concerned, we have good clinical practice guidance documents. We have good manufacturing practice guidance documents. Do we require a good transportation practice type of guidance documents, or if it is so, will it be of any help?
Yeah, for transportations, as per level claim that is mentioned, see, conditions are different. Like for, I would say that for 2 to 8 degree, the product which required to be transported in 2 to 8 degree, though like suppose for the insulin, so there is that required to be stored at 2 to 8 degree, and their accelerated study, that accelerated stability for 6 to 7 days. Yes, of course, that goes up to 30 degree also, but accelerated stability. But it doesn't mean that on label, what is mentioned on label, that is 2 to 8 degree, it required to be transported at 2 to 8 degree. Once we are sending our cool chain items, then either in cool pack or there is a special vehicle. A special vehicle comes, that is with the that comes with the data logger, with the facility, so that such products can be delivered to the distributor levels. And from distributors also, once they are sending the a small unit for cool chain samples, they are also transporting it with the cool pack. So that is important. See, I would say that in like in pathology, as you say, that the pre-analytical part, the way it plays a vital role, especially in the era of pathology report, the kind of importance it has for pre-analytical part. Same way, I would say that in medicines, storage condition plays a role. So it plays a role.
Yeah, I'm coming to your topic, taking further from the discussion about Janaushadhi, and I will move to Mr. Abhay Patel also, pass on this question to Mr. Abhay Patel. Your quality checks and parameters are very stringent. We agree with that. But your costs are very low. Yeah. The drugs provided are at a very cheap, low cost. when we talk about, we can take whether it is possible to have such stringent checks by generic manufacturers for, let's say, we move on to biosimilars, which requires very different kind of transportation practices, storage conditions. Will they be able to provide this much quality at the prices which are being provided by Janaushadhi? Or it is just a dream that the prices of biologicals can be brought down to such a level where quality is not compromised? Now I will ask Mr. Abhay Patel a direct question. How the confidence among the prescribers about biosimilar products should be developed so that they are happy to prescribe products from Intas or maybe any other generic manufacturer, any other manufacturer of biosimilars? And how do you ensure that cost is kept low? Not for making supplies to Janaushadhi, let's say for insulin, which is provided by you to PMBI. Mr. Abhay Patel.
Okay, so I will answer first part of the question, that is around the quality, because quality and safety are the most important aspects to make sure that the product is safe and efficacious. So when we talk about the biosimilars, the biosimilar development pathway is quite long when compared to the other generic molecules or the small molecules, which are around 150 daltons. Against that, the biosimilars are around 150 kilodaltons. So they are quite large molecules, and their development is also very different than the other molecules. So where it starts from the analysis or detailed primary and secondary structural analysis of the originator molecule, because that is the target quality profile that a biosimilar would aim for. And once that is established, those parameters are incorporated into, let's say, some of the cell lines that we are looking for. Now, and then the cell lines are developed and the cell lines are characterized. Of course, they are characterized. There are about 100 cell lines that needs to be developed to select one cell line for a biosimilar. So the point I'm trying to say is that there's a lot of development activities that goes on, and it is time intensive as well as cost intensive. I'm not trying to defend the cost, but I'm just trying to tell you where it comes from. Now, when we say that this cell line needs development, what does it mean? It means that we have to determine which amino acid sequence has to be used. That amino acid sequence has to be incorporated into the gene synthesis, and that has to be converted to a expression vector, and then it goes into the CHO cell lines. This way you create a bank or number of banks, and from this we have to shortlist a bank which will give us a product which is very similar to the originator product. Now that exercise itself also takes a lot of time as well as cost, because the originator products are quite expensive, and we need multiple batches of analysis of both the originator product and the biosimilar to make sure that or they match as close as possible. Now this is only the part of analytical biosimilarity. There is also a part that comes in about the clinical similarity, because the analytical similarity does not make sure that clinically the molecules will behave similarly. So there are additional clinical studies that are done, and these are over and above the PK/PD studies that is done. Objective of this study is mostly to make sure that there are no significant clinically meaningful differences in safety and efficacy of both biosimilar as well as the reference product. So there is a comparison which is done in the clinical setup as well. So there is an analytical comparison, and then there is a clinical comparison. Now, once that is completed, there are many. It undergoes extensive testing. It undergoes stability testing. And once the demonstration of biosimilarity is completed, we apply to the DCGI, and then we get the approval of this molecule. Now, having said that, at Intas, we have just one facility that manufactures both biosimilar bulk as well as biosimilar injectables. So everything that is related to the biosimilars at Intas get manufactured in the Intas facility. What does that mean? It means that our facilities are approved by EU GMP. We also have approval from Health Canada, and the product that is supplied to EU and Health Canada, the facility which supplies the product to EU and Health Canada is the same facility that supplies product to the market which is in India as well. So we do not have two different facilities. It's the same facility that caters globally as well as to India. So that is one part of making sure that the quality of the product that is manufactured is same for all the patients across the globe. That is one. Second thing is we come to the cold chain where which plays an important aspect for the distribution of this product. Now, during the panel discussion, there was a discussion around variable climatic condition as well as the cold chain requirement for those. So the packages that are used for this cold chain transmission are different types of packages, but given that India has varied climate, and even within the same geographical locality, we could have temperatures going from 47, 48 to around 7 or 8 degrees during the winter. The cold chain package has to be developed in such a way that it does not create any additional. The product inside maintains the same temperature that it has to be maintained. So most of these products are 2 to 8 degrees Celsius product. So these cold chain shipment, both shipment as well as the package, both are also validated. The validation activity also is a part of development of the product. So, and in the end, of course, each batch is tested, released. There are extensive testing which are done during the processing as well as at the end of the manufacturing. So this is how we ensure that the quality of the biosimilars that is manufactured is same as the innovator, as well as we make sure that it maintains the required storage condition before it reaches the patient.
Thank you, Mr. Abhay. So keeping the costs manageable is always a challenge. Life care innovations is involved in new formulation development, and you have many patented formulations for already existing drugs. So now we have the third profile who is an innovator. I can call you as innovator. Life care innovation is one of the innovators. So how do you ensure that your products, do you do a cost effectiveness analysis, and how do you see your products? Whether they are in absolutely good quality at an affordable price, or there are you find some challenges in meeting the both ends, providing good quality at affordable prices as innovator?
So as I said, quality is equivalent to the safety of the product, and those are the two parameters that are not compromised. Having said that, that does not mean that the product has to be expensive or not affordable. There are different ways to manage the cost of the product or to manage the cost of the product, but I'm not really an expert in that. But what I can say, because I handle the quality aspects of the biosimilars, the cost aspect is mainly managed by the operations person. But
Mr. Abhay,
I would like to add. Sorry, go ahead.
Yeah, Mr. Abhay, let that be answered by regarding the balancing out the cost as well as the quality. We have Ms. Komal Khandelwal, who is representing Life Care Innovations, which is not a very big kind of, I would say, that a multinational company like Pfizer, but yes, they are very good in formulation development. They have their own patented formulations. Now, when they are developing a patented formulation, how do they keep their costs low? Or maybe how do they achieve this target of affordability and the quality? Let's have the answer from Ms. Komal Khandelwal or the Life Care Innovations for that.
Hello. Thank you, sir, for the question.
Before you start, I would like to congratulate you that Life Care Innovations They got a National Tech Excellence Award in 2022, and congratulations for that. Please continue.
We at LifeCare Innovations, we have innovatively designed products. Our foundation is strongly science-based, and we maintain the superiority of our products by following the principle of QBD, which is quality by design. So we somehow we are deeply involved in studying the mechanism of the formulation, that how can the design be improved so that it can be highly efficacious when compared to the existing formulations. In our drug, which I'll be covering in my panel discussion, in that drug, what we did is we reduced the dose. We reduced the dosage and increased the efficacy of the product. Because of the low dosage, the cost of the total treatment of the entire therapy was reduced to 4 to 10 times when compared to the other formulations. So this is how we have the research-based product. We are the innovators. We are the superior drug when compared to the other formulation on the basis of pure science. And that is how we achieve, that is how I can say the product is economical, though it is patented.
Thank you, Ms. Komal. It was... we had ideas from all the learned panelists, and in the end I would like to just conclude our discussion that one thing is very sure, that prescribers are not at all averse of using generics. This is for sure. The only thing is that quality assurance has to be there. So we need stringent quality control, and for that we would also be needing more these quality testing labs, and then it will be better if we have good transportation practices kind of guidelines, which specifically caters to specialized products. Thank you very much.
So we concluded, if there are any comments from the audience regarding the quality related issues, or maybe a generic versus branded, the debate always goes on. So if there are any queries from the audience. So thank you. Yeah.
I thank the moderators and the panelists for an excellent discussion we had, and I think it was quite insightful, and we heard from both the sides their perception about this unending debate of generics versus branded. My point, which just came as a afterthought. Probably we are always debating all these issues all the time in the hospital, and trying to convince the clinicians that, okay, Jan Aushadhi and all the other generics are equivalent, supposed to be theoretically, scientifically equivalent. But then the doubts linger always. And whenever there is a case of ineffective drug, or the drug is not working, somebody has a personal experience, some clinician, so then doubts start again emerging. So my question was, we are living in a world where information, and you have to convince with data. See, our So our prescribers, our patients, at the end of the day are consumers for drugs. And the only way you can convince is by data. Pure data. Of course, scientifically we know that we can generate data by chemical analysis, by doing bioequivalence studies. See, when we export, we are probably the country is the largest exporter of generics to the developed world. But then they go through the process of proper analysis and bioequivalence studies, and they provide the data to the regulatory authorities and they approve the drug. So many of the developed world, USA, they are using our generics happily. But we have doubts here in our country because our regulatory framework is not that strong. So is it possible that especially PMBI and other generic manufacturers, they can provide data which can convince the end users, the clinicians? So that's my question. And thank you for a great discussion.
Hello. For the export purpose, the manufacturing companies who is doing the export, they have to mainly provide COPP data, and beside the other formalities that yes, that particular company and product has to be registered there. And in other country, whether European countries, and there is European pharmacopoeia, and African country mainly BSP or USPs. These two pharmacopoeias are being used. Now, as far as this part here in India is concerned, we are also now we are taking COPP as per IP. We are taking COPP as per IP. So this part is one. Second thing is that recently, last year only, we have done 150 branded versus generic product testing in our impanel laboratory. And this is from all over India. We have 14 labs, including NIPER. We have 14 laboratory, impanel laboratory, and that all state-of-art setup, the best setup. And there we have tested almost 157, exact number I will tell you, and none of parameters I would say that where we found that yes, product which PMBI has.
So we can put it like that we are assured that PMBA is providing quality assurance of the products. But do you have let's pass on the mic to the innovator, another innovator, Lifecare Innovations. What is the feedback mechanism that you have in place? Let us say about your products, which are new products, which are innovator products. Once they reach the market, how can the end users give you a feedback about the quality or their quality concerns? Do you have any mechanism like that, like for pharmacovigilance, post-marketing surveillance activities are required, are done by big multinational companies? Do you have such mechanism in place in your organization also?
At Lifecare Innovations, we routinely submit the PSUR reports on annual basis to the DCGI office. Lifecare, like I told before, I just want to add on to Dr. Puneet Dhamija's question that time. We make innovative products. Our product is innovatively designed. Our foundation is strongly science-based on the fact that we make the product by improving the design of the product. If I enter into our flagship product, Fungizome, to explain you what Fungizome is, Fungizome was developed by Dr. J.N. Verma and Dr. Lily Verma. They are the co-inventors of the drug. We are all aware that for the past decades, the treatment of systemic mycosis has been a major concern. For the past decade, there is not even a single chemical, entity which showed
recording in progress,
which showed that it is potent and toxic and does not cause any toxicity. Amphotericin B is a gold standard molecule which we know from the historical time. But the problem with amphotericin B was that it attacks the human cell. At the same time, it is giving safety, it is providing the efficacy, and it is killing the fungal cells. So because of that, it has the inherent potency, but at the same time, it has some sort of toxicity. That is why the that is why the chemical, this amphotericin B has to be, mixed, has to be combined with the liposomal technology so that a new liposomal drug can be made. So prior to Fungizome, all the drugs, they employed liposomal technology, but they could not, reduce the toxicity to some extent. They were like, the amphotericin B has 66% nephrotoxicity, and the other liposomals, they have 27% nephrotoxicity. In Fungizome, as we studied the science and the effect, we reduced the toxicity to 0. So that is how we did some changes in the design, which reduced the toxicity to 0, increased the efficacy to more than 90%. The drug is available, the drug acts on various MDR strains, which the other drugs do not. We routinely, as far as safety is concerned, we routinely submit the PSUR reports to DCGI office. And as he asked that as we are the innovative molecule, but still we say that we are the affordable molecule when compared to the other generic drugs, because our dosage, we have reduced the dosage to, the others have dosage of 3 to 10, and we have reduced the dosage to 1 to 3 mg per kg per day. So because of that low dosage, the efficacy, because of the low dosage, the total treatment cost, the treatment therapy could be reduced to 4 to 10 times. So that is how the drug is affordable, that is how the drug is safe. And as far as generic medicines are confirmed, there is, we know that generic medicines are the backbone of Indian pharmaceutical industry, like forever, and they will be. But and there is nothing wrong in using generic medicines, there is nothing wrong in prescribing generic medicines, but there has to be certain evidence that there should be certain evidence for the drug, for the safety, efficacy and dose. And if that particular evidence is not present in the market, that is not present in the records, then that particular drug should not be given permission to manufacture.
So Ms. Khandelwal, can we conclude it like that, quality assurance comes at a premium and there is a scope for cost effectiveness analysis which can convince the prescriber that actually not the per unit cost but overall treatment cost is low. So it is a separate science. So we concluded that generics can be prescribed provided quality assurance is there, and quality assurance might come at a premium. So
plus some aggressive approach towards mass education and basically tilting the prescribers note towards generic is also something which is required. We fully agree that the generic drugs are wonderful drugs, but then they have to be in the mainstream more and more as the day passes. And as rightly said by Ms. Komal, that is still the backbone of the Indian pharmaceutical industries, because we are catering by and large quite poor patients in our terrain. Thank you.
So basically it can be concluded in 3 A's, that is assurance, quality assurance, availability, be it transportation, storage, procurement, all. And the most important, the third A is awareness. Awareness among the users, awareness among the prescribers, awareness among the end users, the patients. And to conclude and to end it on a lighter note, Mr. Mishra, कितने जनौषधि चिकित्सक अभी तक नवाजे जा चुके हैं?
कितने जनौषधि चिकित्सक?
There was a scheme to increase awareness and incentivize the prescribers.
Yeah.
They were titled as Janaushadhi Chikitsak.
Now this number is increasing. Exact number I can give you, but I see
It's being done. It's being done.
Yeah.
हो रहा है.
Per day number of turn up, it is more than 7 lakh per day. 7 lakh patients per day, they are taking medicines from Janaushadhi Kendras.
I'm talking about chikitsak.
Chikitsa.
Doctors.
Prescriber. Now I would say that in Chennai, I was in Chennai. One of the onco specialists, onco physicians, lady doctor, she had the Janaushadhi Kendras. I had been to Karnataka. One of the leading physicians, he was having Janaushadhi Kendra, and he said, अब तो साहब ये मेरे लिए सोने के अंडा देने वाली मुर्गी हो गई है. And मतलब this way how he is, and there is per day turn up more than 700 patients per day, and he had done lot of effort, yes, and slowly more and more doctors. I would say that recently when we opened that, yes, 2000 for opening the Kendras for 2000, so many of the requests we got from the doctors. So numbers are increasing, and slowly the people's confidence on generic part, this is increasing. Third thing is that yes, you will find that in coming days, even the more and more companies, by seeing our models, more and more companies is trying to replicate this model. They are also coming. But ultimately, see, whosoever is copying or whatsoever they are doing, ultimately they are trying to reduce the cost of the treatment. So ultimately we are in the business of healthcare. We are in the business of improvising human life. So ultimately something good is happening, and because of this initiative, now at least I know that is 5-6 names, because they personally came and met in office that yes, we are doing this. One of the entrepreneurs met in Dr. Bindra's program. He also said that just by getting the inspiration from Pradhan Mantri Janaushadhi Kendra, now we are also starting, and they also started doing sizable business. So likewise, things will improve.
Yeah, definitely. Thank you, Mr. Mishra, Dr. Ravi, and Ms. Komal. Thanks a lot. It was a very enlightening brainstorming session. Thanks a lot. We sign off.
May I now invite Professor Shailendra Handu and Professor Dishi Dashmana to kindly felicitate our moderators and panelists of this session. Thank you all the panelists and moderators. With the end of this panel discussion, we now proceed to the valedictory function of Sopicon 2023. May I now request Dr. Manisha Bisht to announce the poster competition winner of the Upasala Prize of Sopicon 2023. Ma'am, kindly be invited to the podium.
Thank you, Khushbu. Good evening, everyone. We have come to the last part of our conference, and it is the most awaited part. So before announcing the results, I would like to thank all the participants who have participated in the poster session. We are very thankful for the overwhelming response. We had nearly received 72 posters, out of which 36 were for online presentations and 36 were for offline presentation. And due to time constraints, we had to convert certain posters into free poster presentation. So basically out of 72 posters, 27 we had converted into free poster session. And after that, 45 remaining posters, competitive posters, we had asked them to present. Out of these, 26 posters were present online yesterday and 19 were present offline today. And each of both these sessions, they were judged by two judges, two different set of judges. And the criteria that we had used for marking the posters, which was a structured, structured marking system, and total marks was 50. And out of which 10 marks each was assigned to originality, material and methods, poster presenting style, impact and usefulness, and ability to handle question. So out of total score of 50, both the judges, they gave their marks, and we combined the marks of both the judges, and together with all the today's session and yesterday's session, we tabulate the result. And so the Uppsala Best Poster Award today, it goes to poster number P34, Dr. Manju Agarwal. It was a physical poster, I think it was presented today, so it goes to Dr. Manju Agarwal. And the Sopicon office bearer will soon contact the Best Poster Awardee, Dr. Manju, they will be contacting. Thank you everyone for your patience and overwhelming response. I now welcome Dr. Khushbu again.
I now invite Dr. Puneet Dhamija to the podium for valedictory remarks.
I want to take this opportunity to express my gratitude to all the members of organizing committee, our keynote speakers, moderators and participants, and our supporters, Karnataka Antibiotics and Pharmaceuticals Limited, PMBI, Life Care Innovations, Intas Pharmaceuticals, Zydus India Limited, Dr. Reddy's, Fresenius Kabi, for making this event possible. IT Cell and Balaji Telecom, they have provided the technical support, without which this event would not have been possibly conducted in such a smooth way, with minimum glitches. I would also like to thank the sanitation department, the security department and the photography department who have taken pains to manage the venue and cover this whole event. And the PRO cell, which is involved in press releases and media, conveying the message to the media. Most importantly, I would like to thank the residents, volunteers and the students of this institution for extending their support for this activity. Inspiring support, I would not like to forget that without the support from the co-organizing secretary, Ms. Manisha, Dr. Manisha Bisht, without the inspiration from Dr. Shailendra Handu, the head of department, this event would not have been possible. And it was the support from, it was actually a teamwork where everybody, every part worked like a team, and that made this event possible. So I would like to thank everybody, and we hope that we carry forward with this message of drug safety and affordability further in our careers and in our lives. And I would also like to thank Mr. Tapan Sharma for the support which he has provided as a representative of PMBI for us. Every time whenever we have any issues, we contact Tapan and he is ready to resolve the issues at any particular point of time. Thank you everyone. Thanks for being here and participating in this event. Thank you.
Thank you all delegates. Now we can proceed for tea, which is being served at level 2 of this auditorium.